students, welcome to Diksha Vedantu Foundation channel. So as we all know, your 10th midterm exams are approaching and I hope everybody is very busy in their preparations. So yes, to help you with your preparation, to make your preparation much more easier, we have come up with 100 most expected questions from science. So this includes your both chemistry, physics as well as biology. So now we'll be discussing 100 most important questions that you need to prepare so that similar questions you can expect in your board as well as in your midterm examinations. Okay, so what you have to do is you have to see these questions and then you have to pause the video, you have to try the answers and then you have to check the solutions in this video. Okay, so now I hope everyone is ready. So we have already done revisions of all these chapters, so now we should be able to solve these questions. Okay, so first I'll be covering the chemistry. Okay, after that biology questions will be covered and lastly physics questions will be covered in this video. Okay, so this is a comprehensive video of the complete science and you can expect like these 100 questions are all those that have been asked many times in the previous year, most important, most repeated questions. So do practice these questions. Okay, so yes, now let's get started without any delay. Okay, so first of all I'll tell you in this video what all things we are going to cover. So there will be very short answer questions. So there will be 15 MCQs including all the three PCM, physics, chemistry and biology. Then from very short answers, assertion and reason type. So students, generally we feel students find a lot of difficulties in this assertion and reason type questions. So do not worry, we are going to practice almost 15 questions from here also. Then short answer question 2 marks will be discussing 30 questions. Short answer questions 3 marks will be doing 32 questions. Case based questions 4 markers will be doing 12 questions and then long answer questions, that is 5 markers, will be doing 20 questions here. Okay, so you can see a lot of questions we have to practice today so that we can master each and every chapter, each and every topic and can score really well in our midterm examinations. Okay, so now let's start with chemistry. So in chemistry I'll be telling you the question numbers, how I have distributed them. So question number 1. Okay, so to make you practice with more questions itself, in question number 1 I have included 5 MCQs. Okay, so there will be total 5 MCQs in this question number 1. In question number 2 there will be 5 assertion and reason. Okay, similarly for all the 3 subjects. So you can see in MCQ you will get 5 questions from all the 3 subjects. Okay, next all the 3 sections. Okay, subject is science only but all the 3 sections. Next question number 3 to question number 12 will be short answer 2 marker questions. So there will be 10 questions in this. Okay, next question number 13 to 22, these will be short answer 3 marker questions. You will be having 10 questions here. Next question number 23 to 25 will be case based questions. We'll have 3 questions here and then question number 26 to 30 long answer questions. We will have 5 questions here. Okay, so now when I'm telling this MCQs 15, these have been included in one, like 3 questions total. This is 3 question. Okay, so now you could have understood this is 3 questions, this is 3 questions, this is 30, this is 32, this is 12 and then this is 20. Okay, so now if you see it is coming around 3, 3, 6, 7, 8, 9, 10. Okay, 100 questions will be there, right? So in each of this one question there are 5 MCQs. So we have done in this way so that we can give you more questions for practice. Okay, fine. So now coming to, I hope this is clear to you. Question numbers I have given you. Now let us start. Okay, so first, so I have made these 5 questions like question number 1, A, B, C, D, E. Okay, 5 MCQs. So the first question is from the chapter chemical reactions. So now if you see, Rima took 5 ml of lead nitrate solution in a beaker and added approximately 4 ml of potassium iodide solution to it. What would she observe? Okay, so now if you see what is taken, lead nitrate. Lead nitrate is taken with potassium iodide. So here what happens? Double displacement reaction happens. You need to pause the video and answer the questions. Okay, so now if there will be a double displacement reaction, what I'll get? PbI2, right? This is yellow in color. So what we will be able to see, a yellow precipitate is formed. So correct answer is option number B. Okay, next. Okay, which of the following is correct observation of the reaction showing in the above setup? So this is a setup given to you, which is the correct observation. So first option is given, brown color powder of magnesium oxide is formed. So if you see here a white color powder of magnesium oxide is formed, right? So that is not brown color, so you can eliminate this option. Colorless gas which turns lime's water milky is evolved. So this is which gas we are talking about? Carbon dioxide, right? And we know when we burn, there is no production of carbon dioxide, so this also wrong. Next, magnesium ribbons burns with a brilliant white light. So if you can see here, this area, it is a dazzling white light, okay? So that is why this option is your correct answer, okay? I hope all of you are answering with me, okay? So you have to pause the question, write the answer, and then you have to check with me, okay? Next, what is formed when zinc reacts with sodium hydroxide? Zinc reacts with sodium hydroxide, reaction of metal with bases. So now we have come to the questions of acids, bases and salts, right? So now if you see, it will give you sodium zincate, right? Sodium zincate. So what is it? Sodium zincate and hydrogen gas. So the correct answer for this question will be option number B, okay? Next, again one question from okay, next question is from metals and non-metals. Which of the following oxides of iron would be obtained on prolonged reaction of iron with steam? So we have remembered Fe does not react with cold water, hot water, it directly reacts with steam, right? So this will give you Fe3O4 plus H2, okay? So Fe3O4 will be formed, so correct answer is option number C. Next, what happens when a pellet of sodium is dropped in water? So you know sodium even reacts with highly reactive metal, it even reacts with cold water, right? So what will happen? It will catch fire, but does it form oxides? No. Sodium, potassium will form hydroxides, right? So it forms oxide, so this is also wrong, this is also wrong. It catches fire and forms hydroxide, right? Correct answer is option number C. So now let us see the next assertion reason type question. So this is from again acids, bases and salts. So the first one is the acid must always be added to water with constant stirring, okay? So it is saying that acid must be added to water. Now the mixing of an acid with water decreases the concentration of H plus ions per unit volume. Yes, this is correct. So when volume increases, concentration decreases, but yes. Now this reason is not explaining this assertion, right? The reason for this one is that when you add more heat is generated and water can absorb that heat, that is why this process is done. So there is nothing relation with concentration as a reason here. So that is why assertion is correct, but yes, reason is not the correct explanation for the assertion, okay? So correct answer will be option number B, okay? Next question, copper. So from again from acids, bases, copper sulfate crystals are wet because it contains water of crystallization. Water of crystallization is the fixed number of molecules water present in one formula unit of salt. Now see, again when copper sulfate contains water of crystallization, that is correct, but it is not wet, okay? It is a dry salt only. So that is why here assertion is false. So when assertion is false, we have option number D, okay? Because this is not wet, okay? Next one. So next we'll go to metals and non-metals. Hydrogen gas is not evolved when a metal reacts with nitric acid. Nitric acid is a strong oxidizing agent. So yes, this is correct. When metal reacts with nitric acid, okay, what we will get? We'll get salt plus H2, right? Now what happens? This nitric acid being a very strong oxidizing agent, it oxidizes this H2 to H2O. Right? So that is why we cannot find H2. Now here I'll tell you one more question. What are the two metals which can give you H2 with dilute HNO3? Comment down quickly, okay? So here the answer will be that assertion is also correct and reason is the correct explanation, right? So option number A. Next, question number 2 is zinc can easily displace copper on reacting with a solution of copper sulfate, okay? So yes, zinc, if it will have copper sulfate, so we know this reagent reaction, it is ZnSO4 plus copper, right? So now copper is more reactive metal as compared to zinc. Copper will be more reactive metal. See, we know that this reverse reaction is not possible. Now zinc is able to displace copper, means zinc is more reactive than copper, okay? So that is why assertion is true but reason is false. So that is your option number C, okay? So like this you can practice much more assertion reason questions as well. Okay, now we will go to second, that is type 3 questions, where we will be discussing short answer questions, 2 markers. Okay, so the first topic is again from chemical reactions. So what happens when steam is passed through red hot iron? So you have to write the reaction. First one, when steam is passed through red hot iron, okay, so that will give you Fe3O4 plus H2, right? So there is oxide of iron is formed, Fe3O4 plus H2. Okay, that is the first thing. Write the equation, this will give you 1 mark. Next, is hitting iron to red hot a physical or chemical change? So this is a physical change. When you are hitting, physical change. Why? Because no chemical okay, so you can write down that no new substance is formed here. Okay, no new substance has formed. No new substance is formed. Okay, so you can write down this much, that no new substance is formed for the answer. Clear? Okay, next coming to question number 4. Okay, what is observed when a solution of potassium iodide is added to a solution of lead nitrate taken in a test tube? So first you have to write what is observed. So first of all you need to write what is observed, that a yellow precipitate is formed. What you are observing, it is asked. Okay, yellow precipitate is observed. Now then you need to write why it is observed. So that is, you can write due to the formation of PbI2. So what is the full form, what is the full reaction here? So we have Ki plus lead nitrate, right? So that will give you lead iodide plus KNO3, right? So we will get this one as yellow precipitate. Next, what type of reaction is this? So what is this type of reaction? This is your double displacement type. Okay, so answer should be simple, double displacement. No need to define it, no need to write an example. Whatever the question is asked, only write that much. What type of reaction? It is a double displacement reaction. Okay, so you will get 2 marks here. First 1 mark here and 1 mark here. Okay, next, why is respiration an exothermic reaction? So first point you need to write what is an exothermic reaction. Definition of exothermic reaction, first point you need to write. What you will write here? That whenever any reaction heat is released, that is called as exothermic reaction. Next, you have to write the respiration reaction. In respiration reaction, what happens? Glucose, right, reacts with oxygen to give carbon dioxide plus water, right? And here you need to write heat. So as in respiration, heat is produced. As in respiration, heat is produced. It is an exothermic reaction. It is an exothermic reaction. Okay, so here you will get, see how will be the marking distribution. If you write this one here, this one here, will get 2 marks. Okay, so 1 second. Okay, now we can go to acids, bases and salts. That is your next chapter, 2 marker question. Why does an aqueous solution of acid conducts electricity? So why? First point you have to write, when conduction of electricity, who can conduct electricity? So you have to write free ions. Free electrons or ions can conduct electricity. Now second thing you need to write, that when acid is dissolved in water, when acid is dissolved in water, okay, it produces ions, okay, which can conduct electricity. Which can conduct electricity. Okay, so this one you can write 1 mark and this is 1 mark. Okay, next, why diluting an acid why is it recommended that acid should be added to water and not water to the acid. So first of all you can write why acid should be added to water. Okay, first of all write acid plus water is an exothermic reaction. Okay, next you will write that if acid is added to water, heat generated or heat liberated is accepted by, is absorbed, heat liberated is absorbed by water, right? Then second point you write if acid is added to water, okay, a huge amount of heat is generated, a huge amount of heat is liberated, and here there is nothing to absorb this heat, right? There is only acid and you added water, so there is no water to absorb this heat and it can lead to explosion. Okay, clear? This will give you 1 mark. Why acid should be added, why water should not be added. Okay, 2 points, 2 marks. Next, what happens when nitric acid is added to eggshell? So here also you know eggshell formula is what? Calcium carbonate. Okay, nitric acid, HNO3. So when acids react with carbonates, what we get? We get salts, we get CO2 and we get H2O, right? So we'll get calcium nitrate plus CO2 plus H2O. So now what is happening? This calcium carbonate is reacting with this one. So that means it is dissolving. So we can write that calcium carbonate dissolves. Okay, so reaction 1 mark, this statement 1 mark, total you can get 2 marks. Okay. Next, in nature, aluminium is found in the form of compounds while gold is found in the free state. Give reason. So now we have come to metals and non-metals type questions. Okay, 2 markers. So you can write aluminium first of all is reactive, right? Because it is reactive, it reacts with all other elements and is formed in the compound state. Next you can write gold is a non-reactive metal and it does not react with any metal and it stays in the free state. Okay, so here you write 1 for aluminium and 1 for gold. Okay. Next, explain why calcium metal after reacting with water starts floating on its surface. Write the chemical equation for this reaction. Okay, so first point you write down here, when calcium reacts with when explain why calcium metal after reacting with water. When calcium reacts with water, it forms calcium hydroxide and hydrogen gas. Okay, this hydrogen gas bubbles comes out where calcium sticks. Okay. So that is why it is visualized, or it seems that calcium is floating. Now if you just need to write the reaction, so whenever it is asked to write the reaction, you have to write the whole reaction, means the balanced reaction you have to write, right? So calcium plus water will give you calcium hydroxide plus H2. Okay, so if you want to balance, we can balance it in this way. 2, 4 hydrogen, 2 oxygen. Okay, balanced one. Next question. Sorry, ionic compounds conduct electricity in molten state, not in the solid state. Why? So again here first statement is conduction of electricity is due to free ions. Right, free ions can conduct electricity. Second point what you need to write, that why molten state? So in molten state there are molten state, in molten state or in the aqueous state. Okay, or in the molten state or in the aqueous state there is free ions, right, and that is why they conduct electricity. Okay, so this one will give you 1 mark, this one will give you 1 mark. Okay, next question, what happens to potassium and sodium, if they are kept in open, why are they immersed? Okay, here there is a question mark. Next, why are they kept immersed in kerosene? The first point you can write, sodium and potassium are highly reactive, right, are highly reactive. So when they come in contact with moisture, they catch fire, right? So when they come in contact of highly reactive with water, okay, when they come in contact of moisture, right, they catches fire, or they catch fire. Okay, that is the first thing. Now why it is stored in kerosene and oil? It is stored in kerosene to prevent their contact, from air. Okay, if they do not come in contact to air, then they won't catch fire and there will be no explosion. Okay, so with this we are done with the 2 marker questions. Now we'll go to 3 marker questions. Okay, so short type 3 marker. First we'll start chemical reaction. Mark the chemical reaction is a redox reaction. Justify your answer. So in redox reaction you have to tell which one is undergoing oxidation and which one is undergoing reduction. Okay, so first you can see here MnO2 to MnCl2. Here what has happened? Oxygen has been removed from manganese. So removal of oxygen is what? Reduction, right? So here it is reduction. Now if you see HCl to Cl2, from HCl to Cl2 hydrogen is removed. Removal of hydrogen is oxidation. So from here to here is oxidation. Okay, so you can write down that first point, you can write the definition of oxidation and reduction. Second point, you can write down MnO2 to MnCl2, oxygen is removed, so it is reduction. And next one you can write HCl to Cl2, hydrogen is removed, so it is oxidation, and so it is reduction. 3 markers, this will give you 1 mark, 1 mark and then 1 mark. Okay, fine. Next, identify the oxidizing agent among the following. Now what is oxidizing agent? So see, the species which undergoes reduction, okay, it behaves as an oxidizing agent. Species which undergoes reduction behaves as an oxidizing agent. So we have to find here which species is undergoing reduction. Now if you see Fe to Fe3O4, oxygen is added, that means it is undergoing oxidation, but we need to find the species which is undergoing reduction. If you see H2O to H2, right, so here oxygen has been removed, so removal of oxygen is again reduction here. So which is undergoing reduction? H2O. So which is the oxidizing agent? H2O. So the first one, H2O. Okay, second one, if you see copper oxide to copper, oxygen is removed. Oxygen is removed means it is undergoing reduction. If it is undergoing reduction, it will behave as an oxidizing agent. So here the correct answer is copper oxide. Now next, V2O5. So if you see V2O5 to V, oxygen has been removed. Oxygen removed means it is reduction. Reduction means it is oxidizing agent. Okay, so here it is V2O5. Okay, next question. Next we'll go to, okay, next question. Name the type of chemical reactions presented by the following equation. Okay, you have to tell me the type of chemical reaction. Okay, if you see first one, 2 things are combining to give you 1 compound. So this is what? Combination reaction, right? Second one, if you see BaCl2, Al2SO4 whole 3. So then Al, so this one has come here and this one has come here. This is double displacement reaction, right? Next, FeSO4, Fe2O3, SO. So one compound gives you more than 1 product, so that is decomposition reaction. Clear? Very simple question, just you need to write, you can easily get 3 marks. Next one, question number 16. A milkman adds a very small amount of, so now we have done 3 markers, 3 questions from chemical reactions. Now we'll go to the next chapter, that is acids, bases and salts. Now, a milkman adds a very small amount of baking soda to fresh milk. Why does he shift the pH of the fresh milk from 6 to slightly alkaline? Okay, so this is done to prevent curding of milk. Okay, the milk does not get converted to curd if you add a slight amount of baking soda. Okay, now what is the reason for that? Why does this milk take a long time to set as curd? Now see, curd is produced, or curd formation takes place due to the production of an acid called as lactic acid. Okay, so now what this baking soda will do, okay, this baking soda, okay, neutralizes the lactic acid, right? Baking soda neutralizes the lactic acid, so now there is no lactic acid to form the curd, right? That is why the formation of curd is little delayed here. Okay, so next one, name the acid present in ant sting. So that is which acid? Formic acid. Okay, and write the chemical. So first you need to name it. So A will be formic acid. Okay, then B, name formula HCOH, and then also give the common method. So common method is applying some baking soda. Why? Because this one is basic in nature, right, and this one is acidic in nature, so that can be neutralized. Okay, fine. Next question. You have two solutions, that is your A and B. So we have two solutions A and B of pH of 6 and 8. Okay, which solution has more hydrogen ion concentration? So if we know that less pH, that means more acidic. Okay, more acidic, that means more H plus ion. Okay, so you can see pH is 6 here, so that means here less pH, so that means it is more acidic. Which solution has more hydrogen ion concentration? I can write A. Okay, then which is acidic and which is basic? So A is acidic, okay, and B is basic. You know, pH above 7 are called as base, pH below 6 are called as acid. Okay, very simple question this one. Okay, next, metals and non-metals will go. 3-4 questions will do from here. So give reason. Platinum, gold and silver are used to make jewelry. So first one, the reason is they are non-reactive. Okay, because they are non-reactive, they do not get any like oxidized, or there is no reaction of these elements, and they can be used for long period of time. Okay, next one, sodium and potassium and lithium are stored under aluminium. So you can under oil. So you can write because they are highly reactive. They are highly reactive, right, and catches fire. Okay, next, C. Aluminium is a highly reactive metal, yet it is used to make utensils for cooking. So the reason for this is it is a good conductor of heat, because it is a good conductor of heat. Okay, clear? So 3 marks you can easily get here. Okay, next, name the following. A non-metal that is lustrous. So a non-metal that is lustrous is iodine, that is there. Then a metal that is liquid at room temperature is mercury. Okay, and then an allotrope of carbon that conducts electricity is your graphite. Right, just you need to write 3 words, easily you will get 3 marks. Okay, next one. So a metal form M forms an oxide having the molecular formula of M2O3. It is dissolved both in dilute sulfuric acid and in sodium hydroxide solution. Identify the metal and write the equations for the following reactions involved. So if you 3 M2O3, okay, so oxygen valence is 2 minus, so this must be M plus 3. So when they crisscrossed, we got M2O3, right? So what is this metal with which is reacts with both acid and base, means it is amphoteric. So which is the metal which is having plus 3 state and it is also amphoteric in nature? So that is your Al2O3. Okay. So what is our metal? The metal is aluminium. Okay. Now the reaction with any that is sulfuric acid. So H2SO4. Okay. So now this is a acid. Okay. Right. Okay. So we need to write Al2O3 reaction. Okay. So now see, this is an acid, that means this will behave as a base. Okay. So what will happen? Salt and water will form. So we will get aluminium sulfate, that is Al2SO4 whole 3, okay, and water. Okay. Next, with the dilute NaOH is also it is there, plus NaOH, right. So if this is base, this will behave as an acid, right. So sodium, first salt will form. So the salt formed here is called as sodium aluminate. Okay, NaAlO2 plus water. okay, must balance the reaction. So if you see 2 aluminum is there, 2 hydrogen is there, oxygen 7 is there. So here we have 12 oxygen, okay, sulfur is 3. So I will make it 3 sulfur, then we'll get 3 hydrogen is formed. Okay. So 6 hydrogen, so we can make this also 3. Now if we balance, we have this side oxygen, we made it more, right? No, correct. So 2 aluminum, oxygen 3, and here it is 12 plus 3, 15. Here it is 12 plus 3, 15. Here it is 6 hydrogen and then 6 hydrogen, and then here we have 3 sulfate groups and then 3 sulfate groups. Okay, this is balanced. Next, if you see here Al2O3, Al2 is there, so I can make it 2 Al, and then 2 sodium, so I can make it 2 sodium. Now let us see, Al2 is there, 2 aluminum is there, oxygen is 3 plus 2, 5. Here we have 2 plus 2, 4 plus 1, 5, 2 sodium, and 2 hydrogen and 2 hydrogen. Okay, so this reaction is also balanced. Okay, so if you write first one, second one and third one, total you will get 3 marks easily. Okay, next, in the electrolytic refining of metal, what did you take as anode, cathode and electrolyte? So anode, impure M should be taken as anode, okay, pure M should be taken as cathode, okay, and salt of M should be taken as electrolyte. Okay, next. Okay, so this one is done with 3 marker questions. Now we will go to 4 marker questions. Okay. Okay, so these are all your case based questions. So oxidation is the process of gaining of oxygen, losing of hydrogen. Reduction is the process of losing of oxygen or gaining of hydrogen. The substance which undergoes oxidation is the reducing agent, while the substance which undergoes reduction is known as the oxidizing agent. Oxidation and reduction always takes place together, and these type of reactions are known as redox reactions. Some of the examples of redox reactions are given below. Okay, you can see some of the reactions of redox reactions. Next, give 2 examples. So now we will see this case based questions here. So give 2 examples of oxidation from your everyday life. So what are the 2 examples of oxidation from everyday life? You can say one is your rusting, okay, and another is rancidity. That is your oxidation of food oil particles. Write the oxidizing agent in the reaction of 3 and 6. Okay, let us see. 3 and 6, we need oxidizing agent. Oxidizing agent means it should undergo reduction. Okay, so now if you see here 3 in the reaction 3, one second, yeah, so in this third reaction that is given to us, which is undergoing reduction, copper is undergoing reduction, right? Why? Because you can see copper charge here is +2 and copper charge here is 0, right? Since the oxidation number is decreasing, copper is undergoing reduction. If copper is undergoing reduction, then it will behave as an oxidizing agent. Okay, so I can write here in 3, which is the oxidizing agent, copper. Now coming to 6th one. Okay, 6th one, if you see that which is undergoing reduction, copper oxide is there, okay, and copper is there. So from here to here oxygen is removed, so that is why it is removal of oxygen is reduction. If reduction, that means it is an oxidizing agent. So what is the answer for the 6th one? In the 6th reaction, this one is copper oxide. Okay, next, which of the following is an oxidizing agent? So you should know that alkaline KMnO4 and K2Cr2 are very strong oxidizing agents. So the correct answer for this question will be both B and C. Okay, so here the answer is both B and C. Okay, okay. Next, out of oxidation and reduction, which reaction takes place at anode? So always remember, anode is always oxidation. Okay, at anode there is always oxidation, at cathode it is always reduction. Okay, fine. Next, go to the next question. Acids, bases and salts. Okay, the solutions of glucose, alcohols, hydrochloric acid, sulfuric acid, etc. And fix 2 nails on a cork and place the cork in a 100 ml beaker. Connect the nails of the two terminals of a 6-volt battery through a bulb and a switch as shown in the figure. So figure we can draw like this. Okay, you have to take different solutions. We have two nails here, okay, and these nails are connected to battery, okay. Now, and it is also connected to a bulb, okay. So we can make it like this, okay. So this is bulb. Now pour some dilute HCl in the beaker and switch on the current. Repeat with dilute sulfuric acid. What do you observe? Okay. So now if you repeat with, first of all, when you observe, when you put HCl, the bulb will glow. You will observe that bulb glows. Second case also, bulb will glow. When you take H2SO4, there also bulb glows, okay. Next, repeat the experiment separately with glucose and alcohol solution, okay. So if you see alcohol solution and glucose, third one, okay, the bulb will not glow here. Bulb will not glow. What do you observe now? That is your bulb will not glow. Does the bulb glow in all cases? So the bulb glows only in the first and second, and not only in the first and second cases, okay. Now what is the reason for this? See, when you take HCl and H2SO4, these acids can ionize, right? So when they form ions, they can conduct electricity. If they conduct electricity, there will be glow of bulb. Now if you take glucose and alcohol solutions, these do not produce ions. Because they do not produce ions, they cannot conduct the electricity. If they cannot conduct the electricity, the bulb will not glow, okay. Very simple answer, okay. Next, okay, so here, okay, we need to answer these questions, sorry, okay. What was the changes occur in the case of acids, that is HCl and H2SO4? So you can see that when you add HCl, there was a small, the bulb will glow, but when you add H2SO4, more ions are produced, so the bulb will glow brighter, okay, bulb will glow brighter here. Next, why do glucose and alcohols do not conduct electricity? Because they cannot produce ions, okay, cannot produce ions. Next, why do acids do not show acidic behavior in the absence of water? Again same thing, they cannot produce ions. They can produce ions only, they cannot produce H plus, okay. So they can produce H plus only in the presence of water, okay. So because water is not there, there cannot be any, they cannot produce H plus. So if water is not there, they cannot produce H plus. If H plus is not produced, then we cannot call it as an acid, okay. Next, does rainwater or distilled water will conduct electricity? So you can write rainwater will conduct electricity. Why? Because it has a lot of ions in it, okay. Fine. Now let us go to the next case based question from metals and non-metals. A metal X combines with a non-metal Y by the transfer of electrons to form a compound Z. State the type of bond. So what is this type of bond? This is ionic, right? Transfer of bond is ionic bond. What can you say about the melting point and boiling point? So the melting point and boiling point of ionic compounds is very high. What will this compound dissolve in kerosene or petrol? No, right? They will dissolve in water because they are ionic compounds, but kerosene and petrol are non-polar solvents, so they will not dissolve in this. Will this compound be a good conductor of electricity? So you have to write yes, but in molten or aqueous state, okay, fine. Now let us go to the next question. So now we'll go to the 5 markers questions, okay. So the first one is from your chemical reactions here. Okay, so reaction is given, which substance is getting oxidized. So what is oxidized? Addition of oxygen, right? Addition of oxygen is oxidation. So you can see H2 to H2O, there is addition of oxygen. So that is why H2 is getting oxidized. Which substance is getting reduced? Removal of oxygen. Copper oxide to copper oxygen has been removed. So copper oxide is undergoing reduced. Now which is the oxidizing agent? Oxidizing agent is that one which undergoes reduction, right? So reduction is undergone by copper oxide, that is an oxidizing agent. Which is the reducing agent? So reducing agent is that which undergoes oxidation, right? So here H2 is undergoing oxidation, so this is H2. What type of reaction is this? Both oxidation and reduction is happening, so it is redox reaction. Okay, very simple question. Next one. A compound X is bitter in taste. It is a component. So now we'll go to acids, bases and salts. Okay, a compound X is bitter in taste. It is a component of washing powder and it reacts with dilute HCl to give brisk effervescence due to the colorless odorless gas Y. So if you see washing powder, what is the component? Na2CO3. Okay, okay. So this has to be X. Then it reacts with HCl to give what? Salt, that is NaCl plus CO2 plus water, right? Now this CO2 is what? Brisk effervescence, colorless odorless gas. So now this is your gas Y, okay, which turns the this one turns lime water milky due to the formation of Z. So what is this carbon? When you take calcium hydroxide, that is your lime water, and treat it with carbon dioxide, it converts it to calcium carbonate. Okay, this is your this is why it looks milky in color, right? So due to the formation of Z, so Z is formed there. Now when excess of carbon dioxide is passed, milkiness disappears due to the formation of P. So if you see, now we have calcium carbonate plus we have CO2 plus water. Okay, now when you pass this, we get calcium bicarbonate. Okay, this calcium bicarbonate is P, which is a soluble compound. So that is why we cannot see the milkiness disappears, because the precipitate itself gets dissolved. Okay, so the correct answer we will get here X, Y, Z and P. Okay, next question. What will be the action of the following substance on the litmus paper? So first one, if you see dry HCl, on dry HCl there is no change, because we told for acidic behavior we need water, right? Next, moistened NH4 gas. NH4 means it is a base, right? So if you remember, what does acid do? Acid changes blue litmus to red. And what do base do? Base changes red litmus to blue. So when you take ammonia in a moistened ammonia gas, it will behave as a base and it will turn red litmus to blue. Okay, next, lemon juice. If you take lemon juice, it is an acid, so it will convert blue litmus to red. Okay, next we have carbonated soft drinks. So carbonated soft drinks, again these are your acids, so acids will convert again blue litmus to red. Okay, and last one, okay, last one is a curd. Curd is also there, so curd is also acid, lactic acid, it will also convert blue litmus to red. Okay, so I'll write here what is your C, lemon juice. Next we have carbonated drinks, okay, and then we have curd, and the last one is soap. Okay, so soap is a base, it converts red litmus to blue. Okay, fine. Now next one, an element E. So now we'll go to metals and non-metals. An element E combines with the oxygen to form an oxide E2O, which is a good conductor of electricity. Give the following information. How many electrons will be present in the valence shell of the element? So if you see E2O, okay, so if you look at the formula of E2O, then oxygen is in -2 state, right, and this is in +1 state. So that means there this has lost 1 electron only, right? So in the valence shell there will be only 1 electron. Okay, write the formula of the compound formed when the element combines with chlorine. So E is in +1 state, Cl is in -1 state, we will get ECl, right? What will be the formula here? ECl. Next, now see, magnesium, atomic number of magnesium is 12, right? So we can say that it will have 2, 2, 8 and 2. So it has 2 valence electron, and oxygen has 6 electron. So what does the magnesium do? It gives 2 electrons to oxygen. So now if I see what, I'll get magnesium 2 plus and I'll get oxygen 2 minus, right, because it is gaining 2 electrons. Now if they two combine, we will get MgO, right? Next, name 2 metals which give dilute hydrogen gas with dilute and HNO3, that is your magnesium and manganese. Okay, please remember these 2 elements, very important. Okay, next, now let's see the last question. Okay, the last question here, that is explain the following. You have 5 situations have been given, you have to explain them. Okay, so now reactivity of aluminium decreases if it is dipped in HNO3. So you know HNO3 is a very strong oxidizing agent. So in presence of HNO3, aluminium gets converted to aluminium oxide, right? So now because it is not a pure metal, now its reactivity decreases. Okay, very simple. Next one. Carbon cannot reduce the oxides of sodium and magnesium. So you can write down sodium and magnesium. What is these metals are highly reactive. Okay, these metals are highly reactive, right? Now if they are highly reactive, they have more affinity for oxygen. Okay, they have more affinity for oxygen. Okay, so now the carbon cannot reduce this because they have more affinity for oxygen. Okay, next coming to NaCl is not a conductor of electricity in the solid state, whereas it does conduct electricity in aqueous solution as well as in molten state. So this you know, NaCl when it is in aqueous state, it can produce ions, and these ions are responsible for conducting electricity. Okay, next, iron particles are galvanized. So if you know, iron gets easily rust, right, or corrosion. So to prevent that, to prevent rusting of iron, to prevent that, what we do? Galvanization. Okay. Next, metals like sodium, potassium, calcium and magnesium are never found in their free state. So that is again same thing, because these elements are highly reactive. So they react with oxygen, carbonates, sulfur, and that is why, or any other nitrates, and that is why we cannot find them in the native state. Okay. So yes students, now we have done 30 questions from chemistry, including all the 3 chapters. I hope you would have revised all these concepts and written the answers. Okay, from here now onwards we will start biology. So biology ma'am will take up this class now. Thank you, and please prepare all these questions thoroughly. Okay, thank you, and I'll hand over this class now to bio ma'am. Okay.
Hello my dear chitkus, now it's time for biology. Who all is excited? Well, yes, I hope that you have completed all the 30 questions of chemistry, because Aishwarya Ma'am explained it really well. And now it's time for biology, as you know. And instead of 30 questions, we would be solving 40 questions. Why? Because biology has more weightage for your boards, and also one more thing, because we have more number of chapters as well. And here I am, my name is Jasmine Joy. So let's begin. Let's see the number of questions, children. You have 5 MCQs, 5 assertion and reason, 10 short answer, that is 2-marker question, 12 3-marker question, 6 case study based question, that will be of 4 marks, and then 10 long answer question, that will be of 5 marks. So let's begin. Over here, your first question is from this chapter, that is control and coordination. Select a pair of plant hormones from the following. You have to look for the plant hormones, which is mentioned over here. Okay, the first one is cytokinin and insulin, children. Cytokinin is a plant hormone, but what about insulin, children? Is it a plant hormone? Do you think it's a plant hormone? I don't think it's a plant hormone. It's our hormone. It's human hormone. Okay. Now, gibberellin again is a plant hormone. But what about adrenaline? Adrenaline is not a plant hormone. Now, going ahead, abscisic acid is a plant hormone. What about adrenaline? Again, it's not. Auxin and abscisic acid, both of them, they are plant hormones. Now, since we are talking about plant hormones, I have to talk about what are the different type of plant hormones. So, children, there are 2 different types. Okay. One is the growth promoting ones, okay, and the other ones, they are growth inhibiting ones. Okay, so let's team them up. Growth promoting ones, there are 3 of them. One is auxin, okay, the other one is gibberellin, and the third one is cytokinin. Now, auxin is responsible for something that is known as cell elongation. So, when a cell is elongated, for example, if this is like this and now it is elongated to something like this, obviously it will lead to growth of the plant also. Okay, because plant will need more space now, so it will lead to growth. So, auxin is a plant growth promoting hormone. Talking about gibberellin, it helps in germination. It helps in germination of the seed. So, you know there is a seed which is in a dormant state. Now, when it breaks the dormancy because of gibberellin, and now this gibberellin allows the seed to grow into a small sapling or a plant. Okay. Now talking about cytokinin, as the name suggests, cyto as in cell, kinin as in break. So basically it helps in cell division. Now obviously, if there is one cell and this cell divides into two, we would be needing more space. So it will lead to cell growth, or basically the plant growth in general, right? So they are all growth promoting hormones. Talking about growth inhibiting hormones, you have two of them. One is abscisic acid, abscisic acid, and then the other one is ethylene. Okay. What is the use of abscisic acid, children? They are there for any for example, any bad condition. Just imagine your best friend. Okay, so basically when the plant is undergoing something really bad, it's not having some favorable situations, for example, less water is there, things like that happens. So at this point of time, we do not want the plant to grow, because the plant will die if it tries to grow, because it does not have all the things to grow, right? So instead of that, the plant is said that you have to come in this particular stage, which is known as dormant state. Okay, it is known as dormant state, where the plant is not doing anything, is just there, and now when the right time arises, it can use its gibberellin to germinate. Okay, so abscisic acid does the opposite work of gibberellin. It does not allow germinations to happen. Instead, it says that seed you have to be, or the plant basically, you have to be in a dormant state. And also abscisic acid is also known as what, children? It's a stress hormone. So whenever there's plant experiences stress, no, just like us, so they also release this hormone that is known as abscisic acid, which allows it to thrive to this stressful period. Okay. Now talking about ethylene, ethylene is a gaseous hormone. Okay. For example, you have, when you keep a ripe banana, instead in between all the raw bananas, the other ones also become ripened. Why? Because ethylene is being released. It is gaseous. It is gaseous, and it is majorly responsible for fruit ripening. Okay, so both of them, they are growth inhibiting. Three of them, they are what, children? They are growth promoting. Please remember this. Shall we move on to the next one? Okay, we shall move on to the next one over here. I hope you got the answer. Now, okay, now it won't go because, okay. The part of the human brain which controls muscular coordination is what? Medulla, pons, cerebrum or cerebellum? You tell me, medulla, pons, cerebrum or cerebellum. So basically, I'll tell you one thing. Medulla and pons, no, both of them, they are responsible for the breathing activity, the respiratory activity, for the heart rate and everything. Okay, they are responsible for all the cardiovascular activities, that is the heart rate, breathing rate and everything. Okay, especially the medulla. Okay. Now, apart from this, they are also responsible for all the involuntary action. For example, salivation. I see a food and saliva comes in. This is involuntary. All of them happens because of medulla. Okay. Now, talking about cerebrum, it's the largest part of our brain. Cerebrum is the largest part of our brain, and it is involved in thinking, memory and all of this thing. For example, as of now, when you are studying, your cerebrum is the most active part of your brain. Okay. Now, talking about cerebellum. Cerebellum is responsible for maintenance of posture, okay, the muscle coordination and everything. Balance. So you might have seen people who drink, no, they do not have balance, they walk like this. Why? Because the cerebellum is getting affected. And how can you always remember this thing? I always say there is double L over here, which looks like posture, straight thing. Right? So in that way you can remember like two legs. But please remember, medulla also has this and cerebellum also has this. Okay, but how can you differentiate? Medulla might be in somewhere middle, so it is going to affect some middle part of your body, that is your heart. You can remember like that. Okay, medulla is not in the middle, obviously it's over here, it's towards the lower side of the brain. Okay, so that is about the human brain, and the muscular coordination is done by, children, cerebellum. Okay. Again, this is of control and coordination. Going ahead, what do we have? Heredity. Okay. In a cross between two tall pea plants, a few dwarf pea plants were also obtained in F1 generation. It is possible only if the gene combination of the parental plants is. So they are saying, so there is F1 generation. Okay, there is F1 generation and there is F2 generation. F1 as in your parents basically when they fuse, whatever kids are being formed, they come under F1 generation. Now when we undergo sexual reproduction, whatever kids we produce will be coming in F2 generation. So in F1 generation, they are saying in a cross between two tall pea plants. Okay, so there is they are saying tall pea plants are crossed. Okay, that is the question. Now, but a few dwarf pea plants were observed in F1. In F1, dwarf were present. Now, dwarf means the allelic combination should be this. Only if this happens, then only dwarf combination can come in. Okay, because it is a recessive trait, and recessive trait are only observed when there are two alleles of the same kind, both of them which are recessive. If dominant trait, it comes like this. This capital T stands for tall. Okay, if dominant allele is there with the recessive allele, the dominant allele will show over it, and this plant will be what, children? Tall. Okay, so now over here, we want this particular thing to happen, then only dwarf product can be produced in the F1 generation. So they are asking what will be the parental combination. Capital T, capital T, capital T, small t. Chalo, let's see, we have to solve each of them. Capital T, capital T, capital T, small t. So over here, this T can come. Over here, this T can come. Here this T, here this T. Now whatever you do, if you cross this and this, you get capital T, capital T. So this will be tall. If you cross this and this, capital T, small t. Again there is a capital T over here, so it will overshadow it. Okay. Now if you cross this, whatever you do, you won't be getting dwarf one. But if you see over here, if one parent among them has this dwarf thing, okay, the allele for dwarfness, then probably things could work. So let's now try for this and this. Here and here anyways it won't work, because in one parent's end we have capital T, so it will overshadow for any such cases. Okay, so let's try this. So we have capital T, small t, and capital T, small t. Okay, just a second. Yeah, small t and small t. Okay, that's a parental combination. Now parent can give this gene in the gametes, so it will be capital T, or it can give this gene, so it will be small t. This is a gamete, okay. Now again, can be small t in this gamete, can be small t in this gamete. These are the chances that what all parents can give. So these are the gametes, children. These are the gametes. Now they will fuse. So over here, if I fuse this with this, I get capital T, small t. Here capital T is there to overshadow small t, so it will be tall. Now, again capital T, small t, capital T is there, so it will be again tall. But now, small t, is there anyone to overshadow? No, it will be dwarf. Small t, again there won't be anyone to overshadow, it will be dwarf. So you got dwarf as well. So this could be one parental combination, okay, that the parent could be capital T, small t, and the other parent will be small t. Now let's look for this also. Can we look for this? Because since you could have a confusion, let's look for that also, okay. Now, I am making in green. So, there is capital T, small t, and there is capital T, small t. This is the parent. You have to write your answers like this whenever you are looking for genetics, okay. Now, the gametes that will be formed will be, so the gametes can have either this, that is capital T, or this, that is small t. Similarly, over here, capital T and small t. Now, when they fuse, it's capital T, it has to be tall. Capital T, small t, capital T is there, so it has to be tall only. Okay. Now capital T, small t, again it will be tall. Always try the dominant trait over here. Okay, on this particular side. Now small t. But over here, if you see, basically there is only one plant which will be dwarf. Over here, there will be two plants which will be dwarf. So over there, what is the thing? In a cross between two tall pea plants, okay. Two tall pea plants, a few dwarf pea plant were also obtained in F1 generation. It is possible only if the gene combination of the parental plants is what? Is it this or this? Now you are saying here also dwarf plants are produced and here also dwarf plant is produced. So which one is the correct answer, children? Will you look for the number of dwarf children that are being produced over there? No. They are giving you a condition. It's a cross between two tall pea plants. Is it tall? Is it a tall pea plant? No, it's a dwarf pea plant. Whereas this is tall. So this parental combination is not possible. We need two tall pea plants. But over here if you see, this is also tall and this is also tall. Right? So the correct answer will be, children, this one. This is how things can be made to be very confusing for you guys. So if you want the explanation, this is the explanation that I gave over here. Here we are fusing two tall pea plants, whereas there we are using one tall pea plant and the other one is dwarf. Okay? So the correct answer will be what? This thing. Going ahead. Okay, the opening and closing of stomatal pore in the leaves of a plant is due to. Why does the stomatal pore, which is this, open and close? What are these cells that we have over here? Guard cells. Okay. Now, diffusion of carbon dioxide in and out of the guard cell? No, that is not the case. Movement of water in and out of the guard cell? Let's keep it on hold. High pressure of gases inside the guard cell? No. Stimulus of light in the guard cell? No. The answer is this. Movement of water in and out of the guard cells. The opening and closing is because of that. I'll tell you why. Now, over here, when water enters the cell, at this point of time, it will get swollen. Okay, the guard cells basically get swollen. Now you only imagine, if something is like this, you are basically, making a balloon to be big. So at this point of time, when you're passing on air, it will go big and it will curve itself, right? Similarly over here, some balloons which were like this, now it will curve and it will be like this when they are swollen. Do you see a space in between? Yes, the space, right? What is the space? This is a stomatal pore. So this is the opening of the stomata. It's because water enters. So at this point of time, the cells are swollen and the stomatal pore basically opens. On the other hand, if you think, the water moves out. So now, something was like this, the water moved out, okay, we removed the air from the balloon, what will happen? It will slowly go back to the same state. At this point of time, stomata closes off. So basically, during shrunken state, it closes. I hope you got it. Instead of swollen, we also say turgid state. It is much better for plant cells. Okay, so the correct answer will be this, children. Going ahead, what do we have? Okay. Last part of the MCQ, okay? An organism which breaks down the food material outside the body and then absorb it is. Chalo. When you are given this condition, I already have to think what is it? What are those things which breaks down food outside the body? It has to be either a fungus. It has to be either a fungus or a bacteria. Because only both of them, they are what, children? Saprophytes. And saprophytes are the ones that feed on dead and decaying things. And for dead and decaying things, basically if it's a mushroom, it releases enzymes outside. These enzymes basically break down the food into smaller particles. And whatever nutrients are there, it's been taken in. They are not taken. Have you seen ever the mushroom with its body is taking the food and eating it and chewing it and having it? No, it does not work like that. You never know what's happening because they are releasing enzymes outside. So let's see what suits the best. A plant parasite cuscuta? No, cuscuta takes in things. It's a parasite, so it's not the answer. Going ahead. An animal parasite tapeworm? Again, no, it takes inside the food. So, no. A bacteria rhizobium? No, children, bacteria rhizobium, it's nitrogen fixing bacteria. It is also not working like that. I said only few bacteria and the fungus. But over here, the perfect answer is a fungi, that is rhizopus. What is rhizopus, children? When it is given over here, and you don't have to worry about anything else. Okay, you don't have to worry about anything else. What does rhizopus do? Rhizopus is also known as bread mold. Okay, and they are saprophytes. You might have seen them settling on the bread, the moist bread, right? So that is one thing, and rhizopus is the answer. Bacteria rhizobium? No, it's not the answer. It has a different mode. It's not saprophytic. It's not saprophytic. It has a very different mode. They are basically autotrophic, chemoautotrophic. So that is something different. Okay, I hope you got this. Now going ahead, what do we have? So if you know this option, then you won't be confused between this and this. Rhizopus for sure, fungus for sure. Fungus has the more number of chances. Okay, few bacteria are known there which are saprophytes, but most fungus, they are what, children? They are saprophytes. Okay, so if you want more examples, there is yeast, mushroom, all of them, they are fungi. Now they can ask you what is that single celled fungus that is saprophytic. So yeast is single celled, children. Yeast is what? Single celled fungus. Okay. Now going ahead to assertion and reason. Assertion. In humans, males play an important role in determining the sex of a child. Reason, males have 2 X chromosome. Okay. Assertion is completely true. In humans, male definitely play an important role in the sex of a child. Why over here? You see, male and female, if you see, okay, the genes that the female will be having is always XX. They will be having one X allele and the other X allele as well. Whereas when you talk about males, they have one X and the other one is Y. Now, depending on what do they give is the sex of the child. So let's see. So over here, if in the gamete, if in the sperm, X is there, and if in the sperm, Y is there. Okay. So first let's go with X, and this is the gamete, children. Over here, females do not have an option, they only give X. Now if this and this fuses, it will be XS, that is, this child will be what? Female. Going ahead, if this and this fuses, again it will be XX, again it will be female child. So now you understood one thing, that if the male partner is giving X chromosome in the gametes, then the child will be female because they will also be giving X. On the other hand, now when you talk about Y, if Y is coming from here and X is coming from here, obviously it will be XY, and this will be what, children? A male child. Again, so basically whatever gametes come down, whatever gametes, okay, basically what allele is there in a gamete, that makes the whole difference. Okay, so, and that is from the males, because males only have the option. So yes, in human, males play an important role in the sex of a child. The assertion is true. I am writing. Assertion is true. Now what about reason? Males have 2 X chromosome. No, children, as you can see over here, males have 1 X and 1 Y chromosome. So reason is false. Reason is false. Okay, got it? It's from heredity. Okay, going ahead, life processes. Assertion, in humans, the inner lining of the small intestine has small finger-like projections called villi, which are richly supplied with blood vessels. Agree? Yes, they are richly supplied with blood vessels, and we humans, we definitely have villi. Okay. Now, villi increase the surface area for the absorption of digested food. Villi increases the surface area for absorption of digested food. That is also true. So, for example, you think if the inner lining of a small intestine was like this, okay, the food can pass through only these surfaces. So, we have limited surface. Now, instead, if the same thing, if you occupy the same position for making linings like this over here. So over here you see with the same distance we have folds, and with the help of these folds we can take more nutrients inside, because they are definitely increasing the surface area for absorption. Okay, now we know assertion is true, reason is true, but is the reason explaining the assertion? Let's see that once more. In humans, the inner lining of the small intestine has finger-like projections called villi, which are richly supplied with blood vessels. Okay, now reason is villi increases the surface area for absorption of digested food. Is it true? Yes. Okay, is it explaining the assertion? Yes, we definitely have this for absorption of, so both assertion and reason is true. And reason is explaining A. Okay, and they are richly supplied with blood vessels. So in the blood vessels only these nutrients go. Okay, going ahead. Okay, assertion is blood clotting prevents excessive loss of blood. Agree? All of them agrees, right? So yes, it is true. So assertion is true. If clotting is not there, children, the blood will just go on outside. And what is that condition? Is a genetic condition where the blood does not stop bleeding. Hemophilia. There the clotting enzymes are damaged. So over here, assertion is true. What about reason? Let's see. Reason is blood clotting is due to the blood plasma and white blood cells present in the blood. I'll say reason is false. Why reason is false, children? Assertion is true, reason is false, because clotting is done because of not white blood cell but the platelets, and platelets, children, are known as thrombocytes. So in our blood we have three types of cell. One is WBC, one is RBC, and one is platelets. WBC is known as leukocytes, RBC are known as erythrocytes, and platelets are known as thrombocytes, and these thrombocytes or basically platelets are the one which is responsible for blood clotting, not them. So this reason is entirely false. Going ahead. In humans, if gene B is responsible for black eyes, okay, I am writing. So whenever they have mentioned B in capital, that means it is dominant, okay, it is dominant. So let's write, decode. So B gene is for black eyes, okay, responsible, and gene B, small b, is responsible for brown eyes. Small b is a recessive trait, okay, which is why they have mentioned in small letters, brown eyes, okay. Then the color of eyes of the progeny having combination, gene combination capital B, small b, BB or BB will be black only. You tell me. Now this is dominant, you know, this is small, this is recessive, you know, okay. And now they are saying if this is capital B, this is the combination, then the child, whoever is having this, they will have black color. Okay, that is fine because this is dominant, so this will overshadow it and the color will be black only. But now the next condition, if it's BB, then also it will be black. Will it be black? You tell me. Is there anyone to overshadow the small b? And small b is for brown eyes, right? And both of them are the same. Will it be black? No. This is wrong. Next statement. Or it has to be capital B. In this case also it will be black. But again, as I said, here it won't be black. So the assertion itself is what, children? Wrong. Okay? Assertion is false. Now talking about reason, let's see. Reason. The black color of the eye is a dominant trait. Is a dominant trait over here? Yes. As they have mentioned over there, with this piece of information, it is the dominant trait. So reason will be what? True. Assertion is false and the reason is true. Okay. If they had interchanged the whole thing and now B was small here, capital B was saying brown eyes, then obviously that will be dominant. So capital B, whenever the capital L comes, just understand it's a dominant trait. Okay. This came in heredity. Now going ahead. Assertion, the energy which passes to the herbivores does not come back to the autotrophs. What do you mean by autotrophs, children? What do you mean by autotrophs? So the producers are the ones which are known as autotrophs. Autotrophs as in those organisms which can prepare their own food, right? And now they are saying the energy which passes to the herbivores. So producers, basically they are eaten up by herbivores. Agree? For example, a deer. Here what was the example? A plant, obviously. So does the energy which passes to the herbivore come back to the autotrophs again? No. It is a unidirectional thing. The energy goes like this. It will come back later on during the whole cycle, but it's not a direct process where direct energy which is there in the herbivores will come back to producers. No. Have you seen plant eating a deer? No. Instead, a deer eats a plant. So energy will be going in that way. It will be always unidirectional. So the assertion that is there, the energy which passes to the herbivores does not come back to the autotrophs, it is true. Assertion is true. Okay. Now next thing, the flow of energy in a food chain is unidirectional. Yes, it is unidirectional. So reason is also true. And is it explaining the assertion? Yes. Explains the assertion. It is unidirectional, which is why it does not come back. Okay. So assertion and reason is true and the reason explains the assertion. That is the correct answer. Now, going ahead with our 2-marker question. Now we have completed assertion and reason and MCQs. Let's see the 2-marker questions, children. Okay. Write the role of insulin in regulating blood sugars in human body. Do you know? Mention the disease caused due to it. Do you know that? Write the role of insulin in regulating blood sugar levels. So how does insulin work? So you have to tell that. basically insulin is a hormone which is released by what children? Pancreas. So your first statement will be insulin is a hormone released by the pancreas, which is a heterocrine gland. If you want to mention that, you can mention that. Hetero as in it is both doing endocrine function, that is releasing hormones and releasing enzymes. But as of now, since they are asking about blood sugar levels, let's concentrate on that. So now insulin, basically what does it do? You have to tell about that. Okay, insulin converts blood glucose into glycogen and let it store in that form such that blood glucose level will be low, okay, or basically normal. Okay, and where does it store? In the liver. Now, if this is not happening, what is the disease that is caused due to this? Now, you might have seen people taking insulin shots, right? They take insulin injections. What are this? Basically, their pancreas is not working well to produce enough amount of insulin. At this point of time, someone is giving externally such that this insulin does the same work. It converts the glucose which is there into glycogen. Now, if this is not happening and you are taking insulin injections, what is the disease that is there? It is known as diabetes. It is known as, children, diabetes. Okay, so you got the answer for this. That is 1 mark. Now, how is the timing and the amount of release of insulin in the blood regulated? How is it regulated, children? You tell me. How is the timing and the amount regulated? That is the next set of answer. The timing and the amount of insulin regulated is by the amount of blood sugar in the blood, okay, or basically sugar level in the blood. Sugar level in the blood. If it is high, if blood sugar is high, insulin is produced, okay, which converts what? Glucose to glycogen. Now, if write in points, okay, please in boards write in points. Here also I am not writing the whole point, but it's just for you to understand how you should be framing your answers. Now, next thing, if blood sugar is low, insulin production will also be accordingly low. Blood sugar is low, okay, do we need to convert it into glycogen? No. So, it will be obviously less or it will be nil, okay. In that point of time, what happens? Basically, glucagon is released. That is another enzyme which breaks down glycogen to glucose. If our blood sugar is less, at this point of time, the opposite of this thing happens. Okay, glucagon becomes active. So, as of now, they are not asking about glucagon. They are talking about release of insulin in the blood. So, you have to talk like that only. It is dependent on the amount of blood sugar in the blood. Okay, the sugar level in the blood. If the sugar level is high, insulin is produced, which breaks down glucose to glycogen. Okay, converts the glucose to glycogen. If it is low, insulin won't be produced. So, hence, the same thing is solved. Okay, so this is how you have to answer. This is a 2-marker question. One for this, one for this. Okay, and I'll tell you one more thing. How is basically papers evaluated? So, teachers, they get a paper sheet in which the answers are written. Okay, point to point, that's all. They do not have long lengthy answers like that, and this person could be a chemistry teacher, could be a biology teacher, could be a physics teacher. So your paper could be checked by any of these teachers, okay, who might not have great understanding of other subjects like biology or chemistry, or basically they might have understanding of biology but not of chemistry and physics. So whatever answers is there in the blueprint, they will just see that and give you marks. So your answer have to be accurate. You get it? Okay, so please understand that. And basically also I will include one more term over here. It works on negative feedback mechanism. Just a second. This particular thing also works on negative feedback mechanism. You can just say the timing is also controlled by feedback mechanism. What do you mean by feedback mechanism? Basically over here there's your negative feedback, as in this is high, sugar is high, so we have to make it low. This is negative feedback. We do not want that to be high, we want it to be low, so we release something which would make it low. This is negative feedback. Now on the other hand, if it was like this, the sugar is high and you're releasing something for making it more high, that is positive feedback. So at this point of time they are showing negative feedback effect. So you can say the timing and the amount of release of insulin or any other hormone is also based on feedback mechanism, where if something is more, okay, where something is more, depending on it the hormone will be released. If it is something is more, for example glucose is more in the blood and you have to make it less, then they show negative feedback effect. Okay, so insulin basically works on negative feedback role. Okay, that is also one very important thing, please mention it somewhere. Now going ahead, name the type of blood, oxygenated, deoxygenated, transported by each of the following, mentioning the path. That is from one organ which place to another organ which place. Okay, so we have to say it's okay. So basically, vena cava. Vena cava. What organ to what organ? Basically, your body parts to your right atrium. To your right atrium. Right? If you remember, I can make a small diagram over here. So this is our heart, children. Left atrium, left ventricle, right atrium, right ventricle, and over here we have the lungs. Okay, why is the color not changing? Okay, and over here we have the body parts. Okay, so oxygenated blood, the thing that we take in, we have so much amount of oxygen in the lungs. So from the lungs, oxygenated blood comes to left atrium. Bring in oxygen. You can remember like that. It goes to the left atrium. Now from left atrium, it goes to the left ventricle. From the left ventricle, it goes to the body parts. Now this thing is done by aorta. That is an artery. This thing of bringing the blood to the left atrium is done by a vein. It's an exceptional vein which carries oxygenated blood. So this is pulmonary vein. Okay, pulmonary, wherever the term pulmonary comes, just understand they are exceptional. So now oxygenated blood has come over here, aorta has given to the body parts. Now deoxygenated blood will be moving from here to the right atrium, and this is with the help of vena cava. You have two vena cava, superior vena cava which collects from the upper part of your body, inferior which collects from the lower part of your body. Now from right atrium it goes to the right ventricle, from the right ventricle it again goes back to the lungs because we ought to make it oxygenated again. Now who carries this to the lungs? Basically it is pulmonary, again as I said it will be an exception. Pulmonary artery. So artery usually carries oxygenated blood, but this is an exception and this is also an exception. Okay, this is the loop. Now over here, you know, from the body parts to the atrium, right atrium, it is vena cava that is carrying it. And what type of blood, children? Deoxygenated. Deoxygenated blood is being carried. I hope you got it. Can you tell about pulmonary artery now? Can you tell me about pulmonary artery now in the comment section or something like that? If we talk about pulmonary artery, okay, over here pulmonary artery carries blood from right ventricle to the lungs. So basically you can just mention heart over here in bracket. And what type of blood is being carried? As I said, it's an exception. It also carries deoxygenated blood. Done? So I hope you got this. This is this, this is this. Okay. Next question. With the help of a schematic flow chart, show the breakdown of glucose in a cell to provide energy in the presence of oxygen, in the lack of oxygen. So you have to just draw the diagram which is there in your NCERT textbook. Very easy thing. Okay. So you have glucose, which is what, children? A 6-carbon compound. It has 6 carbon. Glucose basically breaks down into what? Pyruvate, which is a 3-carbon compound. And how many pyruvate do you have? 2 pyruvate. So basically you only think if there is 6 of something, okay, there is 6 carbon or basically 6. So 6 if you want to divide into 3, because this is just a 3-carbon compound. So obviously it will be 2 compounds that is being formed. So you have 2 pyruvate which is being formed, And apart from 2-pyruvate, no children, you also have some energy being produced over here. Some energy also being produced over here. Okay. Now, this happens in the cytoplasm of the cell. This happens, if this is a cell, then there is cytoplasm, then there is mitochondria and everything, there is nucleus and all the things. So, this is happening in the cytoplasm of the cell, and this process is known as glycolysis. They are asking, in the presence of oxygen, what will happen? In the presence of oxygen, okay, this pyruvate basically, in the presence of oxygen, okay, it will get entry into what, children? Into mitochondria. So, until now it was there in the cytoplasm. The pyruvate slowly takes an entry into mitochondria, gets converted into something known as acetyl-CoA. It undergoes a huge set of reaction, okay, huge set of reaction, and finally gives down to CO2, H2O and ATP, that is the energy. So, what does it do? CO2, H2O and ATP. Okay. So, over here this is happening in the presence of oxygen, which is why we say we need to breathe in and take in oxygen. This oxygen is utilized for this particular process, for the whole breakdown of glucose. Now, the breakdown of glucose is finally completed. It has just started off, now it is finally completed, and this whole process is known as respiration. Okay. When it happens in the presence of oxygen, you call this process as aerobic respiration. You call this process as aerobic respiration, and during respiration we say CO2 is given out. This is the CO2 that we breathe out. This is the CO2 that we breathe out. What do you think? Where does CO2 come from in our body? Okay, so this is the CO2 that we give out, along with H2O is released and also ATP. Now, this ATP is used to run all the reactions of our body, and also ATP is utilized for this particular process. So, ATP which were made before were utilized over here, and we are making new ATP as well. Now, in the presence of oxygen it is done. In the lack of oxygen, what will happen? In the lack of oxygen, children, it would not go to the mitochondria. It would not go to the mitochondria, but still you have to make something like this, and you can write, in the lack of oxygen, okay, and this happens in the cytoplasm itself. In the cytoplasm, here only this is happening. What will happen? What is produced, children, in the lack of oxygen? Lactic acid is being produced, and apart from that, one more thing is being produced, energy. We need to produce energy also. That is why we are doing respiration, right? But this amount of energy and this amount of energy will be quite different. This is 36 molecules of ATP, whereas it will be 2 only. Okay, you see the difference, but still ATP is being produced, but here lactic acid is being produced, and this is what, children? This is an aerobic respiration, where oxygen is not being utilized. This is an aerobic respiration, and this lactic acid is the one that causes what, children? Cramps. This causes cramps. So, basically you have seen people who run, okay, when they feel breathless and all, at this point of time, what happens? Basically they get cramps. Why? Because you are not getting enough amount of oxygen. If there is no enough amount of oxygen, they cannot do aerobic respiration. Instead they shift to anaerobic respiration, because we still want to produce ATP. But at this point of time, one bad thing happens: lactic acid is being produced, and when it accumulates way too much, it will cause cramps in the muscles. Okay, so you have to draw this thing and show them exactly what happens. Please mention where it is happening also, and the presence and in the absence of oxygen you have to write like that also. Please mention the energy form and everything. Okay, in a cell this happens. Now, there is also one more case. Okay, there is also one more case for lack of oxygen only, but this is not happening in our body. It is happening in yeast. It is happening in yeast. At this point of time, again in lack of oxygen, same, in lack of oxygen. Okay, in yeast what is being produced, children? In yeast they are producing ethanol, that is an alcohol that is being produced, plus CO2 is produced, plus energy is produced. So, ethanol is produced, CO2 is produced, energy is produced. This is also happening in the lack of oxygen. This is also happening in the cytoplasm, just like this. Just that the product is different, because the thing that is undergoing this is also different. This happens in yeast. So yeast, you might see wine is formed when wine has a little amount of alcohol. This is the alcohol, ethanol. And then carbon dioxide and then energy. So you might have seen we put it very tight lid, okay, so that oxygen is not getting inside. So if oxygen is not happening, then only oxygen is not entering, that's when fermentation would happen, and that's when the wine is produced, or basically this whole reaction takes place there. Okay, so you can show this. Going ahead, what do we have? Oh, I actually had a page to write this. Bad, Jasmine Ma'am. Now next, how do harmful chemicals get accumulated progressively at each trophic level in a food chain? So for explaining that question, what do you need to do? You have to make a pyramid. Okay, you have to make a food web, or sorry, food chain in the form of pyramid. So over here, plants, and then deer, okay, and then what can we make? Just a second, okay, yeah, plants, deer, and then what can we make? Okay, instead of deer, I'll just make a quick change. So yes, so there is rat, there is snake. Are we mentioning about us? No. And there is hawk, that's a bird which feeds on snake. Now see over here, the plants, in plants, no, I put a, for making them grow, I put a pesticide, and this pesticide was, children, DDT. This pesticide that I was using was DDT. Okay, now DDT, when I put in one plant, there is one plant over here, there's another plant, there's another plant, there's another plant, okay, each having, for example, each having 2 mg of DDT, 2 mg, 2 mg and 2 mg. So if I see, basically one among them will be having just 2 mg. Okay, one among them will be just having 2 mg. But now when the rat eats it, rat can eat multiple plants. It ate this, this and this, for example. Okay, one rat. Okay, so for example, not one rat, I am making 2 rats. So this does not look like rat, but yes, this is a rat, okay. This is not a bee, this is a rat. My drawing skills have been terribly bad for some days now. Okay, now basically a rat ate these 2, another rat ate these 2. Now this particular rat will have 4 mg of DDT. Over here it will also have 4 mg of DDT. Agree? This guy was only having 2 mg. But over here, one rat is having what, children, I'll make it more clear, it will be having 4 mg of DDT. Okay, so if we see, this was a trophic level. Over here I had this many, okay, some they didn't have 2 mg, some did have, but when a rat feeds on it, it feeds on lots of organisms, lots of plants together, so obviously it will be having more. Now if you talk about the snake, okay, so again you have a snake. Okay, you have a snake. This snake will feed on this. So it has 8 mg in itself, and you will have another snakes as well, which feeded on another rats, right? So they will be having also that much amount of being stored. It will be more than 8 mg, or it could be more or less like that than only. For example, 6 mg. Okay, it feeded on another 2 rats, which had 6 mg. So in this way, if you see, now when a hawk eats this and this, so 8 plus 6, how much is there? 8 plus 6 is 16, 14, okay. So basically 14 mg of this is there accumulated over here. So the question is, how do harmful chemicals get accumulated progressively at each levels? Because this is a trophic level, this is another trophic level. You have to mention trophic level 1, trophic level 2, trophic level 3, trophic level 4, and you have to say the plants which are there in one trophic level has enough amount of pesticides, but there are so many plants being eaten up by a rat, which is there in trophic level 2. Now this particular chemical does not move outside the body, instead it stays inside the body, and this process is known as bioaccumulation. What do you mean by bioaccumulation? Well, you have to explain. Toxic chemicals accumulates. Why does it accumulate? Because rate of intake is more than rate of excretion. So things does not move outside because these chemicals that is being taken in, it cannot be broken down easily. So it stays there in our body. So we are keeping on having it. okay, and it's not getting outside, so basically it is getting accumulated. Now again, when in organism in the trophic level 3 eats much more amount of rat, which has enough amount of chemicals being accumulated because of bioaccumulation, they will be having more. In this way, if you go at the top and the top, the amount of chemicals, it increases, and this process is known as, children, biomagnification. Biomagnification, where toxic chemicals levels increase in each trophic level. I hope you got this. You have to explain both biomagnification over here and bioaccumulation over here. You have to make this diagram, say T1, T2, T3, T4, that is trophic levels, and you have to say that little amount of DDT is taken about by plant, but it is taken by different plants, and these different plants are eaten by so many different rats in trophic level 2. It is getting accumulated, it is not moving out because of the process that is known as bioaccumulation. Now rats are being fed by the snakes. Again, this is getting accumulated, but now the amount has increased. Why? Because the snake won't be eating on just one rat, it will be eating on multiple rats over here in trophic level 2, right? So obviously the amount has to be increased. Now it goes in hawk is the highest trophic level, okay? So basically over here the amount will be slowly increasing because again of bioaccumulation. So you have to make utilize of this and this while explaining your answer. If you want the example of a pesticide, you can say DDT. Okay, or if you are not able to give an example of a pesticide, say X toxic pesticide is being used, and then explain your answer, but you have to explain in points, okay, with these particular things in mind. This is a 2 marker, so according to 2 marker you have to answer. Do not write too much, just explain this and this a bit, okay. Going ahead, there is no E here, okay. How is an electrical impulse created in human nervous system? Identify the parts of neuron that helps the nerve impulse to travel towards the cell body, away from the cell body. So let's see. So basically, if I draw the structure of a neuron, okay. So you can explain like this, okay. Make a proper diagram, okay. As of now, I am not making too much amount of details over here. Now, okay, listen to me very carefully, okay. This is the structure of the neuron, and I will answer the second part first. Who is the one that carries the things towards the cell body? So where is the cell body? This is the cell body in the middle, and the cell body, no, it has certain extensions. So there are certain extensions over here. There is one long extension over here. This extension that you see over here, this is known as axon, okay. And these extensions that you see over here, they are known as dendrites. And dendrites, they have certain receptors over them, okay. They have certain receptors over them. Now going ahead, axon, these things, the terminal portion of axon is known as axon terminal. And at the end of them, they have something that is known as synaptic knobs. These synaptic knobs are nothing but they are rich in neurotransmitters, which are chemicals, okay. So basically I will tell you what happens. Once some excitation happens in the neuron, they will release out these neurotransmitters which are over here, which are nothing but chemicals, okay. These are what, children? Neurotransmitters. Neurotransmitters, okay. Now these neurotransmitters will go and bind on the next neuron's dendrite, that is over here on the receptors, and now it will send the signal forward as well, okay. So this space that comes between one neuron and another neuron, what do you call this space as? Synapse. Okay, so now nerve impulse, who is the one that carries towards the cell body? It will be dendrites. So this is your answer number 1. Okay, second thing, away from the cell body, who is taking away from the cell body, children? It is axon. So this will be your answer number 2. Agree? So I am marking it in green so that you do not forget it. Now, how is electrical impulse created in human nervous system? Basically, electrical impulse is created when our receptors are active, when our sensory receptors are active, and when receptors of dendrites are active, receptors of dendrites are active, what will happen? The ion channels which are over here in the neuron, it will open up. Ion channels in the neuron opens up. Now what will happen? There will be intake of ions. Intake of ions would lead to change in membrane potential. So there is a normal membrane potential that is minus 70 millivolts. So when that change happens, they will send down a signal. They will make open other ion channels, and ion channels will open other ion channels, and the same thing goes on until the end, such that there will be other ion channels which will let calcium ions inside, and then these calcium ions basically says that neurotransmitter, you have to be released now. So this is a chain of reaction that happens because what? The membrane potential is changed. So intake of ions would lead to change in what? Resting membrane potential, which is minus 70 millivolts. Okay, which is minus 70 millivolts. Now, obviously, when minus 70 millivolts go down, it will change to minus 55 millivolts. At this point of time, the ion channels will go quickly open up, and this is known as an action potential. This process is known as an action potential, or basically you say an electrical impulse is being created. So how can we explain this answer very easily? Electrical impulses are created when there is change in the membrane potential of the neuron. How can I say? Electrical impulse is created when there is a change in membrane potential of the neuron because of intake or exit of ions. Because of intake or exit of ions. Okay, and this impulse is being carried ahead. So you don't have to explain all this thing if it's very difficult for you. Can you just say this? That the resting membrane potential, which is normally there in a cell, it is disturbed because of intake of ions and exit of ions. And this causes the electrical impulse to go forward. You can say that, and it happens, if you want to explain betterly, you can say that this happens when the receptors of the dendrite are active, when some chemicals binds on the receptors of the dendrites, ion channels open up, and there will be lots of ions inside, which would change the resting membrane potential, that is minus 70 millivolts, okay, to minus 55 millivolts. Okay, how does it change? I'll tell you. Basically, what ion channels are opening up? Sodium ion channels are opening up, and sodium is a positive ion. Now something positive coming in, obviously the minus charge will get reduced. So it is now minus 55. Again, this will make open potassium ion channels and lots of other things which you do not need to explain as of now. But you can just say the membrane potential is changed, which is how the electrical impulses is created. Okay, if it goes beyond minus 55, it is going to create an electrical impulse over there. Now you know who is the one that carries towards cell body and who is the one that carries away from the cell body. Okay, so cell body is the place where all your organelles are present, okay, except centrioles. Centrioles are not present because if centrioles were present, they would be responsible for cell division, and neurons do not do cell division. So except centrioles, all of them are present, so it controls all the metabolic activity. They have the neurons, they carry the impulse, and they are the one that will be releasing what? Neurotransmitters. Okay, so all of them has a property. Fine, going ahead. With the help of an example, explain how does feedback mechanism regulate hormone secretion. So over here I told you what is feedback mechanism. Basically, if something is more, okay, if something is more, you have the possibility to make it less. And this process is known as, this particular process that you see is known as negative feedback. Negative feedback. Example? Example, what do you have an example? Chalo, let's see. Over here, blood sugar level is high. Blood sugar level is high. At this point of time, you want to make it what, children? You want to make it low. This is negative feedback. Okay, so some things are going to happen over here. So since blood sugar level is high, what will happen? Insulin will come in play, and insulin basically converts glucose to glycogen. And now what will happen? Blood sugar level decreases. Now it is what? Decreased. You wanted to do this and you did this. Now here negative feedback is happening because something was more and you are making it less. You are not making it more. On the other hand, if you want example of positive feedback, even though they asked you just one example, but I will tell you about positive feedback. When something is there in more amount and you want in more amount, positive feedback. So over here, so during when the baby is born, okay, you know the mother's body, uterus basically contracts, okay, and that's how the baby is moving outside. So this contraction happens with the help of a very important hormone which is known as oxytocin. Okay, so basically you can remember like this. So oxytocin leads to contraction of uterus, contraction of uterus, and this contraction will lead to more oxytocin. This oxytocin will lead to more contraction, and the cycle goes on. That's how the baby is moved out. So over here, as you see, you're making it more and more. It's not like something is produced more, so you're making it less. No, contraction was there, you're making it more with the help of more oxytocin. This is the example of positive feedback. This is the example of negative feedback. This is something that you need to explain over there. Okay, done. This is a 2-marker question. Explain in your own words. Make flow charts if you can. It will be much more helpful for you. Give the name of the enzyme present in the fluid in our mouth cavity. What is the fluid that is present in our mouth cavity, children? It is the saliva. Okay, and what is the enzyme that is present in the saliva? I just know about one enzyme, and that is what? Salivary amylase. Okay, state the gland which produces it. Who produces saliva, children? Who produces saliva? Can you tell me? It is what? Salivary gland that produces saliva. You have multiple salivary glands. Over here, you have so many, you have almost, I think, 1, 2, 3. You have 3 salivary glands, pairs. Okay, you have 3 pairs of salivary gland over here. Now, going ahead, it produces saliva, and what is the enzyme that you have? Salivary amylase. Okay, what would happen if the digestion process, what would happen to the digestion process if this gland stops secreting this particular enzyme? So, what is the use of this particular enzyme, children? It is responsible for conversion of starch to something that is known as maltose. So, and this happens in a basic pH, that is almost 6.8 pH. So, now if this particular thing is not being produced, what would happen? If the gland is not producing this, what will happen? Obviously, starch would not be converting to maltose in the mouth. So, usually I will tell you an extra thing, 30% of the starch is being done by this, okay, rest 70% is being broken down in your pancreas. So, basically you have to say 30% of starch conversion which happens over here would not be happening in the mouth. This is something that happens. So, basically starch is not getting broken down to maltose. This won't be happening. So, they are directly asking you the function in a very twisted way. That's all. So, it's a simple question, answer in a simple way. Do not write too much about it, just point to point. Salivary gland is the one that produces saliva, or basically the thing that they ask first. What is a fluid? Basically salivary amylase is the thing. What is that thing that produces? Salivary gland is the thing that produces. What does salivary amylase do? Wherever you have written salivary amylase, write starch to maltose, and then write below that this secretion would be, this conversion of starch won't be taking place in the mouth. Okay, because it is happening in the mouth itself. That's all. Next, what is vegetative propagation? List 2 advantages. What do you mean by vegetative propagation, children? Where basically you are using plant parts, vegetative parts of the plant. What you are doing? You are using vegetative plant parts for reproduction. Okay. What is the example of vegetative plant parts? You have leaf, okay, you have stem, you have root. You can give an example. Give an example of the most common example, bryophyllum. Bryophyllum leaf produces what buds, children? Adventitious buds. Adventitious buds at the notches. When fallen down, it would form what? Plants. So basically, if this is your bryophyllum plant, over here you will be having small buds. Okay, and why are they known as adventitious buds? Because buds are usually produced where nodes are there. So this is the place where usually buds are produced. Now buds are being produced somewhere else, unusual places, so you call this adventitious buds. Now these buds falls down and forms a whole plant. So here, do you think gametes are involved? No. Here vegetative plant parts are involved, that is leaf. Okay, so you have to say vegetative propagation is where in plants, vegetative plant parts are responsible for production of new plants instead of the gametes. So the part that produces gametes is what? Flower. So this particular part is basically not being utilized. Flowers are not being utilized for production, instead vegetative plant parts are being produced. Okay. Now list 2 advantages. Can you tell me the advantages? It is fast. It is fast way of reproduction. It's a faster way of reproduction. Second thing, it would guarantee production of desired traits. Production of desired traits. Now, this is natural vegetative propagation, there is artificial vegetative propagation which helps in all this thing. Okay, what happens there? Basically, you have seen rose plant. A rose plant basically is cut. Okay, a rose plant is cut and this particular plant is made where it has a nodes and it is put somewhere else and it is allowed to grow. Now over here I am making it fast. If I would wait for this particular rose plant to give me flowers, it will take a lot of time. But if I am breaking a part of it and putting it somewhere else, basically I have got 2 parts from the same plant and it will be really faster. Now second scenario is when I am doing grafting. In grafting what happens? When I am having a plant part, I am putting another plant part over here. Now I would have 2 plant parts in a similar plant, okay, and I will be having my desired traits, whatever traits that I want, I will be having that over here. Okay, so that is one very important thing of it, and that is the thing that I mentioned over here. So it is a faster way of reproduction and would also generate progenies with desired traits. Okay, done. Let's go ahead. List 2 distinguishing features between sexual and asexual reproduction. So asexual reproduction and then you have sexual reproduction. Can you tell me the difference, children? In the chat box, if you're seeing this video, can you tell me in the chat box what are the differences? In asexual reproduction, do you think there is involvement of 2 parents? No. No involvement of 2 parents. Here you need involvement of 2 parents. Is there fusion of gametes here? No. No fusion of gametes. Here you do not need gametes. Okay, so no fertilization. Over here, fusion of gametes will be there because 2 parents are producing, and hence fertilization will also be there. Fusion of gametes means fertilization, right? Now, if you want one more difference, they are asking for just 2, but I can give you one more. Less variation because only one parent is involved. Okay, but over here more variation because obviously two parents are involved. Okay, this is question number 11. You can answer like this. Going ahead, ozone, although a deadly poison, performs an essential function for the life on earth. Justify the statement. So you have to tell me, why is it considered to be a deadly poison? So you have to say ozone is what, children? Ozone is nothing but O3. So oxygen molecule, when it combines with O, it forms ozone. Okay, now ozone at what say ground level, that is when it is there in our level over here, it's not there in the stratosphere, instead it is over here at ground level, is a deadly poison as it causes respiratory issues and stuff like that. Okay, respiratory issues, eye damage and everything. But in stratosphere, it forms a layer. Okay, you know, right? It forms a layer, that is ozone layer, which does what, children? Which absorbs UV radiation, especially UVB radiation. Okay, and hence protects us, or basically protects the environment from any damage. Okay, as what? As UVB radiation or UV radiation can cause cancer or other diseases. You have to answer like this. Okay, in points. I hope you got this. This is a 2-marker question. Explain according to that. Do not invest too much time into this. Okay, going ahead, we are now starting 3-marker questions, and now I want you to take a break, drink some water, have a 5-minute stroll around. I will also drink some water and I will come along. Okay, so I hope you are carrying your water bottle with you and drinking water. Okay, Chutku people, I hope that you drank water and now you are energetic. You had a quick little stroll and you came back. So let's go ahead with the 13th question with 3 marks. In human excretory system, name the following parts in which urine is produced. So where is urine produced, children? Any idea? I have an idea. It is produced in what? In the kidneys. So basically you have the kidney, it is produced over here, so you have the answer, that is kidney. Okay, in the kidney what do we have, children? Nephron. So please mention that also, in the nephron which is there in the kidney, so that your marks would be safe. Okay, going ahead, what do we have? Where is the urine stored? So let's see. So basically urine which is formed in the kidney goes to some place like this. Ayyo, just a second. Okay, what do you call this place where it is stored? It is known as urinary bladder. Okay, so this is the second answer. Agree? It does not have this big opening, please. Okay, so basically, which connects 1 and 2 mentioned above? So this is 1, you know that this is 1, and what is that structure that is connecting 1 and 2, children? What do you call this? Ureter. Okay, you call it ureter, and ureter is the one that carries the urine. Now, which provides passage for urine to pass? Now, this passage which is there, which allows urine to pass, what do you call this? Urethra. This is what? Urethra. Now you tell me, was the diagram important? Yes. Okay, now going ahead. So you don't need to make the diagrams there. Over here you just have to name them. So it's better you just name them. Don't draw the diagrams over there. It's not needed. Now list 2 factors on which the amount of water reabsorbed along the tubular part of the nephron depends. So there is nephron. Okay, there is something that nephron. Do we have any space? No. I can show you over here. Okay, so basically the nephron, it looks like this. It's a very easy diagram that I am going to make. Okay, and it opens to collecting that. So basically you have blood vessels over here. These blood vessels basically then moves around the whole thing like this. Okay, so whatever thing is been there which we do not want comes to this particular thing, that is our glomerulus. From glomerulus it goes to this particular tubes, and they are asking you on what factors does the reabsorption depends, as in things are getting reabsorbed to the blood vessels. On what factors does this depend? And what is the thing? Basically water. How is water reabsorption depend? So water reabsorption depends upon two things. One thing, excess of water in your body, or basically you can say level of water in your body. If it is too much, reabsorption won't be taking place. If it is too less, reabsorption of water would be taking. Okay. Next thing, amount of dissolved salts in your fluid. Amounts of dissolved salts, that is basically what? Ions in the blood. So you know osmosis and everything deficient that is there, right? So basically if there is some, for example, if there is blood vessel and the blood vessel has too much amount of ions, there's too much amount of concentration. So at this point of time, to make the concentration less, what do we do? The water is being put over here, so that the amount of solvent it now increases and the amount of solute, which is the ions, it decreases, right? So amount of dissolved waste in the blood also determines how much amount of water is being reabsorbed. If it is too less, okay, if the dissolved salts are too less, the water won't be reabsorbed much. If the amount of dissolved salts is way too much, the water would be reabsorbed. So this is on this particular factors that your tubular reabsorption works. Okay, so level of water in your body, if it is too less, then reabsorption would be there. If it is way too much, reabsorption won't be there. Amount of dissolved salts, if it too much, water would be obviously reabsorbed. If it is too less, again no. Okay, will you remember this? Dissolved salts and the level of water. Okay, and it's a very important question. They have twisted this question and given you so many times. This is from life processes. Going ahead. Explain with the help of a terrestrial food chain as in land food chain, okay, how some harmful chemicals enter our bodies through the food chain. Why is the concentration of these chemicals found to be maximum in human beings? So the same thing that I explained to you where I was talking about biomagnification. So I won't be explaining again, but still I can tell you how you can frame the thing, okay. So let's see. So over here you have food chain starting with terrestrial, so it has to be plants over here, yeah, on the land area, and there has to be something which is over here which would be eaten by the, let's say, okay, so for example there are certain worms. What could be there? Just a second, just give me a second, okay. So here you need humans, and humans, as you know, can feed on cattle, okay, for example, okay, I can make like this. So here we have goat, okay, and then here we have human on the next trophic level. Agree? Okay, they have to be a non-vegetarian to be like this, right? So over here you see the plants again has DDT in more amount. This would be eaten by goats, yeah, and the goats would be eaten by human being. Now, why is the concentration of these harmful chemicals found to be maximum in human beings? That is the question. Because human beings, my child, is occupying the highest trophic level. It is occupying what? The highest trophic level, and the highest organisms on the highest trophic level will be feeding on organisms on the lower trophic level. Agree? It can feed on this, it can feed on this also directly. Okay, either way it can feed on that. And but in both of them, both of these organisms which are there in the lower trophic levels, bioaccumulation has taken place. Bioaccumulation has taken place. That means chemicals are being accumulated. Why? Because rate of intake exceeds more than rate of excretion. Agree? So bioaccumulation, things have gotten accumulated, it's not getting broken down. The DDT is there in plants, so many plants, and these so many plants are eaten by goats, and goats are eaten by the human beings. So obviously things are getting accumulated, it's still not going down, but the amount of things that are being eaten is being more. Okay, it is being more. Now they are going to show biomagnification. This bioaccumulation would lead to what? Biomagnification, as in the level of toxic substances increases in trophic levels. Increases in while we go up to the trophic levels. So we have to explain like this only. Basically, plants have DDT in the lower trophic level, and now these plants are being eaten by the goat, which will also be accumulating this DDT, because this particular DDT won't be breaking down, it is just accumulated within them. The amount of them increases if they feed on more and more plants. If they feed on more and more plants, obviously it will getting more accumulated. Now when you talk about humans, when this particular human is going to eat the goat, again the bioaccumulated toxic substances is now within the human body. Okay, now since it is occupying the highest trophic level, it will obviously show biomagnification, because biomagnification means the organism at the higher trophic level would be having more amount of chemicals, as it is being accumulated throughout all the trophic levels. So they are taking the accumulation of all these organisms throughout the trophic levels, which is why it increases. So you have to make a chart like this and then explain your things. Please mention trophic level 1, trophic level 2 and trophic level 3, and then explain and say that humans occupy the peak trophic level, which is why this is happening. Okay, so if plants were there, plants were just they got it, but the individually it is less. When you talk about human beings, individually there is lots and lots of them, because you are having more of it. Okay, you are having more of it. I hope you got it. Okay, now going ahead, what do we have? State the role of ATP in cellular respiration, children. This is all one mark each. Okay, we can think like that. So state the role of ATP in cellular respiration. ATP provides energy. Energy. What is cellular respiration? For breakdown of glucose to carbon dioxide plus H2 plus energy again. So this cellular respiration needs ATP, because why? ATP, that is adenosine triphosphate, mention that adenosine triphosphates stores energy in its bonds. Okay, now when they are utilized, what will happen? This bond will break. When utilized, bonds break and energy is released. Energy is released for what? For reactions to happen. Reactions to happen. Okay. Now, what ensures sufficient exchange of gases in plants? What is there to ensure sufficient exchange of gases in plants, children? Okay. So over here you have stomata. Okay, and lenticels. Lenticels are found in woody stems. Stomata is there in your leaves, and also what? Your stem also has stomata. Okay, green stem definitely has stomata. Apart from stomata and lenticels, what do we have? We have intercellular spaces. Intercellular spaces. So you might have seen the plants, the cells are like this. So there are spaces like this through which exchange of gases can easily take place. Okay, so you have three things, 1, 2 and 3. Now State the conditions on which the direction of diffusion of gases in plants depends upon. So what is that condition on which the direction of diffusion takes place? First thing, atmosphere, atmospheric condition. Okay, if it's too much, for example, there's too much amount of moisture in there, okay, it's too hot, okay, it's too hot, the stomata will obviously close up because you do not want to lose up too much amount of water. At this point of time, obviously the exchange of gases will also be stopped. Okay, so that is one thing. Apart from that, internal condition, for example, in daytime, what is happening? CO2, which is produced with the help of photosynthesis, okay, sorry, CO2 which is produced with the help of respiration is taken up by photosynthesis because that process is happening in the daytime. Whereas in the night time, CO2 diffusion takes place most. Why? Because photosynthesis is not happening. Light reaction of photosynthesis is not happening. So you can explain like that. First thing, atmospheric condition which is there, depending on that, the stomata can open and close, and which is why diffusion of gases can get affected. Now next thing, internal condition, where for example some chemical reaction is happening and where these gases are being used, so obviously their diffusion rate will be less. Okay, so for example CO2 is produced with the help of respiration, but that CO2 is utilized in photosynthesis during daytime. But at night time, light reaction of photosynthesis is not happening, so you do not need CO2 at that point of time. So basically what will happen, this CO2 which is being produced during respiration will be given out, that diffusion will be taking place, whereas oxygen, its diffusion will be less because oxygen will be taken in for respiration to happen. So this is how you have to explain your answer again. Works? Okay, can we go ahead? So atmospheric condition and internal conditions. Next thing, what is the internal energy reserve in the case of plants and animals? So plants, you know, this process happens, that is CO2 plus H2O gives C6H12O6 plus oxygen plus H2O plus energy, right? This C6H12O6, which is, children, glucose, is stored in the form of starch in the case of plants. Now in the case of animals, this glucose, basically, you know, what does insulin do? Glucose is converted to glycogen. This is in the case of animals, and whenever the animals need it, we can break it down back into glucose. Okay, so this is glycogen, and also one more thing, it is stored in the liver. Please remember that. Okay, how desert plants perform photosynthesis if their stomata remain closed during the day? Can you tell me how do they perform photosynthesis if the stomata is closed during the day? So basically you have to explain. During daytime, stomata is closed to do what? To prevent water loss. To prevent water loss. Hence, at night, stomata opens to take in what, children? To take in CO2. Okay, but what is the use of CO2 over there? CO2 is needed in the daytime and light reaction is happening, right? So in the daytime, what will happen, children? In the daytime, energy is being produced with the help of light reaction. Yeah, light energy is converted to chemical energy, and this chemical energy is something that will convert CO2 to what? The glucose. So this should be there in the daytime. So what will we do? But we cannot open the stomata during the daytime as it can lead to water loss. Transpiration would be high. So what can we do? Stomata is opened at the night to take in CO2, and this CO2 is converted to malic acid, children. It is converted to malic acid and stored. Okay, now what will happen? In the daytime, in the presence of energy, what will happen? Malic acid is broken down. Malic acid gives CO2 and the whole process takes place. So how it is different? How desert plants perform photosynthesis if the stomata remain closed during the day? Because the malic acid is being stored at night itself, and then during the day when enough amount of energy is there, this malic acid is again getting converted to CO2 and this CO2 is being utilized. So you have to explain like this, 1, 2, 3, 4 points. Okay, can you explain like that? It's a 3-marker question. I think 1 for this and 1 for this, sorry, 1 for this and 2 for this. It gives you enough answer for 2 marks. Right? So explain use of malic acid and everything, okay? This thing is very much important. Going ahead, what do we have? Why is it not moving? Move, dear. Okay, it's not moving, so I have to make it move. Do we have space? Okay, our environment. Next question. Construct a food chain of 4 trophic levels comprising the following: hawk, snake, plants and rat. This we already made. Do you remember? We already made this. So let's, for your happiness, let's make it once more. So over here we have a food chain and they have said hawk, snake, plants and rat. So plants, do they occupy the topmost trophic level, children? No, they occupy the lower trophic level. So here we have plant, and then plants are fed by the rat. The rat is fed by the snake. The snake is fed by the hawk. Okay, this is how you can make the food chain. Please mention T1, T2, T3. T1, T2, T3, trophic level 4. Now, next question. 20,000 joules of energy was transferred by the producers to the organism of second trophic level. How much? 20,000. Okay. Calculate the amount of energy that will be transferred by organisms of the third trophic level to the organisms of the fourth trophic level. So what is happening? Basically we have to calculate the organism which is there in the third trophic level, what will it pass to the organism of T4 level. So we know 20,000 energy is passed in the T2. Now T2 will be only giving, so we have to calculate this also for that. Then only if we calculate this, then only we can calculate this. So if T2 trophic level 2 organism is having 20,000 joules of energy, what will it give to T3 organism? Basically 10% of it. So 20,000 into 10 divided by 100. What do we have, children? 2,000. Okay, that is the energy that is given to T3. Now they are asking what amount of energy, calculate the amount of energy that will be transferred by the organism of third to fourth. So third is having this much, you are asking about T4. What will it get? So again 10%, again 10% of it, so it's just 200 joules of energy that is being transferred. So use the 10% law. Why? Where does all the other things go? Basically rest of the things are being utilized for its plants, for the organism's own need. Why my tongue is getting twisted? Listen to me very carefully, okay? I'm so sorry for that. So basically plants, they cannot give everything to the rat. Why? Because plants also have their needs, they will do their needs, and then what are the rest that they will give. But during this process of giving also, during energy transformation that is taking place over here, some amount of energy is lost as heat and stuff like that. So which is why we say roughly 10% of the energy is only given to the next trophic level, that is a 10% law. That is only 10% of energy that goes from one trophic level to the next trophic level. So in this way 200 joules of energy will be there for T4. Okay, T4. I hope you got it. And now if they are asking you what is there for the plants, so you have to do into 10 and then find for plants. That's all. Okay. I hope this is clear to you. It's a very simple question. Going ahead. Why does a kitchen garden called an artificial ecosystem while forest is considered to be a natural ecosystem, children? Kitchen garden, why is it a artificial ecosystem? Because first thing, human made or man made. Second thing, needs intervention of human being. Now Kitchen garden, obviously you need to put some water, you need to give all the things, all the good things, nutrients that could be provided, right? That is because of us that it is sustaining. It's not self-sustaining, it is human made, and it needs intervention of what? Human being. Whereas do you go to the Amazon rainforest to give them water? No, it does not have anything to do with us. We do not have to go anywhere. It is self-sustaining thing. Okay, so you have to talk about them also, then only you will get marks. So on the other hand, if we talk about forest, it is what, children? Not man-made. Okay, or at least if not man-made, if you cannot put that, you can say no intervention of man is needed. No intervention of human is needed. Agreed? Done? Can you think of that? Yeah. Now, while designing an artificial ecosystem at home, write any two things to be kept in mind to convert into self-sustaining ecosystem. So you want to make your kitchen garden and I would say anything artificial ecosystem which is there at your home, you want to convert it into natural one. What can you do to make it natural? What are the things that you should be keeping in your mind to convert? Basically, I'll make sure that the producers, the consumers all are in balance, right? So all the biotic factors, that is producers, consumers and all of them, and abiotic factors are in balance, especially the biotic factors. I'll say producers number and consumer number has to be more or less fine only. Okay, just imagine that the producer is way too much, consumer is not there. So producer will keep on increasing. So we need producer, and if you think producer is very less, consumer is very high, so that is also problem. So we need a balance. Okay, so we need a balance. Please listen to me, that will be better than reading it out. I know for sure. I am trying my best to make it readable, at least normal. Okay, are in balance. Works better. Producers, consumers, all of them in biotic factors. Okay, next thing that I will do is check for decomposers. I will add some decomposers, or else decomposition won't be happening. If decomposition won't be happening, the initial amount of, for example, what happens usually, there is a plant, this plant is eaten by a organism, this organism dies. Okay, whatever important nutrients are there, it is taken broken down by decomposers. Now it gives back the thing, and then the plants take this for making it up again, right? So we need decomposers. The role of decomposers is very much important for recycling nutrients. For recycling nutrients. So I will make sure of these 2 points. Okay, this is a 3-marker question coming from our environment, children. Going ahead. Okay, life processes, again 3-marker question. How does paramecium obtain its food? Tell me, how does paramecium obtain its food? Remember paramecium? So paramecium guy, it's a slipper shaped animal which looks somewhat like this over here. Okay, so basically paramecium has lots and lots of cilia around it. Okay, and it also has contractile vacuole. It has its own nucleus, some organelles and all of that. Okay, now listen to me very carefully. If there is a food material over here, this food is rolled with the help of what? The cilia, and last enter this particular position. This is known as oral groove. Oral groove of paramecium. Okay, now oral groove basically goes up, and to the end what do we have? Cytostome. That is the mouth of the cytostome, which is the mouth of the paramecium. So over here, this particular food is taken in, and now it opens into something like this, that is the food vacuole, and the food will be inside the food vacuole. This is what, children? This is the food vacuole. Okay, so first process will be what? Ingestion. The first step will be, where should I write it? I'll write it here. First step is ingestion, basically where the food is taken inside the body with the help of cilia, and it is put, so I am writing the important terms, with the help of cilia, it is put in the food vacuole. Okay, next thing that we have to write, ingestion, after that digestion, the food has to be broken down, right? So what will happen at this point of time? There will be another organelle which is known as lysosome. It will slowly go and fuse with it, release all its enzyme and breaks down the food. So digestion is where lysozyme I wrote, oh my god, lysosome releases an enzyme, breaks the food. Okay, now third step, what do we have? Ingestion, digestion, and then absorption. Absorption is when, okay, you have broken down things. Okay, so now inside the food vacuole things are broken down. You want to take in whatever important things are there into the cytoplasm. This is known as absorption. Absorption is basically important nutrients into cytoplasm. That is absorption. Okay, next thing, after absorption, what do we have? We have something that is known as assimilation. Assimilation. What do you mean by assimilation, children? Assimilation is basically, you have taken things in, but when our body is utilizing this thing for metabolic function, it is known as assimilation. Utilization for metabolic functions, that is assimilation. Okay, now fifth one, after assimilation we do not want anything. Now let's put out those things that is not needed. So basically this particular food vacuole with the waste will go and open up here, and this opening is known as cytoproct. Okay, so it will open up to release all the undigested food. So this is known as egestion. Egestion, where undigested food goes out. 5 steps for paramecium. Okay, how does paramecium obtain? You have to tell this. Agree? Now list the role of each of the following in our digestive system. Let's begin this. Hydrochloric acid, it provides an acidic medium. For 2 things. First thing, for killing the for killing the pathogen. I am writing here, killing pathogens. Second thing, why do we need acidic medium, children? They also need acidic medium for conversion of enzymes. For conversion of enzymes also we need acidic medium. Now talking about trypsin. Why do they need trypsin? Okay, trypsin is a, what conversion does it do, children? In the stomach there is pepsinogen enzyme. This is inactive enzyme. We need to convert into active enzyme, that is pepsin. For it needs a acidic medium. If acidic medium is not there, this conversion would not be taking place. Okay. Now trypsin again is an enzyme that breaks down proteins to peptides, and we need to break down proteins to peptides, right? We need to break down proteins to peptides, and then peptides are broken down to amino acids, and these amino acids are taken in my body for making new proteins. Okay, so we took some proteins from outside in the form of food, and we are going to make new proteins out of it. Okay, that we definitely need. Now muscular wall of stomach, why do we need it? For mixing of food and secretions of stomach. We want the food and the secretion of stomach to basically mix, the enzymes and everything to mix, and also for movement of food outside the stomach. Also for movement of the food outside the stomach. Now why do you need salivary amylase? We already spoke about this, but since it is asked, I will write here. Salivary amylase is there for conversion of starch to maltose. Easy question it is, no? You can answer, right? Okay, so for this thing, muscular wall of the stomach, as I said, for mixing, for movement of food, for movement of food and for breakdown of food also in some other way. Okay, physical breakdown of food. So you can write all of this in muscular walls of the stomach. Yeah. Going ahead, children, what do we have? What is double circulation? Okay, that has come at last. Why is the separation of right and left side of the heart useful? How does it help birds and mammals? What is double circulation? So basically, when in a cardiac cycle, blood moves twice through the heart, it is known as double circulation. Didn't understand? Ma'am is there to help you. Listen, so over here, just imagine there is heart, okay, left atrium, left ventricle, right atrium, right ventricle, okay. Now at this point of time, you have lungs over here and you have body part over here, okay. Lungs will be giving oxygenated blood. This oxygenated blood will be going to left ventricle and then the body parts, okay. The same blood, deoxygenated blood, comes to the right atrium, right ventricle to the lungs. So you see the blood has come once to the heart and it is going back to the heart to the lungs. Twice the blood has come. Now I can divide total double circulation into two parts. I can say double circulation is of two types. One is pulmonary circulation and the other one is systemic circulation. What do you mean by pulmonary circulation? Whenever the blood has to go from the lungs to the heart and back from heart to the lungs, this is known as pulmonary circulation. So the flow of blood between what? Lungs and heart. That is what you mean by pulmonary circulation. So it has come over here, oxygenated blood, and then deoxygenated blood is taken back to the lungs. Okay, this is pulmonary circulation. Systemic circulation is when oxygenated blood is given to the body and from the body system it is given back to the heart. So this is between body and heart. Can you write this? It's okay. It's okay if you write this. Please, 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 with the help of this particular flow charts and stuff like that, make proper lines like this and explain that this is the flow and here you have oxygenated blood, or you just write O over here and then inside you mention oxygenated and this D is for deoxygenated, so that people can definitely understand this, okay. And now, why is the separation of right and the left side of the heart useful? Because, how can you answer that? Do not want mixing of oxygenated and deoxygenated blood, which is why we do not want, we want a clear separation between right and the left side of the heart. Okay, going ahead. How does it help birds and mammals? How can this particular thing help birds and mammals? So, if mixing of the blood is not happening, that means the blood that is there, it will be rich in oxygen as well as over here it will be rich in carbon dioxide. Now, why do we need oxygen, children? Birds and mammals, especially why they are asking that, because birds and mammals are homeotherms. Homeotherms, or basically children, they are known as warm-blooded animals. Warm-blooded animals. What do you mean by homeotherms, ma'am? What is warm-blooded animals? Basically, the ones which are capable of maintaining a constant body temperature. For example, it's not like outside, if it's very icy, my body also will become ice. If it happens like that, probably we'll die. We need a constant body temperature anyhow, that is 37 degrees Celsius, because our enzymes are able to work in that particular temperature only. Okay, so birds and mammals, both of them, they are homeotherms, okay, the one that maintain a constant body temperature, or you call them warm-blooded animals. And to maintain a constant body temperature, children, you need much more amount of energy. and energy will be produced if more amount of oxygen is there. If the oxygen is getting mixed with deoxygenated blood, does our cell receive enough amount of oxygen? No. If our cells which are crying for oxygen do not receive enough amount of oxygen, will they be able to do cellular respiration? Will they be able to do cellular respiration, children? No. And cellular respiration is the one that gives us energy at the end. So you need, if you give O2, what will happen? Basically this guy which is sad will become happy. It will be able to do cellular respiration and will produce enough amount of ATP for maintenance of what? Constant temperature. Now, if the blood is getting mixed, you are not getting this oxygen, and none of this will happen. Okay, which is why we are homeotherms, warm-blooded animals, and we need this much amount of thing. Can you explain it in your own words? Birds and mammals are homeotherms or warm-blooded animals. They maintain a constant body temperature, which is for which they need enough supply of oxygen, which could help in the process of cellular respiration for the production of what? ATP. That will help in maintaining constant body temperature. It's as simple as that. Nothing to mug up. That is why I'm not writing over here. That is why I'm not writing over here. I don't want you to mug up things. I want you to understand things and frame it in your own words. Okay? That's how it should work. Works for you. I hope you got this answer as well. Now, what are those animals which are not, warm-blooded or homeotherms? Reptiles, amphibians, spices, all of them, they are what? They are cold-blooded animals. They do not care what temperature is. They go around bushes, and then if there is too much hot, they go around bushes. They stay there to make themselves, thanda. Okay, cool. Whereas if it's very much cold outside, they will go under the sun and try to bask under the sun, or go to some warm place and just be there. Okay, that's how they work. So they do not have much energy requirements. So the question can be opposite also. Why does the reptiles and amphibians does not have a clear separation between left side and the right side of the heart? Okay, if you have seen reptiles and amphibians, they have one right atrium, left atrium, and one ventricle where oxygenated and deoxygenated blood, everything mixes. So why they do not have? Because they don't need to maintain a constant body temperature. Their energy requirements are low. That's why. Can you explain like that? Yeah. Now going ahead, what do we have? Again a 3 marker. Where is the 4 marker? Where is the 4 marker? Okay, 3 more questions and then the 4 marker. Okay. Write the percentage of solar energy captured by the autotrophs. Yes, ma'am, I know, I'll tell. So basically, if this is sun, 1 to 5% of the solar energy is trapped by autotrophs. In your NCERT, it's given 1%. Okay, but it is 1 to 5. So you can write 1 to 5, but approximately it is 1%. Okay. Now what is the energy transferred from autotrophs to the next level in the food chain? From autotrophs to next level, trophic level 2. You tell me how much amount of energy? 1 to 5? No, we have the 10% rule. 10% amount of energy. Remember, autotrophs basically they are the lower trophic level, and then comes heterotrophs. Okay, so they are basically passing 10% of energy. This is 10% law. What are trophic levels, children? What are trophic levels? These are certain levels that organisms occupy in a food chain. Levels that organisms occupy in a food chain. Why do different food chains in an ecosystem not have more than 4 to 5 trophic levels? They do not have more than 4 to 5 levels because there is only 10% of energy that is being transferred. Now, how can you explain this answer? Basically, children, you have to be very careful. You have to give the example. For example, trophic level 1 has 2222. Okay, 2000 joules of energy. Now, 10% of 2000 will be what? 200. That will be passed to the other one. Here, it will be 20 joules, and then 2 joules. Is there any energy left to be passed down to the next trophic level? No. Which is why we have a limited trophic level, because as you go upper the trophic level, the amount of energy will be getting reduced, because the energy transformation that is taking place over here, it follows 10% rule. Okay, so can you give that answer very properly? Because only 10% energy is transferred from one trophic level to another, and you have to give an example like this, okay, where you have to mention, you can use the same example: plant, rat, snake, hawk. Okay, you can use the same example and then explain. Because only 10% another and to another, hence if trophic levels are more, energy won't be there, if enough amount of energy won't be there for getting it transferred. Okay, won't be enough to get transferred. You have to explain like that. I hope it works for you. Yeah. Now going ahead, what do we have? Explain the process of transport of oxygenated and deoxygenated blood in human body. We just explained it. If you want, I can just show you the brief of it. So over here, make a box like this. Left ventricle, right atrium, right ventricle. Make lungs in a box. Then mention this. This is what? Oxygenated blood. Then show this. Then show this to this to this. That is what? Body parts. And mention that this is what? Again, oxygenated blood. Now from here to here, make deoxygenated blood. Deoxygenated blood. From here to here, show an arrow. Then from here to here, show an arrow. And again mention deoxygenated blood. So left side of our body, heart basically, of our heart, not our body, is transporting oxygenated blood, whereas right side is transporting deoxygenated blood. How can you remember it? Lao, bring in oxygen, you can remember like that. Or Los Angeles, oh, you can also remember like that. Either way. Now you have to, whenever you mention this, you have to name the organ. Basically, the one that carries the blood towards the heart is obviously a vein. So it is carrying towards the heart, so it has to be a vein, blood vessel. Now the one that carries away from the heart, A for away, A for artery, it has to be an artery. Again, towards the heart, it will be a vein. Away from the heart, it will be an artery. Now wherever lungs is there, so associating to it, it will be always pulmonary. So this is a pulmonary vein, this is an exception. Pulmonary artery. Why it is not working? Okay. Pulmonary artery, that is also an exception. Okay, here artery, this artery's name is aorta, that is the biggest artery that we have. Okay, and this vein, it is vena cava. Vena cava. Explain in the form of writing also. Make this diagram and also explain in the form of writing whatever you are saying. Okay, I hope it works. Now, name the gland and the hormone secreted by it in a scary situation in human beings. So, the glands that are responsible for getting activated when you are in a scary situation. For example, a deer is coming and standing in front of you. Okay, bear, okay, bear is coming and standing in front of you. You will be scared. Why you will be scared? Because it's obviously going to kill you, right? So at this point of time, what is that gland that is going to release some hormones? What are these hormones also, they are asking. So you have to tell about our most famous adrenal gland, children. And adrenal gland, they are responsible for release of something that is known as adrenaline. It is known as adrenaline. So adrenal is responsible for release of adrenaline and also adrenaline. Okay, adrenal as in where are they located? For example, there is our kidney. On the top of a kidney, there is a triangular shaped gland which is known as adrenal gland. Okay, so adrenal as in near, renal as in your kidney. Okay, list any two responses shown by your body when these hormone is secreted in the blood. So children, when these hormone, you also call it you also call it epinephrine and norepinephrine also. Okay, so basically what do? They do two things. They are responsible, they are said to be fight or flight hormones. Fight or flight hormones. Why? It is responsible for fighting. Okay, if I see something, I will fight with the bear, or probably I will fly away, I will just run away. Okay, that is why it is known as fight or flight hormone. And what do? They increase the heart rate. Increase the heart rate so that much more amount of blood is transported, more oxygenated blood is transported. That is one thing. Okay, apart from that, it will also lead to increase in blood pressure. So heart rate, if it is increasing, blood pressure increases. Blood pressure is what, children? If this is a blood vessel, the amount of pressure that the blood is acting on the vessel, it is known as blood pressure. So too much amount of blood, heart rate is increasing, so blood pressure also increases. Okay, and vasodilation also occur. Vasodilation occurs. What do you mean by vasodilation? Basically, your blood vessels, some of them, they become dilated so that enough amount of blood would be going around. And some places, for example, you want the blood vessels of your muscle cells to be dilated so that you can run. So blood vessels of muscle cell will get dilated. Okay, whereas blood vessels that is going to your stomach and all, it will get constricted. Vasoconstriction. Kahan ka? Basically stomach and all. Okay, so you don't want everything to go to the stomach as of now, you want to run away, right? So you cannot be always thinking about your stomach and your food or whatever, even though I am very hungry right as of now and I am thinking about stomach, but still I am telling about that. Okay, so it is responsible for increase of heart rate, your blood pressure increases, vasodilation occurs in the muscle cells, okay, in the blood vessels of the muscle cells, vasoconstriction, which will get constricted in the stomach and things like that, so that blood can be redirected to the places where it is actually necessary. So this is something that you can remember. Right? Going ahead. In the given diagram, name the parts labeled A, B and C. Write the functions of A and C. Okay, let's label it first. So A is the one that is carrying the signal. So the one that carries the signal, children, it is known as sensory neuron. Sensory neuron. First thing, it is also known as efferent neuron. It is also known as efferent neuron. Now, B is somewhere in here. This is your spinal cord. Okay, so basically B is inside your spinal cord. So B structure that I see over here, I call it relay neuron, or it is also known as interneuron. It is totally within the spinal cord. Okay. Now going ahead, C. What is C, children? C, they have mentioned it to be this. What is C over here? Don't say it is an effector organ. No. It is an effector, motor neuron. No. See, they have mentioned it to be an effector organ. So you have to say effector organ. That is basically muscle over here. So they didn't mention this. Instead, they mentioned this thing. So this is the effector organ. Please do not make mistakes like that, just like how I was going to do. So write the function of A and C. A is responsible for carrying the information, carrying the info from receptors, sensory receptors basically, from sensory receptors to where, children? To interneuron. Okay, agree? Now, what is the use of C? Effector receives the signal from motor neuron and undergoes contraction. Undergoes contraction to pick away her hand away. Okay. Reflex arc have evolved in animals. Why? So reflex arc, children, why do you think it has evolved in animals? It has evolved because you cannot depend on your brain. For example, there is some hot thing over here. Okay, very hot thing. Now I am going to put my hand on this. I cannot think like, should I take away my hands or not? If I think like that, probably it's gone. So if I touch something hot, I immediately have to put away my hands because it will increase my survival. So to increase survival rate, we cannot depend on our brain to think and take the action. Brain to think and take the action. Hence, reflex arc has evolved. Can you explain that? That's all. It's as simple as that. So reflex arc basically uses your spinal cord. Information does not go directly to the brain and then come back. It's not like that. It goes to the spinal cord and now information is given to your brain as well, but the spinal cord is the one that makes the quick response. Okay. Information is done that something of this thing has happened, next time probably it won't be happening. We won't be doing something like that. We won't be going and touching a hot pan. We would be very careful. So that is the thing. Okay. And with this, children, we are completing the 3 marker questions as well. So again, I would like you to take a break, and because we would be solving 6 4 marker question and then again almost 10 to 12 5 marker. I think 10 of them. Yes. So we have to do that. So for that we need a break. So if you want, you can go quickly revise whatever you have learned, or else you can just wait for some time. Yeah, and have a quick chill and then come back. So I will also have a quick chill and come back. Okay. Okay, champions. Let's begin the 4 marker case study based question, which has to be interesting. So let's see. In one of his famous experiments of tracing the inheritance of trait, when Mendel cross-pollinated a pure tall plant, pea plant, with a pure dwarf pea plant, okay, he got only tall pea plants, okay, in F1 generation. Okay, all of them, they were tall. Fine. When F1 tall pea plants were self-pollinated, Mendel got F2 generation pea plants in which both tall and dwarf pea plants were present. List any 2 pairs of contrasting characters other than those mentioned above used by Mendel in his experiments. So basically he chose pea plant because it has 7 contrasting traits over here, but if they are asking for something, let's see. So one is the shape of the seed, round and wrinkled. Okay, this is shape of seed. And if you want color of seed, basically yellow and green. These are the 2 contrasting traits. Okay, works. Now differentiate between dominant and recessive trait. What do you mean by dominant trait and what do you mean by recessive trait? So dominant trait are the ones, okay, which will be shown up in an organism. Basically, dominant trait are the ones which will be the phenotype of an organism. Or basically, rather than that, it will be wrong to say that dominant trait will still be visible if there is only one allele of the trait, if there is only one allele of the trait that is present. Okay, that means if capital T is there, then obviously it is present, but only if one allele is there, that is capital T, then also this particular trait will be shown up. So dominant trait will be the one will be visible if there is even if there is only one allele of the trait that is present. Now talking about recessive trait, is the one, okay, where two alleles have to be the same, and also it is the one that won't show its trait, that won't show the trait if a dominant allele is present. If a dominant allele is present. So basically it is the one where both the alleles have to be the same. So in this case also it is the same. So how can you differentiate between that and this? So it can be present only if the alleles are same, and also it won't be present if a dominant trait is present. Okay, that is what you mean by recessive trait. Fine. Now state the ratio of the tall pea plants and the dwarf pea plants in F2 generation. Give reason for appearance of dwarf pea plants in F2 generation. Okay, so we have to tell the ratio of tall pea plants, dwarf pea plants in F2 generation, and give reason for the appearance of dwarf pea plants in F2 generation. Chalo, let's begin. So starting, okay, over here they said you have parents, and these parents were pure tall, pure dwarf. Okay, so pure tall as in both of them will be having capital T, capital T. Pure dwarf as in both of them will be having small t, small t. This is what you mean by pure. It is homozygous. Whenever they are same, the alleles are same, children, you call it homozygous. Okay, now you have capital T, small t, small t. Now let's see the gametes. So gametes will be either it can be T or it can be T. Both of them obviously would be giving the same gametes. Similarly, it can be T. What do you mean by all this thing? Basically, just imagine if there is one chromosome and there is another chromosome. One is having capital T, another one is having capital T. Either of them, only half of the chromosomes gets carried in the gamete. If this can come or this can come, either way it's capital T. Over here also it will be either way small t only that will be coming forward. Now gametes are there. Now let's see F1 generation. So over here capital T fuses with small t, you get capital T, small t, this is tall. Okay, capital T fuses with small t, again capital T, small t, this will be tall. Capital T, small t, it will be again tall. Capital T, small t, again it will be tall. So all of them, children, are tall. Okay, and this, okay, this particular thing that you cannot see, you say that this is the genotype of the plant. That this particular plant which will be there, it is genotype, the thing that you cannot see. Tall, this is a character that you can see. This is the phenotype of the plant. So if you are asked what is the phenotype of the F1 generation plants, all of them are tall. If you are asked about genotype, all of them are capital T, small t. Okay. Now going ahead, let's talk about what the question is. Now this time the parents are them, okay, and now we are going to self-pollinate them. So let's make the parents. Okay, so parents, children, are capital T, small t, and capital T, small t. Now this time imagine one chromosome, another chromosome. One chromosome has capital T gene and the other chromosome has small t gene. So either one of them can be passed on. Okay, so gamete, if I see, it can be capital T or it can be small t. Here also capital T, it can be small t, right? Now F2 generation, this is filial 2 generation. This was first generation which arise from your parents. Now the children that have arise, they are fusing. Now what will happen? This and this fuses, you get capital T, capital T. This is tall, okay. This and this fuses, you get capital T, small t. Again, this is a capital T over here, so it's a dominant trait, so it will overshadow the other one. Here you see, you have capital T, small t, again tall, and then capital small t, this will be dwarf. Now you see, 3 of them they are tall, 1 of them they are dwarf. 1, because only one has 2 small genes, okay, you call it homozygous small t. So this will be only the dwarf one. Now if you are asked, tell me the phenotypic ratio. Phenotypic as in how do they look, and ratio if you want. So 3 appears to be tall and 1 is dwarf. So phenotypic ratio, children, phenotypic ratio is what? 3 tall and 1 is dwarf. The dominant one appears over here always. Don't write 1:3. Okay, so this is tall and write like this, dwarf. Now if you ask about genotypic ratio, the one that you cannot see. So over here 1 is small tt. Okay, so there is 1 small tt, 2 of them with capital T small t, 2 of them, and 1 with capital T capital T. So you tell me the ratio. 1:2:1. That is the genotypic ratio of the F2 plants. Phenotypic ratio is this. Did you get it? That is what they were asking. State the ratio of tall pea plants and dwarf pea plants in F2 generation. This is the tall and the dwarf one. This is what they were asking. They were asking about the phenotype, tall and dwarf. They are asking about genotype. Anyways, you have to write this and this both. Please write both of them. Okay, please write both of them and mention like this. Okay. Now one more thing, they are asking, give reason for the appearance of dwarf pea plants in F2 generation. So you have to tell why dwarf pea plants are appearing in the F2 generation. This is a 2-mark question. This is 1 mark each. Okay. So 1 mark you got for this if you got it correct. You don't have to make the entire thing. You have to start from here. This is for your explanation that I made. You have to start from here. It will take hardly 2 minutes. Okay, that's what I think. Now, okay, give one, okay, and you have to please mention gametes, parents, F2 generation and everything, okay. What is the reason basically, what is the reason basically why dwarf plants is visible? Because over here, reason, okay, there is no dominant allele to overshadow, or basically you can say okay, you can rather say why it is visible, why dwarf is visible. Instead of writing this, you should be saying because homozygous recessive allele have come together, and there is no, okay, that's enough. I think that's enough rather than writing the whole Ramayana there. Okay, because there is the homozygous, as in 2 same ones, recessive alleles that have come together, which is why you can see dwarf trait. Okay, which is why you can see dwarf trait. And also you can say because the parents that were fusing, they were not homozygous but they were heterozygous. Parents were heterozygous tall. They were not homozygous tall. Homozygous tall as in this. Okay, so all of them, they are heterozygous tall, which is why you got this thing. If they were homozygous, probably you would not have gotten this particular allele. So 2 reasons, because homozygous recessive alleles have come together, homozygous as in 2 same type of recessive alleles have come together, and parents were heterozygous tall as in this. They were not like this, instead they were like this. Agree? You can write the answer like this and explain. Fine, this is from heredity. Going ahead. In order to trace the inheritance of trait, Mendel crossed pea plants having one contrasting character or a pair of contrasting characters. When he crossed the pea plant having round and yellow seeds with pea plant having wrinkled and green seed, he observed that no plants with wrinkled and green seeds were obtained in F1 generation. Okay, when F1 generation pea plants, so he is saying he took 2 plants. Okay, one was round yellow and then wrinkled green. This is the shape of the pod and this is the color of the seed, okay? Same thing, shape of the seed and the color of the seed. When they crossed it, he found that all of them, they came out to be round and yellow. And none of them were wrinkle green in F1 generation, okay? But then when F1 generation plants were crossed, something like this, they were crossed, okay? It was found that there were other combinations also occurring there. Write any two pairs of contrasting characteristics of pea plants used by Mendel other than those mentioned above. So apart from that, what do we have? We have the height, which we mentioned in the previous one, that is tall and dwarf. Apart from that, you have the color of the flower. Color of flower, what is it, children? It is either purple or white. So purple one is the dominant one, okay? Purple one that you see is the dominant one. Let me see the time, okay? So you have to know about that as well, okay? Going ahead, what do we have? Differentiate between dominant and recessive, we already covered it. State the ratio of combinations observed in seeds of F2 generation. What do you interpret from this? So we have to make the F2 generation. For that we have to make the F1 also. Let's see, let's make it. It's very simple, listen to me very carefully, okay? So the parent, he said it is round, yellow, and it is wrinkled green, okay? Now round yellow, parents as in, for round you need RR, that is a dominant trait, that's why round yellow is visible, okay? So round, for that you have two alleles RR. For yellow also you have two alleles YY, both of them they are dominant. Wrinkled, for that they are recessive, so small RR, and green, it's also recessive, so small YY. Always use the dominant trait as the letter, first letter, okay? Don't write GG there, YY will be the same thing. Now gametes that will be formed over here. So now you see, it can give either this chromosome or this chromosome from this pair, or this or this. Either way, RY will be there. Either way, one R and one Y will be there coming from, however you do. Similarly over here, R and small y will be coming over here in the gametes. Okay, one R and one Y will be coming, one small r and one small y will be coming. Now, if you see the F1 generation, okay, this gamete which is having R and Y is fusing with this gamete which is having R and Y. So you have to think like this. So there is a chromosome with R, there is another chromosome with Y, okay, that is coming, and over here also same thing. So when they have come together, now in this F1 generation, all of them will be R, small r, Y, small y. Chromosome from this end and this end and come, and the dominant one is appearing over here, the recessive one is appearing over here. All of them, they are children, round and yellow. This is what exactly they said over here. All of them were round yellow in F1 generation. But in F2 generation, there were different contrasting states, and you have to tell the different contrasting traits that you observed. So in F2, if you want to make the gametes, how can you make the gametes? You only think, there is one chromosome like this, one chromosome like this, one can have R, other can have small r. Okay, one has Y, other one has small y. So either this chromosome and this chromosome can go to the gametes, or this chromosome and this chromosome. So if they both go, okay, so there is a combination of R and big Y. Okay, there's a combination of R and big Y. There's a combination of R and small y. There's a combination of small r and big Y, and there's a combination of small r and small y. Either this and this can go, either this and this can go, half, anyway one of it will be going from all the pairs. So either this or this can go, either this or this can go. Whatever possibilities are there, I have written over here. Similarly, you have to write on this side also, RY, RY, RY and RY. So I will just explain the one over here, rest of the things you have to find out, okay children? Okay, now listen. If I fuse this with this, I get R, R, Y, Y. If I fuse this with this, I get R, R, Y, Y. If I fuse this with this, I get R, small r, Y and Y. Okay. If I fuse this with this, I get R, R, Y and Y. All of them, if you see, they are round yellow, round yellow, round yellow, round yellow, because this is dominant. So both of them are dominant, round yellow. This is dominant, round. yellow. Why? Because Y is dominant over small y. So this will be yellow. Similarly, R is dominant, so it is R yellow. R is dominant and Y is also dominant, so it is R yellow. All of them will be round and yellow. If you match the form, you will get almost 16 of them over here. Okay, you will get almost 16 of them, not 16. Yeah, 16 of them will be over here. Now, how can you cannot go around in your exam to find all these things, so you have a trick to remember. Okay, the ratio, phenotypic ratio is 9:3:3:1. There it was 3:1. Here it is 9:3:3:1. How can you remember? 9 of them, they are the dominant traits. So all of them will be round yellow. What type of round yellow? It can be this, any genotype will be there, but it will be round and yellow. One will be the recessive one. What was the recessive one, children? Wrinkled green is the recessive one. So the highest one is round yellow, the lowest one is wrinkled green. In between you have 3 and 3, you mix and match. So he said new combination was found, right? So round and green, that is a new combination. And the other one will be wrinkled and yellow. So this is a new combination. So 9:3:3:1, that is the thing. Okay, that is the answer for this question. That is, state the ratio of combination observed in the seeds of F2 generation. What do you interpret from this result? What amount of information do you get from this result? Tell me. The information is that basically the alleles are moving separately. The alleles for one gene, it's not saying if it is round then it has to be yellow only. It can be different also, it is round green also. So alleles for one character, alleles for one character is moving on independently from the alleles of other character, and this is known as independent assortment. It is independently assorting. So the round has nothing to do with the yellow. It can go with the yellow or the wrinkled one. It's the same thing. Just alleles have to pass. So alleles which are passing down, they are not best friends. They can move independently, and this is known as law of independent assortment. Okay, law of independent assortment. So when two characters are there, two alleles are there for two characters, they are moving independently. This is what you learned. Otherwise, all of them had to be round and yellow or wrinkled and green. But it's not like that. You get different combinations. Okay, so that is the thing. I hope this answer is also clear. Going ahead, the most obvious outcome of reproductive process is the generation of individuals of similar design, but in sexual reproduction they might not be exactly alike. The resemblance as well as differences are marked. The rules of heredity determine the process by which traits and characteristics are reliably inherited. Many experiments have been done to study the rules of inheritance. Okay, why an offspring of human being is not a true copy of his parents in sexual reproduction? Why is he not a true copy of parents? So for that you need to understand sexual reproduction. So over here, parent 1, parent 2, parent 1 has their own DNA, parent 2 will also be having their own DNA. Okay. Now, when a baby has to be born, you give your DNA to the baby. Okay, but you do not give your DNA, you give a copy of it. So while copying mechanism happens, so for that the DNA opens up like this. Okay, on the basis of one strand that is over here, on the information that is present, they make a new strand. Similarly, on the basis of this strand, they make a new strands. Later, this particular thing that you see, they formed up, and this is the copy that you made, okay, of your DNA. Similarly, parent 2 will also give its copy. But during this copying mechanism, you call it DNA replication. Okay, during DNA replication, where the parent is giving, I'm not writing down for you, just listen to me carefully. During DNA replication, when the parent is giving a copy of its DNA, there will be errors. There will be errors that will be made, children. Some errors which would lead to variations. Okay, so errors during DNA copying mechanism during sexual reproduction, that is one thing. And also the second reason why we are different from our parents is that we are getting two DNA, two very different DNA. So we can say from parent 1 and parent 2 we are getting two separate sets of DNA, obviously with their own differences which is coming together, which would lead to more amount of variation. But in asexual reproduction there is only one parent, so what is coming and fusing? Nothing is coming and fusing. So only the DNA replication errors being made during asexual reproduction, that is happening in them. That is the root cause of variation. But for us, this is one reason. This is the second reason, that we are getting two separate DNA from both of our parents, which is why we are different from our siblings as well. Okay. So siblings, why we are different? Because of the errors that is during appearing to the variation and the different combination of genes that are going to combine together. Even though we have the same DNA in some or the other way, but not the exact similar thing. Okay. Now, while performing experiments on inheritance in plants, what is the difference between F1 and F2 generation? Children, F1 is the filial 1 generation. Okay, you call it filial 1 generation, basically the first generation which is got from the fusion of parents. Now, this F1 generation, when they undergo self-pollination, you get F2 generation, that is filial 2 generation. Okay, you can explain with the help of an example, capital T, small t, and that one. Okay, why do we say that variations are useful for survival of a species over time? Why do you think variation is useful, children? Because atmospheric conditions can change. Agree? Atmospheric condition can change. For example, just think I have a variation which could help me thrive through very hot weather, but others do not have this variation. Okay, me and someone else and few more else have this variation, but others do not have this variation. Obviously, when hot weather come, I will be able to survive, my friends would be able to survive, but others who didn't have this variation, they won't be able to survive. So atmospheric conditions basically can change. Okay, atmospheric conditions can change and it will help me survive during everything. That is one thing. I'll write it properly. Atmospheric conditions can change, which lets me survive during anything. So that is why we say variations are useful for survival of species. You can give the example of bacteria. One bacteria has the variation, other bacteria does not have the variation. Very hot climate is there at this point of time. The one that is having the variation would survive, whereas other won't. Now, if this would survive, obviously this can go ahead with reproduction and give rise to more of its own kind. Okay, such that all of them can survive during any bad condition. Now, they will also have variation which will help them survive during anything else that comes to the way. Okay, so this includes both human reproduction as well as not human reproduction, how do organisms reproduce, and heredity, both of them. Okay, now, all human chromosomes are not paired. Okay, most human chromosomes have a maternal and a paternal copy, and we have such 22 such pairs. Okay, but one pair called the sex chromosome is odd in not always being a perfect pair. Women have a perfect pair of sex chromosome, but men have a mismatched pair. In one is normal sized while the other is a short one. In humans, how many chromosomes are present in the zygote and in each gamete? So they are asking you how many chromosomes are present in the zygote. So listen, and the gamete. So let's start with the gamete first. So this is male gamete and this is the female gamete. Female gamete will be having 23 chromosomes. Male gamete will also be having 23 chromosomes. Okay, this is about the gamete. So you got the answer for gametes. Now, fusing together you get the zygote with 46 number of chromosomes. They are asking how many chromosomes. So 46 number of chromosomes. If they are asking how many pairs, if they are asking about pairs, then how many pairs? Then it is 23 pairs. Okay, number of chromosomes they are asking, so it is 46 number of chromosome. Fine. Now, a few reptiles rely entirely on environmental cues for sexual reproduction. Comment. So basically there are reptiles like turtles. Okay, now turtles, they work on a principle of temperature dependent sex determination. Okay, where they put their eggs, if they are put in warmer region, it will be female. If it is put in cold region, it will be male child. Okay, why does this work? Basically temperature dependent, as in during warm temperature probably certain enzymes would be active which would help in the formation of certain hormones, for example female hormones and stuff like that, which is why it could come into a female, but it's not entirely just because of hormone, also because of genetics. So that's a very twisted form that happens, but it happens generally, and this is known as temperature dependent sex determination, shown by turtle as well as the crocodiles and everything that comes under reptilian category. Okay. Now next thing, see, the sex of the child is matter of chance and none of the parents are considered to be responsible for it. Justify it through a flow chart. So what is the thing that we explained over there? I think before some time we explained about we spoke about this thing, no? I don't remember where I left it. Okay, anyways, we spoke about the male and the female X chromosome and Y chromosome. You have to just write about that. Okay, I'm not explaining it to you again because it won't be of any use rather than wasting time, but yes. Where are we? Yes. So you have to say XX and XY. So it's a matter of chance that the parent can give you X or Y. Anyways, it's not anyone's fault that it's not under their control, it's under the body's control. That happens naturally. So the male can give one X or it can give one Y. Okay, if male is giving X and the female is giving X, it will be a female child. If the male is giving Y and the female is giving X, it will be a male child. So it's none of their fault, it happens like that. Okay, now that is one thing I wanted to tell. Apart from that, I wanted to tell you something I forgot. Okay. Yeah, the small chromosome that they are mentioning over here, the shorter one, it is Y. Always remember the shorter one is a Y chromosome. Okay. Now going ahead, plantation drives are often carried out especially during monsoon season for protection of our environment. Such programs need a lot of saplings for tree plantation. Plants are propagated by sexual or asexual means in the fields and nursery. So chemical reaction. Okay. Over the years, horticulturists have developed asexual methods that are vegetative parts of the plant to multiply. Many plants can reproduce by this method naturally as well as by artificial means. Okay, which specific part of the plant is used for sexual and asexual means of propagation to produce a new plant? For sexual, what part is used, children? It is the flower, because flower is the one that will be responsible for the seeds and everything. Okay, now for asexual parts, what do you need? All the vegetative parts. All the vegetative parts. What do you have as vegetative parts? Root, stem, leaves, all of them, they come under this category. Okay, this is from how do organisms reproduce? Organisms reproduce. Okay, now going ahead, give one example of a flower and a fruit grown by vegetative propagation. So flower, rose, flower, rose. Fruit grown by, I can say, strawberry, strawberry. No, basically it is done with the help of layering. There is a process known as layering where you take the branch which has the roots and you put it down in the soil and you cover it. Okay, that is one example of layering that is done by strawberry. Rose, you do cutting and then you put, or else you can basically use jasmine also for layering. Okay, it's the same thing. You can give two examples. Now list two advantages of growing plants by vegetative propagation. I already discussed it. One is it is giving you faster results, and also it will be giving you more amount of desired traits. Okay, because you are only choosing your plants, the one that is best, and then you are planting them with the help of vegetative propagation. So that is the two thing that we already discussed. Going ahead, all the reproductive methods of living organisms are broadly categorized into two types: asexual and sexual. Asexual reproduction involves the participation of a single parent without the formation of gametes. Fertilization and transfer of genetic material. This method is common means for rapidly increasing offsprings under favorable condition. Name the type of fission that occurs in leishmania and plasmodium. So leishmania, children, it is the one that causes kala azar. Okay, and over here they are going to show longitudinal binary fission. binary fission. So what happens in binary fission? Basically there is an organism like this that is known as leishmania, which has cilia, sorry, flagella over here. Just a second. Yo, everything gone. Okay. Something like this. It divides from middle. This is longitudinal axis into two leishmania. So this is longitudinal binary as in two. It's dividing into two. Okay. Now plasmodium children, it shows multiple fission. That is basically, if this is a cyst of the plasmodium, the nucleus keeps on dividing, and later on the cytoplasm covers it. So you have so many chotu plasmodium coming around. So this is multiple fission. Okay, that is different from binary fission. Binary as in one body divides into two, you have two organisms. Here you have so many. Now write one advantage of sexual mode of reproduction over asexual reproduction. Give reasons. Okay. 1, variation is high. That's enough. Variations are high in the case of sexual reproduction because two parents are involved, and also because of the errors during the DNA replication method. Give reasons why colonies of yeast fail to multiply in water but multiply in sugar solution. Okay, why do they multiply in sugar solution, children? Because sugar solution provides energy, and if energy is not there, can they multiply? No. So if energy is there, they will multiply, they will show budding and everything. Rhizopus individuals do not grow on dry slice of bread. What is rhizopus? Bread mold. Okay, why does it not grow on dry slice of bread? Because it needs moisture, for formation of hyphae. So usually it's in the cyst stage, right? It's in the spore stage. The spores, when they find a suitable area with a moist climate, that's only when they will form hyphae. These hyphae is going to form the whole structure of the rhizopus, okay, the bread mold. So for moisture, it needs a very wet thing rather than a dry thing. That's done. Now let's get into the 5-marker questions, my dear little champions. Name the disorder which a person is likely to suffer from due to the following reasons. Chalo. Over secretion of growth hormone. So over secretion of growth hormone, it will lead to gigantism. Okay, where they will be too big. Now deficiency of estrogen in females, it will lead to masculinity, but if they are asking about a condition, I will say female hypogonadism. Gonad is basically the ovary. Okay, hypo as in less. So basically your ovaries are secreting less. This is why this is happening. Okay, estrogen is secreted by the ovary itself. Now if this is happening, what are the problems? Basically you won't be having the secondary sexual characters which are probably usually there. Okay, so there will be lesser chances of all this thing happening, as in there will be no proper breast formation and stuff like that, which should be happening normally. Less secretion of thyroxine. Hypothyroidism. Now if hypothyroidism is there, you will be having less your metabolic rate will be really low. Okay, your metabolic rate will be really low because thyroxine is the one that handles your metabolic rate. Okay, also name the gland that secretes each of the hormone mentioned above. Chalo, let's see the gland. So growth hormone is released by pituitary gland in your brain. Okay, hypogonadism, what is it? Estrogen is released by ovary. Hypothyroidism, thyroid gland releases the secretions. Okay, going ahead. List 3 points of differences between nervous and hormonal mechanism for control and coordination in animals. How are auxins related to bending of plant shoots towards unidirectional growth? Chalo, I'll explain this first. The other answer I've already written for you. Okay, so you see, there's a sun over here and we have the plant over here. Okay, plant generally has a uniform distribution of auxin. So I'll write somewhere over here, uniform distribution of auxin. Okay, now what will happen? Since sun is there this side, okay, I have a little bit of changes to make. Yes. Okay. Now what will happen? Sun is there this side. So the oxygen, no children, it will move to the other side. Oxygen will be moving to the other side. Now there will be too much amount of concentration. Oxygen moves to the free side. Free side or the shaded side. Okay. Now what will oxygen do over here? If oxygen is there, what will it do over here? Oxygen will now, oxygen causes cell elongation on one side. Now if there is too much amount of oxygen over here, it will lead to cell elongation. So basically it will look like this. So your cells which were like this, chotu chotu, it will now be bigger this side. If it is bigger, obviously if one side is longer and the other one is shorter, it will tend to bend towards that side. So your sun is over here and is tending to bend towards that side because there is an irregular cell elongation on just one side. Because of the irregular cell elongation on one side, it will slowly bend over there. You have to draw this thing and explain it to them. Okay. Now list 3 points of differences between nervous and hormonal mechanism for control and coordination. Over here I've written speed. The nervous system is faster than the hormonal one because you are using nerve impulses. Okay. Signal type. The nervous system uses electrical impulses whereas the hormonal system uses chemical signals. Okay. Fine. So where there is formation of ions and everything there. Effect duration. The nervous system's effects are short-lived. Yeah, sometimes it can be more, but the hormonal system effects are long term. It will stay for much longer time. Okay, can you write these 3 things over here? These are the 3 points that you have to write. Going ahead. Given below is a diagram showing cross section of a leaf. Name the parts marked as A and B and write one specific function of each. So this is A and this is what, children? This is B. You have to write one function of each. Okay. This thing that you see over here, this is the palisade mesophyll. Palisade mesophyll, which has so much chloroplast and hence it will increase photosynthesis. Okay. Next thing, this thing that you see over here, yeah, it's a stomatal pore. Okay, stomatal pore, it allows diffusion of gases and what? And transpiration, water to move out. Fine, that is the use of B. Now, name the 2 tissues present in the vascular bundles. In what way does each of them help in a plant? So, the tissues that are present in the vascular bundle is basically xylem. Xylem help in transportation of water, okay, and phloem, translocation of food. It helps in translocation of food, children. Flow for phloem and flow for food. Now next thing, in the experiment light is essential for photosynthesis. Why does the uncovered part of the leaf turn blue black after putting iodine solution on it? So over here, imagine that there is a leaf, okay, and they have covered the whole part of this. So this is covered, this is uncovered. Now uncovered part obviously will receive sunlight, okay. It will do photosynthesis. At the end of photosynthesis, what do you get, children? C6H12O6, which is the glucose, and this glucose is stored in the form of starch. Agree? Now, if I put iodine solution over it, okay, this particular leaf will be turning blue black in color because of the presence of starch. So when iodine is made to react with starch, it will give a blue black coloration. Okay, that is one thing that you need to remember. That is why over here, if you see this particular part which is covered, no starch is there, so obviously it won't turn blue black in color. Okay, going ahead. Chalo. Next question. Study the following chart, flow chart showing the breakdown of glucose by various pathways. In this flow chart, in 4 blocks, some blanks are given as A, B, C, D. A and then B, C, D. Okay. Write the names of A, B, C, D. Chalo. Right. Let's write the names of A, B, C, D. So glucose to what? It will be pyruvate. Is it visible? Yes. I should write with black. I'll write with black. Okay. Now, in the absence of oxygen in yeast, what is produced? CO2, energy, and what? What is B over here? Ethanol, children. It is ethanol. C, again, in the absence of oxygen, in the case of muscle cells, lactic acid and energy is being produced. So you have to mention that. Now don't write this absence of oxygen and come here. Please write muscle cells. Okay. Now presence of oxygen in D of a cell, this will happen. In the presence of oxygen, what will happen? This will be produced. But where is it happening? In the D of the cell. So what is it? D, mitochondria. In the mitochondria, aerobic respiration takes place. Now write the source of glucose molecule involved in cellular respiration in plants and animals. So in plants, the source will be what, children? The starch, which will be broken down. In the case of animals, what will be it? It will be glycogen, again, which is broken down to this thing. Okay. Why do athletes sometimes suffer from muscle cramps during running? You know the answer. I told you already, lactic acid. During running, at this point of time, there are certain conditions where the amount of oxygen is less. So our body shifts to anaerobic respiration. Where it will be giving lactic acid. Where it will be producing, children, lactic acid and ATP. So this lactic acid is the one when accumulated gives cramps. Give reasons why the rate of breathing in aquatic organisms is faster than the terrestrial. Because in aquatic organisms, no, children, the thing is so less dissolved oxygen is there. Okay, there is so less dissolved oxygen, which is why they have to breathe faster. They have to take in faster. Okay. And breathing is basically taking water in them. So they are taking in water with oxygen, and this has to be fast because there is little amount of oxygen in every type of breath that they take. That's the problem. This is why they have to breathe a little faster. Okay, that is the thing. Now going ahead. Name and explain the 2 modes of asexual reproduction observed in Hydra. So you have to talk about budding. So for that, make the diagram of the Hydra. Okay, make the diagram and then show that the small bud being formed, which forms into a big bud later on and then detaches, which is known as budding. Okay, so cell there are regenerative, there will be so many regenerative cells over here, which will be giving responsible for forming a new bud and then later a new hydra. Now there is one more function, which is, this is budding. Okay, the other one is fragmentation. Fragmentation, where the body of the hydra is getting divided into 2 and then from each fragment a new hydra is coming. Again, you have to make the diagram and then explain it to them. So from this part a new hydra comes, from the lower part a new hydra will be coming around. That is what you mean by fragmentation. So from each lost body part a new one is coming with the help of a process which is known as regeneration. Okay. Now what is vegetative reproduction? We studied about it. List 2 advantages, we studied about it also. So I am not explaining it again. Going ahead, name the parts of bisexual flower that are not directly involved in reproduction. So you know, if it's a flower, it's a bisexual flower. Basically flower will be having some things like this. So this is the place where the ovaries they rest. Okay, and then you will be having the anther and the filament, and then you will be having, children, the petals, okay, and the sepals. So they are asking you parts of the bisexual that are not directly involved in reproduction. It will be petals, it will be sepals. If okay, that can be involved. Bisexual as in when you have two of them, when both gynoecium and androecium is present. Differentiate between self-pollination and cross-pollination. Tell two significance of pollination. Okay, what do you mean by self-pollination? When pollen from a flower moves to the stigma of the same flower or flower of a different flower or different flower of a same plant. So basically, if this is a plant, it has two flowers over here. So self-pollination is when if it has both the stigma as well as the if it has both the pistil as well as the stamen, from here to here if it goes, that is self-pollination. Also from here to here if it comes, that is also self-pollination. Now cross-pollination, children, is when pollen from anther moves to stigma of another flower of another plant. So in your NCERT it's just written another flower, they didn't mention another plant, but that is not the case, it is also another plant. So from here it is going to another plant. So this will create to more amount of variation. Okay, just like how one human and two human, similar to that. This is known as cross pollination, children. What is the significance, two significance of pollination? Pollination would lead to the continuation of the species, right? Pollination would basically lead to the continuation of the species. Pollination is not there, it will be difficult, right? And it will also continuity of species also lead to variations. Also lead to variations. What is the fate of ovules and ovary after fertilization in a flower? So what happens to the ovule and ovary, children? Listen, if this is the stigma, okay, this is the ovary, and then here you have the ovules. Basically the ovary, it becomes the fruit. Okay, just think about this thing. It becomes the fruit. The ovule that you see, it becomes the seed. What is it, children? It is the ovule. Now inside the ovule, the male and the female gametes fusion, and hence what will be there inside the seed? Inside the seed you will be having the embryo, which is why we say we put seed and then a new plant will come in, because seed will have embryo. Okay, so ovary becomes the fruit, ovules becomes the seed, and then inside the seed there will be embryo. Works? So can you tell that? That's what is asked over here. Going ahead, name the two types of pollination and differentiate between them. We just spoke about them. Okay, so yes, next thing, explain the post fertilization changes that occurs in the ovary of the flower. Same thing that I mentioned over here. The ovary becomes the fruit, the ovule becomes the seed will be having what? The embryo. Okay, given below is a diagram of germinating seed. Label the parts that give rise to future shoot. A is the one that gives rise to future shoot, it is known as plumule. Okay, gives rise to future root system, it is the B, which is known as radicle. Okay, plumule, radicle is the one that gives rise to root, R for root. And then stores food, this thing C is the one that stores food, it is known as cotyledon. So inside the seed, this is the condition. So you have cotyledons which would be providing nourishment, and then plumule, and then radicle. So you eat, sometimes people eat the seeds, right, because it has the stored food for the embryo to grow actually, that is cotyledon. But we do not leave anything, we eat everything. So basically we have this. Okay, going ahead, children, name the organs that produce sperms as well as secrete hormones in the case of human male reproductive system. Okay, testis is the organ that produces sperm as well as the hormones which are none other than androgens or testosterone. Name the part of the human female reproductive system where fertilization occurs. So, if you talk about human reproductive system, it looks like this. Okay. Fertilization occurs over here, which is known as the fallopian tube. So, this is the site for fertilization. Where does implantation of the zygote take? What do you mean by implantation of the zygote? Basically, the zygote has to come and embed over here. It's not the zygote that comes, zygote is being formed here. So, egg and the sperm fuses, you get the zygote undergoes division, now it's known as an embryo. When it's more than one cell stage, you call it an embryo. This embryo slowly comes over here and gets embedded at this particular point. This is known as implantation. And where does it take place, children? Uterus. Okay, explain how the embryo gets its nourishment inside the mother's body. What do we have, children? We have placenta. We have placenta for nourishment. How was placenta formed? Again, I have written over here. So, this is what is placenta. Okay, so basically, let's see. As sperm joins with, you don't have to write the entire thing, just for your understanding, okay. The sperm joins with an egg to form the zygote will undergo division. What is it formed? It forms embryo. One stage of embryo, it is known as blastocyst. At this particular point, the blastocyst goes to the uterus and gets attached there, okay. Now, in the blastocyst, no, it looks like this. There is an outer layer like this, which is known as trophoblast, and there is an inner layer like this, okay, which is known as inner cell mass. Now, this inner cell mass, no children, it will form the embryo ahead, okay, it will form the baby. This forms the placenta. The outer layer, which is known as trophoblast, it will contribute to form the placenta. That's what is written over here. So, outer layer of the blastocyst, known as trophoblast, spreads into the uterine lining. It gets into the uterine lining. The trophoblast develops into chorion. You don't need to read all this thing, okay. Small projections called villi grow from the chorion into the uterus, allowing for the nutrient exchange. So, basically, the trophoblast is going to help in the formation of something that is known as villi, and basically slowly the placenta, okay. Now, mother's blood vessels supply nutrients and oxygen without mixing with the fetus blood. The placenta forms as a flat structure made of both maternal and embryonic tissue. So, mother is contributing with the help of her uterine wall. Mother is contributing with the help of her uterine wall. Embryo is contributing with the help of the trophoblast. Will you be able to remember this? So, this point, this, okay, you can start from this point, that is embryo is formed, okay. This embryo basically slowly gives out structure which helps in the formation of placenta. How is placenta formed? With the help of uterine wall and also the embryonic tissue, which is trophoblast. The placenta produces hormones that supports the pregnancy. Apart from that, basically the placenta connects the fetus to the umbilical cord. So, this cord that you see is the umbilical cord. What happens? I say that the mother's blood and the child's blood is never mixed, okay. The mother's blood and the child's blood is never mixed. So, imagine that this is the baby, okay. There is an umbilical cord and this is the placenta, okay. So, umbilical cord basically has the blood vessels of the baby, okay. Now, mother's blood vessels are also going to come over here. Children, diffusion will be happening. Things that are needed by the child will be gotten in there. For example, oxygen gets in there. Things what are not needed gets in over here, that is CO2. So, basically you do diffusion, but the blood is not getting transferred anywhere. So, this is entirely separate from the mother's blood. So, you get oxygen, your oxygen is taken in, and the CO2 what is given out is taken by the placenta, okay, and then it goes to the mother's blood, and from there it goes out. So, there is diffusion, there is no mixing of blood that is happening over here in most cases. So, how will you write the answer? Basically, you can write the answer like this. Embryo, how does it get nourishment? It gets nourishment with the help of the placenta. How is placenta formed? So, once after the embryo is formed and it forms a stage which is known as blastocyst, at this particular point, the outer layer of the embryo will be helping in the formation of placenta from the embryo side. And the uterine wall of the mother will be helping in the formation of placenta from the mother's side. Both of them, they are going to form the placenta, and the placenta is attached to the embryo with the help of something which is known as umbilical cord. With the help of something that is known as umbilical cord, which has the blood vessels. Now, with the help of diffusion, the oxygen from the mother goes to the child's blood vessels, from the CO2 from the child's blood vessel goes to the mother's blood vessel and is moved out. This is how exchange happens. So this is just for your reference to read it rather than not for you to write it. Please, the way I said, that is how you have to write it. Okay, so just say embryo is there, the outer layer of embryo helps in the formation of placenta, the uterine wall of the mother helps in the placenta from the mother's end. Both of them diffuse, they form the placenta and the baby is connected with the help of umbilical cord which has the blood vessels. Now, these blood vessels basically do not transport the blood here and there, there is no mixing happening, but just the diffusion of the blood happens, okay, diffusion of gases happens, oxygen and CO2, oxygen is given to the baby, CO2 is taken from the baby, and the nutrients also same way. As simple as that. Okay. Now write the function of the following part of a bisexual flowers, as in they have both the thing. Okay. Stigma. What does the stigma do? It provides a landing surface for the pollen. Okay. Next thing, pollen tube. Pollen tube basically is responsible for carrying the male gamete to the ovule which has the female gamete. Now anther. Anther is responsible for production of children what? Pollen grains. Okay. Now going ahead, okay, name the future shoot and the future root of the germinating seed. I told you already, but if you remember you can tell me right now. Future shoot, what is it children? It is plumule. Okay. Future root, it is what children? It is the radicle. Fine. Next thing, name the parts of the flower that develop into seed. Seed, it will be ovule. Name the part that develops into fruit. Ovary. Okay. It's a 5-marker question from how do organisms reproduce. Our last question children, our last question, we did it. What do we have? Mention the role of the following organs of human male reproductive system. Testis is responsible for production of hormones, androgens, and for, apart from hormones, it is also responsible for sperm production. Scrotum is responsible for maintaining 2 to 2.5 degrees Celsius lower than the body temperature. Okay. Vas deferens, what does it do? Vas deferens carries the sperm to the urethra. Okay. Or basically the ejaculatory duct that comes before the urethra. Seminal vesicle secretes what children? Seminal plasma rich in fructose that provides energy for sperm. Okay. Now, what is placenta stated function? I already told you. Function is what? Exchange of gases, nutrients, removal of waste. And with this, we have completed the 40 questions of biology children, and now we just have to move ahead with physics, so stay tuned for that. Soumik Sir will be just coming over and then you will be solving all the physics questions as well. I hope that you understood each and every point of it. If you understood all these things, please let me know in the comment section as well, and if you have doubts, I am here to help you out as well. Do not forget. Bye bye children. Take care and do not forget to hydrate yourself.
Hello. So biology session is already over and it's time for physics now. Aishwarya Ma'am has completed chemistry questions. Jasmine Ma'am has completed all the biology questions. Now here I am. I will help you to guide from question number 71 till question number 100, that is 30 questions based on physics concepts. So what all we are going to cover in this? From question number 71 consists of 5 1-mark MCQs, and then question number 72 consists of 5 assertion and reason questions. Question number 73 till question number 82 consists of 10 short answer type 2-mark questions, and question number 83 till question number 92 short answer type 3 marks question, another 10 questions. Then question number 93 from question number 9 till question 95 is case based question, very important, and question number 96 till question number 100 will be 5 long answer type questions. So let's start with the all the questions that we are going to discuss today. So first we are starting with our MCQ. So first MCQ is based on the chapter light. A ray of light is incident on a plane mirror at an angle of incidence of 15 degrees. The deviation of the ray produced by the mirror. Now if you remember the formula, I have already taught you how you can find the angle of deviation. So angle of deviation, say angle D is given by 180 degree minus 2i, right? 180 degrees minus 2i, so 2i is here 15 degrees, so 180 degree minus 30 degrees, that is 150 degrees. Okay, so the correct answer is option D. Just use this formula, 180 degree minus 2i, okay, or 2 theta I. So this is the angle of deviation. Using this formula you can simply calculate the angle of deviation. Now before I move any further, you should note that all the notes, all the lecture notes are available in our website. You just need to click on the link given in the description and you can download all the notes, all the teaching lessons. Also the video links will be provided here. So from there only you can check all the videos, you can download all the PDFs. Now let's move on. Next question. Absolute refractive index of glass and water is 3/2 and 4/3. So absolute refractive index of glass is 3/2 and that of water is 4/3. Now the speed of light in glass is 2 times 10 to the power of 8 meter per second, the speed of light in water. So first of all, let us try to find the speed of we know that ng is basically c divided by speed of light in glass. Similarly, nw is equals to c divided by v water, right? So this is the speed of water, this is the speed of glass. Now what we have to do, we have to find the value. Now let's do one thing, let us find the value of ng divided by nw. Easiest method, ng by nw is 3/2 divided by 4/3. Now this 3 is the bottom, so it will go up, it will become 9, and this 2 will come down, it will become 4 2 is 8, so it will become 8. Now this is the value. So in terms of this, what will happen? Now let me put this value, c by vg divided by c by vw equals to 9/8, right? We have replaced ng by c by vg and nw by c by vw. Now vw is in the bottom, so it will go up, c will cancel, so vw divided by vg is equals to 9/8. So from there we can find vw, vw will be vg is given this, right? 2 times 10 to the power of 8 times 9/8. So it will be 9/4 times 10 to the power of 8 meter per second. So the correct answer is option A, right? You have successfully calculated the speed of light. Now let's move on. Which of the following phenomena of light are involved in the formation of rainbow. Now this is a question based on human eye chapter. Which of the following phenomena of light are involved in the formation of rainbow? So reflection, refraction and dispersion. Refraction, dispersion and total internal reflection. So the correct answer is option B. In while forming a rainbow, we observe there is a refraction. Okay, if you remember the diagram I showed you. So a ray of light coming here. Okay, first they refracted, then they did a total internal reflection. Okay, then they do a dispersion. Okay, so everything happens here. Because of that we observe rainbow. So refraction, dispersion and total internal reflection, everything happens together. Now question number D. A cylindrical conductor of length L and uniform area of cross section A has a resistance R. Another conductor of length 2L and resistance R of the same material has area of cross section. So same material is there. So first R is rho L by A. Okay, now next resistance R is same material, so rho will remain same, but L is 2L. Now what is A dash that we are trying to find? So what we have to do, we have to compare these two values, simple compare. Okay, rho L by A equals to rho 2L by A dash. Rho cancels, LL cancels, so A dash equals to 2A. So this will go up, this will go in this side, so A dash is 2A. Okay, so answer is 2A, correct option. Okay, now let's move on to the next question. Another important question. A cell, a resistor, a key and ammeter are arranged as shown in the given circuit diagrams. The current recorded in the ammeter will be, now if you see, all the circuits are in one or other form are same. Okay, all the circuits has one resistance, one ammeter, one battery, and everything is connected in order, and in each case key is connected properly, so the current is flowing in the each case. So the same in all the cases, because they are all same. It does not matter how you connect the ammeter, how you connect the resistor, as long as they are connected and everything else is same, the current flowing in the circuit will remain same. So we can say that all current is flowing through all the circuit will be same. Okay, or ammeter reading will be same, because current is measured by the ammeter. So whatever current is flowing, ammeter will be showing the same result. So current flowing in the circuit will be same, ammeter will show the same result. Now assertion and reason questions. In assertion and reason questions, there are 4 common options. Each question has 4 common options. What are those common options? Both A and R are true. Okay, and R is correct explanation of the assertion. So this is option A. So in option A and R reason. So in the assertion and reason question, how the question is given? Question is given in a format where we have given one assertion, that is some statement. Okay, you can say that is a statement, and there is a reason for that statement. So first of all, we have to check if individually all the statements are correct. Statements and reason, both of them are correct or not. If both of them are correct, then we have to decide whether reason is actually the explanation of the statement, or it is just another statement which just another independent statement. Okay, so if both of the statements are correct and the reason actually explains the assertion, then we choose correct option. You have to understand this type of questions, very important. And if the reason is not the correct explanation but it is correct, okay, both the statements are individually correct but they are independent of each other, they do not interrelated, okay, in that case we will choose option B. Right, now option C and option D are very simple. If option A is correct, that is assertion is correct, reason is wrong, reason is false, then we will choose correct answer is option C. And if assertion is false but reason is true, then we choose option D. Okay, now let's move on to the questions one by one. electrons move from lower potential to higher potential in a conductor. It's a correct statement. Okay, electrons move from lower potential to a higher potential, and current moves from higher potential to lower potential, which is actually opposite to the direction of flow of electrons. So this is a correct statement. Now what is reason given? That reason is given as a dry cell maintains electric potential difference across the ends of a conductor. Now it is not 100% correct, but we can assume that this is an ideal dry cell, so it maintains a potential difference. Okay, dry cell is much better than other type of cells, okay, because they have some substances which helps it to maintain the potential difference. Okay, if you are using it for a long time, then there will be some potential fluctuation, but a dry cell is very good, very adaptable to maintain the potential difference. That's why you use dry cell, right? So this statement is also individually correct, but this statement has nothing to do with this statement, right? This is an independent statement which is a correct statement. So what we can say, that this is option B. Correct answer is option B. That is, both are true, but reason is not the correct explanation for assertion, right? So the correct answer is option B. Now let's move on to the next question. When a wire is stretched to increase its length to double, its resistivity remains the same. Okay, obviously resistivity remains the same. Resistivity depends on the nature of the material and temperature. So basically it is said that when a wire is stretched to increase its length to double, its resistivity remains the same. Obviously resistance will change, but resistivity only depends on material and temperature. So this is a correct statement, and this is the correct reason for that. Okay, because resistivity is independent of length, resistivity only depends on the material and the temperature of the substance. So option here, correct answer is option A. Okay, so it is a correct reason, and the statement is also correct. So both of them are correct. So this is the correct explanation for that. So you have to understand if the explanation given is actually the explanation here or not. Okay, so this is the correct explanation, so option A is correct. Now next question. Red light signals are used to stop the vehicles are on the road. Obviously red light signals are used. Red signal, if the signal is red, the vehicle stops, they do not move, because there may be some accidents happening, the lanes from other side is open. So red light signals are used to stop the vehicles on the road. So this is a correct statement. Red light scatters the most to be visible from a large distance. Now this statement is actually completely opposite of the actual statement, correct statement. So red light does not scatter, red light scatters the least. So that's why we see the red light from a very far away. So this is actually opposite of the correct statement. This reason is wrong. Okay, so the correct option will be option C. Okay, if in the exam they say the red light scatters the least, okay, to be visible from a large distance, then option A will be correct. You have to be careful. Okay, I have given this question, but if they say that red light scatters the least to be visible from a large distance, then you have to choose option A, because that will be a correct explanation of this. Because red light scatters the least, that's why we use it as a danger signal, so that a vehicle coming from a very large distance, they can see, okay, the signal is red, so I have to slow down, I have to stop, because otherwise there may be some accident happening, right? Now let's move on to the next question. Myopic eye cannot see distant objects. Correct statement, myopic eye cannot see distant objects. Okay, doctors prescribe converging lens to correct myopia. Okay, so myopic eye cannot see distant object, that is assertion, and reason is given that doctors prescribe converging lens to correct myopia. Now even if this statement is correct, obviously, even if the reason, I assume reason is given correct, then also this is not the reason. Okay, because the reason is that when the rays passing through our eye lens, they converge before they even reach the retina. Okay, that is the reason. So if they say that the shape of the eye is elongated, so that the rays passing through the lens, they converge before they even reach retina, then that would have been a correct statement. This is one of the this you can say this is one of the preventive measure, nothing to do with reason. And in this case also, this is not converging, this is diverging. So for myopic eye, we prescribe diverging lens or concave lens. right? So reason is anyway incorrect. Okay, assertion is correct, reason is incorrect. So correct answer is option C. Now let's move on. The incident ray, the reflected ray and the normal drawn at the point of incident all lie on the same plane. Correct statement. That is the law of reflection. Second law of reflection. Law of reflection is not valid for rough surfaces. Now this statement, both of the statements are independent statement, and in fact this is correct obviously, but law of reflection is valid for all the surfaces. It does not matter rough surface or smooth surface. Okay, it does not matter. So this reason is incorrect. Correct answer is option C. So you have seen that most of the cases, assertion and reason based question, we are getting option C as correct, but in your exam maybe option A and option B can also come into play. So don't rely on this information that only option C will be coming, because the questions that we have chosen, we have framed in that case, we found that option C may be a suitable option, right? Now let's move on to the next part of our journey. Objective part is done. 10 objective questions are done. Now let's look into the 2-mark questions. Why do we prefer a convex mirror as a rear view mirrors in cars? So if you see, when you are driving a car, if you are driving a bike, if you are driving a scooter, okay, there are some mirrors fit in that vehicle, so that you can see whatever is coming from behind. That is called rear view. Rear view means whatever is behind you, if you can see them, they are called rear view mirrors. Right? Now let's look into the reason. A convex mirror has a greater field of view as compared to any other mirrors. Thus we can see a large area behind the car using a convex mirror. So convex mirror, they have a large field of view. Okay, they all are formed here and you can see them from your car. What would happen if you are using plane mirror? The field of view will be less. Okay, if you are using a plane mirror, field of view will be not that great. Okay, they will not always, from distant object they will not coincide. Okay, you have to be very close to observe that, but for this you can see, and for a distant object also. So that is why the area that you can see behind you is larger for a convex mirror than compared to a plane mirror. Even and concave mirrors cannot be used at all, because concave mirrors mostly will form real images that you cannot see, okay, unless you place a screen. That is a very difficult thing. So concave mirrors cannot be used. Convex mirrors are easily used. In fact, if you see, if you are living in a society, or if you are living in somewhere where a lot of vehicle passes, then you will see in shopping malls, etc., you will see there are convex mirror fitted on the alleyway as well, so that you can see if some car is coming from the other side or not. Okay, in fact, in big shopping malls, security guards check what all are happening there. If someone is doing shoplifting, someone is stealing from the shop, via this kind of mirrors, as well as CCTV camera. Not only CCTV camera, there are convex mirror are there also, right? Now let's move on. Next question. A concave mirror produces 3 times magnified real image of an object placed at 10 centimeter in front of it. Where is the image located? Okay, so it is said that a concave mirror generally produces real image, and they have mentioned that real image, okay, of an object placed at 10 centimeter in front of it. Where is the image located? So we know that magnification in a mirror is minus V by U, right? Magnification is given here. The magnifier real image 3 times. Real image 3 times means magnification must be minus 3. Why? Because real images are always inverted. So for real image it will be minus 3 minus V, and U is given, so it will be minus 10. U is always negative. U is always negative. So what is V? V is minus 30 centimeter. Very simple questions. Okay, even if they ask you what is the focal length as a part of this question, that also you can find 1 by V plus 1 by U is equal to 1 by F or 1 by minus 30 plus 1 by minus 10 equals to 1 by f, or 30, okay, minus is common, let's take minus common, both of them are minus, now minus 30, 1 plus 3 equals to 1 by f, so minus 4 by 30 equals to 1 by f, or f equals to minus 30 by 4. 30 by 4 means 15, 15 by 2, 15 by 2 means 7.5. Okay, 7.5 centimeter. You can even calculate the focal length if they ask you using the formula. Now, refractive index of diamond with respect to glass is 1.6 and absolute refractive index of glass is 1.5. Find out the absolute refractive index of diamond. So, what is given that refractive index of diamond with respect to glass is 1.6 and absolute refractive index of glass is 1.5. Now, they have asked you to find the absolute refractive index of diamond. Okay. So, very simple question. We know that relative refractive index is given by, first is this medium, which medium refractive index is given, and second is divided by the with respect to which medium it is given, right. So, this is given as 1.6, ND we are trying to find out, and NG is 1.5, right. So, what is ND? 1.6 times 1.5. Now, what is 1.6? You can calculate it like this: 16 by 10 plus 15 by 10. Okay, 5. So 15, 15, 225, 225 plus 16, 1, 3, 4, 2, 4, 1. $240 divided by 100, so which will be equals to 2.40. Okay, 2.40 is the refractive index of the diamond. Simple problem. Now, let's move on. The linear magnification produced by a spherical mirror is +3. Okay, so linear magnification produced by a spherical mirror. Okay, so spherical mirror linear magnification is minus V by U, that is given as 3, +3. Okay, based on this statement, answer the following questions. What is the type of mirror? First of all, what is the linear magnification? +3, so it is greater than 1, right? What is the type of image? +3. Plus means what? It is erect image. Now, erect and magnified image means what type of image? Erect and magnified image means it is a virtual image, right? The image is erect, image is magnified, so it must be virtual image. Now, which mirror produces virtual and magnified image? Only one mirror that produces virtual and enlarged image, so the type of mirror must be concave mirror. Okay, so the type of mirror must be concave mirror because it is producing a virtual and magnified image. Where is the object located? Now, from this formula we know that. Now, if you see this question, they have not given you anything else. Okay, what is the distance, what is the object distance, what is the focal length, nothing is given. So they are asking you not exact location, but what should be the position, right? So it should be between F and P. between the pole and the principal focus, or pole and the focus. Okay, between pole and the focus, if you place the object, then it will produce a magnified and virtual image for a concave mirror, right? List two properties of the image formed other than size and the magnification. Obviously, size and magnification is already covered. So what are other properties? First property, it is a virtual image. Okay, and second is, it is an erect image. Okay, simple answer. So they have mentioned that you cannot mention size, you cannot mention magnification. Okay, so only an option left is it's a virtual image. Okay, you can mention it is located behind the mirror, that is also fine, because it's a virtual image, it is located behind the mirror. Now that is both options should be same, but other thing is it is an erect image. Okay, because there is a plus sign, that means it will be an erect image. Now let's move on. Explain what happens when human eye focuses nearby objects and distant objects. So human eye is a very delicate instrument which can help us to see close objects as well as very distant object, right? So using same eye, without even changing the focal length of our eye by manually, it changes focal length automatically. But if you are using, suppose you are using a camera, okay, camera is a device. For that, if you want to focus something which is far away or focus something which is closer away, you have to focus the lens manually. Okay, or you have to click on the button and then the machine will adjust the focal length after some time. If you are using a mobile camera also, they cannot focus immediately, you have to give it some time. After some time it adjusted the focal length, right? But for human eye, we can immediately adjust our focal length. We do not have to wait, okay, I am looking it for some time, after 5 minutes my focal length is adjusted. It does not happen like that, right? So using same eye, using same lens, our eyes can focus distant objects as well as close objects, which is very magical. So let us look into the reason. When we are looking at objects closer to the eye, the ciliary muscles contract. This increases the curvature of the eye lens. The eye lens then becomes thicker. Consequently, the focal length of the eye lens decreases. This resembles us to see nearby objects clearly. When the muscles are relaxed, the lens becomes thin, thus its focal length increases. This enables us to see the distant object clearly. So first of all, when we are observing distant objects, what happens? Okay, first of all, what is happening when you are looking closer objects? So ciliary muscles contract. Okay, there are muscles, you know that this is eye lens. Okay, there are some muscles attached to this eye lens, so ciliary muscles contract. Okay, because of this contractness, it increases the curvature of the eye lens. The eye lens then becomes thicker. Okay, consequently the focal length of the eye lens decreases. This enables us to see the objects close object clearly. So to see objects which is closer to eye, we need a lens which actually have low focal length, right? Low focal length, then only objects which are closer to our eye can be forming an image to our retina. Okay, the main problem with this is the distance between our eye lens and the retina is fixed. If we can adjust that distance, then would have been easier, but that distance we cannot change. Only thing that we can change is the focal length of the retina in front of us, right? Similarly, when we are observing distant objects, okay, our ciliary muscles gets relaxed. Okay, so that's why when it is very important, it was said that observe something from distant. Okay, if you are observing TVs or watching movies, okay, watch it from a distance so that your eyes don't get strained. Also, sometimes it is advisable to observe sky at night. Why? Because your eyes get relaxed the most when you are observing the sky at night. Okay, so that actually helps the longevity. It reduces the strain on your eye, right? If you are always watching mobile close to your eye, your ciliary muscles are always contracted. Okay, so if your ciliary muscles are always contracted, then it is after some time it will feel weaker and you will lose your natural ability to adjust. Okay, so then you will need a spec like me. Okay, even though I don't have a problem with close, I cannot see the beyond distant objects. That means what type of eye defect I have, please mention in the comment below. Now another important question, why do stars twinkle but not the planets? Okay, we have seen the twinkling of stars but not the planets. Why so? The twinkling of a star is due to atmospheric refraction of starlight. The starlight on entering the Earth's atmosphere undergoes refraction continuously before it reaches the Earth. The apparent position of the star is slightly different from its actual position. Further, the apparent position of the star is not stationary, okay, but keeps changing slightly. So what is happening is that when there is a star, light is coming, a very thin light is coming, and when it is entering the atmosphere, okay, it is suffering some atmospheric refraction, okay, and we don't see the star exactly here, we see the star somewhere here. Okay, now this position, this refraction changes as the layer of the air layer of the Earth does not have a fixed refractive index. It's changing because of the temperature, because of the air current, because of the various reasons, even the dust particle present in the air. So this position is actually fluctuating. Sometimes it is here, sometimes it is here, and it is fluctuating between that. So that's why it appears that the stars are twinkling. Okay, they are not actually twinkling. It happens because of the Earth's atmosphere. If you go out in the space, you will not see stars twinkle. Their stars will appear stationary, right? Now the planets are much closer to the Earth and are thus seen as extended objects. Extended objects like you, me, your book, okay, candle, TV, mobile, they are all extended objects. That's why they don't twinkle, right? They are big enough. Okay, so big enough objects means they have enough light coming from that towards our eye. So even if they change the fluctuation, even if there is a change in the refractive index, there are multiple images forming and all the images are overlapping with each other. So we do not see that twinkling, right? So that's why they do not twinkle. So for extended objects we do not see twinkling. Twinkling happens only for point objects, that is very, very, very far from us. For example, the stars. Now let's move on. Question number 79. A student sitting at the back of the classroom, that is me, okay, cannot read clearly the letters written on the blackboard. What advice will the doctor give it to her? Draw the ray diagram for the corrections for this defect. Okay, so what is the problem that a student facing when he is sitting behind the blackboard and still not see whatever is written on the blackboard? If you are facing this kind of issue, do report to a doctor. They will take care of your eye immediately. So the student has a myopic eye like me. Okay, so myopic eye is something when eye cannot focus light coming from a very distant objects. Okay, so even if their ciliary muscles are relaxed, we will not be able to see distant objects because our eyeballs are elongated. Okay, it forming before even it reaches our retina, so we will not be able to see distant objects. To correct this, the doctor will prescribe appropriate concave lens. So to correct our eye defect, I am wearing a concave lens, and if you wear a concave lens, then this things happening to your eye and you are finally seeing the distant object clearly, because after it passes through that concave lens, okay, it is focusing again at the retina, so you are able to see distant objects. So this is the ray diagram that you have to draw. Okay, does not matter how great is this eye, it will be a rough image is okay, but remember that this lens should be drawn properly and the final ray of light should focus on the retina. Okay, this is a very common question based on your previous year question papers. Okay, after carefully considering all the question papers, these questions are selected. So if you are wondering how we have selected these questions, now filament of an electric lamp draws a current of 0.5 ampere which lights for 2 hours. calculate the charge that flows through the circuit. So what is the current? Current given is 0.5 amperes. What is the time? Time is given 2 hours. Now what is 2 hours? 2 hours means 2 times 60 minute. But we are not satisfied here. We need in seconds. So this is the seconds, right? Now the question is what is the charge that flows through the circuit? So we know that current is basically Q divided by time. So charge is nothing but I times T. I is given 0.5, time is given. Now remember that when you are applying this formula, the time has to be in seconds. Time has to be in seconds, then only you can apply this formula. Now 0.5 to the 1, 6636, 00. This much coulomb. So 3600 coulomb is the total amount of charge that passed through the circuit. Okay. So this is the total amount of charge that has passed through the circuit. This is the answer. If you answer this, you will get 2 marks completely. What is the electrical resistivity? In a series electrical circuit comprising of resistor made up of metallic wire, the ammeter reads 5 ampere. The reading of the ammeter decreases to half when the length of the wire is doubled. Why? So first of all, what is electrical resistivity? So if you want to define electrical resistivity, you have to start with, we know that R is equal to rho L by A. Now, if L equals to 1 meter or 1 unit, okay, A equals to 1 unit, and R equals to 1, sorry, not R cannot be 1 now. Now let's put these values, then we can see that R equals to rho 1 by 1, that is rho. So the resistance of a wire of unit length and unit cross section is called resistivity of the material. So resistivity of a material is defined by the resistance of unit length and unit area of that material. Okay, this is how you can define electrical resistivity. Now, in a series electrical circuit comprising of resistor made up of a metallic wire, the ammeter reads 5 ampere, right? The reading of the ammeter decreases to half when the length of the wire is doubled. Why? So when the length of the wire is doubled, okay, its resistance becomes double as well, because we know that resistance is rho L by A. So if you increase the length to double, okay, the resistance will also become double. So we can write that we know that current is nothing but V by R. So when you increase the resistance, current will decrease. Now if you have decreased the resistance to 2 times, okay, initially it was this, finally it is V by 2R, because resistance has doubled, current must be halved, so that is basically half I. So explanation is that as you increase the length, resistance become double. So if the resistance become double from Ohm's law, we know that current will be halved. Simple. Now let's move on. State Joule's law of heating. How this effect is useful in electric circuits where a fuse is used for safety device. Now you remember that this is very important, electric fuse. Okay, fuse is very not fused, fuse. Okay, fuse is very important concept for you. Okay, let's look into the answer. Joule's law of heating states that the heat produced in a conductor due to the flow of the electric current is directly proportional to the square of the current, that is I, the resistance of the conductor, and the time t for which the current flows. Okay, mathematically it is expressed as H equals to I square RT, right? So heat generated in a circuit is directly proportional to square of the current, okay, then resistance and time. So combined together we can write them as H is equal to I square RT. Fuses in electrical circuits use Joule's law to protect the devices from the excessive current. Due to high current, the heat exceeds the melting point of the fuse wire. It melts and breaks the circuit, stopping the flow of electricity and preventing the damage to the appliances. So fuse is a device which is used to protect the device. Sometimes the current may become higher than expected. Okay, maybe there is a lightning happening, or sometimes some devices malfunctioned. So in that case, what happens is the current is very high amount of current is passing through the circuit. It is called a short circuit. Okay, now once short circuit happened, okay, the current becomes very high in the circuit, which may fry the electrics of the device, okay, electronics of the device. So to prevent that, there is a safety device called a fuse. Okay, the fuse, what it will do, it will it will get heated up and it will burn itself out. So only burning itself, okay, it is saving the entire device. So later on, what you have to do, you have to just change the fuse, then your circuit will work as it is, right? So only the fuse is getting burned, and that burning fuse actually preventing the entire circuit from burning, right? Now let's move on. So if you are wondering where you can get all the questions, all these answers, then you have to visit our website, and all the link will be provided there. You can check all the videos that we have done for your midterm score booster. Also, you can download all the questions, all the PDF files, and study from there. If you are needing for a class notes, you can download it right here. Okay, just go to the description and click on the link, download the file and study from there, and I can guarantee that you will score very well if you completely follow whatever we have done in our channel. Now let's move on. Question number 83. A mirror forms a virtual, erect and diminished image of an object. Okay, identify the type of this mirror. Draw a ray diagram to show the image formation in this case. Okay, so what is the mirror? So mirror forms virtual, erect and diminished image. Okay, so only one type of image mirror can form diminished image, which is virtual. Okay, if you remember, only convex mirror can form diminished and virtual image. Okay, concave mirror, if they are forming virtual images, then it will be magnified, right? Concave mirror can produce real image as well, concave mirror can produce virtual image as well, but concave mirror produces magnified virtual image. We have seen one such question in our previous part, and convex mirror can produce only diminished virtual image, right? So identify this type of mirror. So identify it is a convex mirror. Now they have asked you to draw a ray diagram. I have given you some tips and tricks to draw the ray diagram. If you remember, if you want separate video on that, please mention that in the comment below that, sir, I want a separate video on tips and tricks on how to draw a ray diagram. I will make some video on that. So this is the focal length. You kept an object here. Okay, when the ray is passing parallel to the principal axis, it appears to come from the focus. Right, and this is C, the ray that is going towards the C. It will go along this, come back along this. So this will be the image. Okay, simple ray diagram. Use your scale, use all the methods that you know how to draw the ray diagram. You will get a better image. Okay. This is the mirror. Now let's move on. It is desired to obtain an erect image of an object using a concave mirror of focal length 12 centimeter. Okay, so now we are obtaining erect image using concave mirror. Previously we obtained erect image using convex mirror, right? Now we are trying to get erect image using concave mirror. What should be the range of the object distance in the above cases? Now we know that when the object is placed between the pole of the mirror and the focal length, okay, we obtain a virtual image. Okay, erect image means obviously virtual image. So between 0 to 12 centimeter should be the distance range. Right, now this is a virtual image because it is an erect image. Will the image be smaller or larger than the object? Okay, image will be larger / magnified. Right, as we know that concave mirror forms magnified, erect and virtual image, okay, when the distance is less than focal length. Where will be the image of this object be if it is placed 24 centimeter in front of the mirror? Okay, now the U is given 24 centimeter, focal length is 12 centimeter. So this is nothing but 2f, nothing but r, radius of curvature. So when we place an object at radius of curvature of a mirror, we obtain image exactly at radius of curvature. So we will get the image at 24 centimeter from the mirror. Okay, so we will get the image at 24 centimeter from the mirror. You can use this logic or you can use the formula, whatever you like, does not matter. Okay, if you think the formula will be easier for you to answer, you can use the formula. If you think no, I have understood that it is 2f, okay, I don't need to use the formula to find the distance, then you can use the formula as well. Okay, so 1 by v. When you are using the formula, you should be careful about the sign, okay, because u is negative always, and for concave mirror f is also negative. So 1 by v is equal to 1 by 24 minus 1 by 12. So this is the risk of answering without using sign convention. So 1 minus 2 minus 1 by 24. So v equals to minus 24. Okay. So this is the answer. Now let's move on. A concave lens has a focal length of 15 centimeter. What is the distance should be the object from the lens that be placed so that it forms an image at 10 centimeter from the lens? Also find the magnification produced by the lens. So in this case, what is given is that focal length of concave lens is given 15 centimeter. Okay, what distance should be the object from the lens should be placed? That image is, now if you consider the sign of a concave mirror, concave lens, the sign of focal length is negative, same as concave mirror, right? And image distance is given as 10 centimeter. Now it forms a virtual image, right? Concave lens forms a virtual image, that is the same side, so it will also be minus 10 centimeter. You have to calculate the u. Okay, so we know that 1 by v minus 1 by u equals to 1 by f. This is the lens equation now. Okay, so 1 by v is minus 1 by 10 minus 1 by u equals to minus 1 by 15. Now you can take the negative, you can cancel that, so you will get 1 by 10 plus 1 by u equals to 1 by 15, right? So from there we will get 1 by u equals to 1 by 15 minus 1 by 10, or so what is LCM? LCM will be 30, 15 2s are 30 minus 3 10s are 10, 3 10s are 30, so it will be minus 1 by 30. So u should be minus 30 centimeter. So the object should be placed at a distance 30 centimeter from the lens. Now what is the magnification? Magnification produced by a lens is given by v by u. V is minus 10 and u is minus 30. So 1/3. So magnification is 1/3 of the object, and as you can see from the sign, it is a erect image, right? Now next question, a 200 watt heater has a resistance of about 25 ohm, okay, whereas a 100 watt bulb has a resistance of 500 ohm, okay. When 220 volt is applied on this, then which of the two can carry large current, may be used to the electrical circuit having 10 ampere rating, will be fitted with a 15 ampere electric board and not with a 5 ampere electric board? Justify your answer in each case. So let us check about everything one by one. So the voltage applied is given by 220 volt, right? We know that power is VI. Okay, now it is said that which one will have a large current. Power of this device is given as 2000 watt. Okay, voltage is given 220 volt and current we are trying to find. So current is 2000 divided by 220. Okay, 100 divided by 11, which is almost equals to 9 ampere. Okay, 9 11s are 99, so I can assume it 9 point something will be there. Okay, so it is almost 9. Now what is the second device? Second device has a power 100 watt only. Okay, so now suppose the current passing through this is I dash. So I dash will be 100 divided by 220. So as you can see, the value of current will be very less in this case. So it will be 5 by 11. So 5 by 11 only. So which can carry large current? Okay, heater, water heater. Okay, water heater can carry large current. Okay, and the maximum current is 9 ampere. May be used with an electrical circuit having 1 ampere rating. Which one can be used 1 ampere? 1 ampere means less than, so you can assume this is almost equals to 0.5 ampere, almost. Okay, so 5 by 10. Now we are assuming 5 by 10, so 0.5 ampere rate current. Okay, so 1 ampere, the fuse should be higher than the maximum current that is flowing through that device. Okay, the fuse should be higher than that, right? If the fuse is already lower than that, then every time you are switching on the device, it will fuse, right? It has to have some higher current, so that if the current is passing gets higher than the safest limit, then only it will burn, right? So only you can use this bulb with a 1 ampere rating. So it can be used bulb. Will be fitted with a 15 ampere electric board and not with the 5 ampere electric board. Justify your answer each case. So what will be used? So 9 ampere is the device that can be used with 15 ampere, but it cannot be used with the 5 ampere. So in this case, water heater. So first step I suggest is to find the current, heater and bulb. So you find the current in each device, then the reasons for each steps will be much easier. You can write the reason for this is that the current in the heater is very large, as you can see, so it can carry a large current. Okay, the current through the bulb is 0.5 ampere, so it can be used with a fuse of 0.1 ampere rating, 1.0 ampere rating. Okay, and since the maximum current passing through the heater is 9 ampere, it cannot work with 5 ampere because then it will fuse, but it can easily work with the 15 ampere circuit, right? So you have to mention, if you find the currents correctly, then it will be easier for you to mention all the points, right? Now question number 87, what is electrical resistivity in series electrical circuit? So electrical resistivity I have already answered. in a series electrical circuit comprising of a resistor made up of a metallic wire, the ammeter reads 5 ampere. I think this question is appearing twice. Somehow this question came twice. My mistake, sorry for that. So ammeter decreases to half. I have already mentioned that electrical resistivity is R is equal to rho L by A. Okay, when L equals to 1, A equals to 1, R becomes rho. So you can define the rho from there, and the reading of the ammeter doubles because current is nothing but V by R. Now if you increase the R to 2R, then current will become half, obviously, as resistance is proportional to the length. Okay, if resistance becomes double, length become double, resistance becomes double, so we can write that current will be halved, right? Now next question, B1, B2 and B3 are 3 identical bulbs connected as shown in the figure. When all 3 bulbs glow, a current of 3 ampere is recorded by the ammeter A. So when all the 3 bulbs are glowing, then the current in the circuit is 3 amperes. Okay, now what happens to the glow of the other 2 bulbs when the bulbs B1 get fused? Okay, so let us understand that. So here, what is the type of connection? If you see here, the bulbs are connected such that, okay, these bulbs ends are connected with the other bulbs ends. They are not connected one after another, but along with another, right? So this is called a parallel connection. So in parallel connection, it does not matter if one of the bulb gets fused, but in series connection, if one bulb fused, all the bulbs will stop glowing. But what will happen to this? First of all, you have to mention that B1, B2 and B3 are connected in parallel combination in this case. So if B1 gets fused, B2 and B3 will continue to glow as it is, right? B1 and B2 will continue to glow as it is. What happens to the reading A1, A2 and A3 and A when the bulb B2 gets fused? So what will happen if the bulb B2 gets fused? When the bulb B2 gets fused, okay, the current through A1 and current through A2 will remain the same, right? In parallel combination, individual currents does not change as long as the resistance is constant, voltage is constant, so the individual currents will remain same, but the total current will decrease because the component which is contributing A2 contributed by A2, that will be now no longer be there. So for example, assume that all the bulbs are identical, so we can assume that current at the beginning, what was the current through all the bulbs? So 3 ampere current is passing here, right? So we can easily divide this current equally among all the bulbs because they are all identical. So 1 ampere here, 1 ampere here, 1 ampere here, they are all identical. So initially the current through each bulb will be 1 ampere. Now once this bulb B2 is fused, then this 1 ampere will become 0. This 1 ampere will become 0, but the current through A1 and A3 will remain same, that is 1 ampere, 1 ampere. Now this current will become 2 ampere. So the current through A will decrease, but current through A1 and A1 and A3 will remain same. Current through A2 will become 0. So A2 will become 0, A1 and A2 will remain unchanged, A3 will reduce. Okay. Now what is the third question? How much power is dissipated in the circuit when all 3 bulbs glow together? Very important question, very interesting question. How can you calculate the power? Power is Vodafone Idea. Remember that power is Vodafone Idea. So V is given here in the circuit 4.5 volt. Current is given 3 ampere. Okay, so this is the power 0.5, 13.5 watts. Okay, simply multiply that and you can answer this question, right? Now let's move on. Draw a schematic diagram of a circuit consisting of a battery of 4 1.5 volt cells, a 5 ohm resistor and a 10 ohm resistor. Okay, and a 15 ohm resistor and a plug key. Okay, and all connected in series. Now find the electric current passing through the circuit and potential difference across the 10 ohm resistor when the plug key is closed. So what is the resistors? So they have told you 4 1.5 volt cells. So first let us consider 4 1.5 volt cells. This is one cell, another cell, another cell, another cell. So 1 2 3 4 4 1.5 volt cells, a 5 ohm resistor, okay, a 10 ohm resistor and a 15 ohm resistor. 5 ohm, okay, then 10 ohm and 15 ohm. Okay, and they are connected all in series, right? And there is a key also, sorry, my mistake, there is a key also. Now find the electric current passing through the circuit. So how can we find the current? What is the total voltage? Voltage is 1.5, 1.5, 1.5, 1.5. So all the batteries are series, so total voltage will be adding them 1.5 plus 1.5, or you are adding 4 1.5s. So what we can do? We can write them as multiplying by 4, so that is 6 volts, right? 6 volt is the total voltage. Now what is the total resistance? Total resistance in this case is 5 plus 10 plus 15, so it is 15 plus 15, that is 30 ohm. Okay, so the current in the circuit is V by R, 6 by 30, so 1 by 5 amperes. This is the current in the circuit. 1 by 5 ampere is the current in the circuit. Now potential difference across the 10 ohm. So potential difference across 10 ohm. So potential difference across 10 ohm is very simple. Current times the resistance. Okay, current is 1 by 5, resistance is 10, so it will be 2 volts. 2 volts is the potential difference across 10 ohm, right? Simple question, not so simple question, but not difficult question either. So now question number 90. So after this we will be having only 10 questions left, so please be patient. We are almost 2/3 are done, only 10 questions are left, so let's complete them quickly. Define the term power of accommodation of human eye. Write the name of the part of the eye which plays a major role in the process of accommodation, and explain what happens when human eye focuses nearby objects and distant objects. So power of accommodation: the ability of the eye lens to adjust focal length is called accommodation. However, the focal length of the eye lens cannot be decreased below certain limit. Okay, the ciliary muscles plays a major role in the process of accommodation by adjusting the curvature of the eye lens. So this is very simple question. Okay, I have similarly answered one more question where I have discussed about how can you see different distance and close objects, so you can refer that question. Now a person needs a lens of power minus 4.5D for correction of her vision. What kind of defect in the vision she is suffering from? So first of all, a person needs a lens of power minus 4.5D. So what is the power? It is negative. Okay, and power, we know that power is nothing but 1 by f. That means if the power is negative, that means focal length is also negative. Focal length is negative means what type of lens? Focal length negative, that means it is a concave lens. Okay, so in concave lens focal length is negative. So what defect she has? She is using a concave lens to correct her eye, so that means it should be Yes, myopia. Okay. What is the focal length of the corrective lens? Focal length is nothing but minus 1 by p. But we will use 100 by p because 1 by p is only in meters, 100 by p is in centimeters. So minus, sorry, the sign won't be negative, sign will come negative automatically. So 100 divided by minus 4.5. Okay, so let me multiply minus 200 divided by 9, right? So it will be approximately 22.2. Okay, so approximate focal length is minus 22.2 centimeter. You can do simple division. Okay, I have practiced it. I don't use calculators to do simple calculations, so I can do it faster. If you are doing without calculator, and if you have trained your mind, you can even do this calculation faster than me as well. What is the nature of the corrective lens? It is a concave lens, obviously. Okay, so for example, I am using a minus 5.5D lens in my eye to correct my vision. So I know negative power means myopia. So you can mention that also in the answer, that since the power is negative, that means the eye is myopic eye, and the focal length you can calculate using the formula. Remember that P is equal to 1 by f when f is in meters. F is in meters. To convert it into centimeter, you have to use the factor 100. Okay, so that is one important point of this. Now let's move on. What is the dispersion of white light? State its cause. Draw a diagram. Show dispersion of a beam of white light by a glass prism. So what is the dispersion of white light? The splitting of white light into its constituent colors. Right, this is called dispersion of light. State its cause, because light bends at different angle. So different colors of light bend through different angles. Different colors of light bend through different angles with the respect to the incident ray as they pass through a prism. This causes the dispersion of light. So when the light ray changes a medium, generally, okay, it splits into different colors of light. That is the one thing, because white light consists of 7 colors and each color bend differently. There is a specific reason. We can discuss that in our next class, that is class 12th. You will learn about what is the exact reason behind dispersion. Okay, then and there was a ray diagram also. So you can draw the ray diagram. I guess you already know the ray diagram. Okay, so the ray of light coming here, and the dispersion starts from this surface. You should be careful. Dispersion starts from this surface, and the top ray is red, the lower ray is violet. So it is red, this is violet, and in between some other rays are there. So remember that when you are saying Vib gear starts from the bottom. Okay, Vib gear starts from the bottom. Okay, Vib gear starts from the bottom. This is the secret. If you start writing Vib gear from the top, your answer will be wrong. Okay. Now, highly polished surface such as a mirror reflects most of the light falling on it. Okay, this is a case based question, question marks 4. So the statement given in such questions are generally long, so you have to be patient, you have to be careful, read the question properly. Okay, a highly polished surface of such as a mirror reflects most of the light falling on it. In our daily life, we use two types of mirrors, right? Plane and spherical. the reflecting surface of a spherical mirrors may be curved inwards or outwards. In concave mirrors, reflection takes place from the inner surface, while in convex mirror, reflection takes place from the outer surface. Okay, so basically they have said that in convex mirror it is bent outward, and in concave mirror it is bent inward. Okay, from concave mirror the reflection happens from this surface, for convex mirror reflection happens in this surface. These are the information given. Now let's look into the question. Define the principal axis of concave mirror. So principal axis, what is the principal axis? Principal axis is basically the straight line which passes through the center of the mirror, passes through the focus of the mirror, passes through the center of curvature of the mirror. So together, combine them together into a single system. The principal axis of a mirror is a straight line that passes through the center of the mirror, comma, the focus and the radius of curvature of the mirror. Right? So if you write that, you will get a full marks. Now a ray of light is incident on a concave mirror parallel to its principal axis. If the ray after reflection from the mirror passes through the principal axis from a point at a distance of 10 centimeter from the pole of the mirror, find the radius of the curvature of the mirror. Okay, this is also 1 mark. So these are the marks given. Okay, so I have taken the question from previous year, and also I have tried to give you the marks. This is a 2 mark question. So they have said that a ray parallel to the principal axis, okay, after reflection from the mirror, okay, it passes through a point 10 centimeter from the pole. So this distance is 10 centimeter. Now we know that from the principal rays that a ray parallel to the principal axis after reflection from the mirror either passes through the focus or it appears to come from the focus. For concave mirror it is passing through the focus, for convex mirror it is appearing to come from the focus, right. So this is the focal length. So basically the focal length, indirectly the focal length is given. Okay, so radius of curvature is what? Radius of curvature is 2f. So 20 centimeter is the answer. So simple, you can write 20 centimeter. Okay. So an object is placed at a distance of 10 centimeter from the pole of the convex mirror of focal length 15 centimeter. So what all are given is the distance is given. Object distance, we know that object distance is minus 10. Object distance always negative. For a convex mirror, okay, focal length is positive. V we are trying to find. Focal length is given, convex mirror of focal length 15 centimeter. So focal length is given 15 centimeter. We know that 1 by V plus 1 by U equals to 1 by F. Okay, so 1 by V equals to minus let me clean this up quickly. One by V plus 1 by U equals to 1 by F. Okay, 1 by V plus 1 by minus 10 equals to 1 by 15. So 1 by V is 1 by 15 plus 1 by 10, right. 32 plus 3, so that is 5 by 30. Okay, so V equals to 30 by 5, that is 6 centimeter. Be careful while solving this. You have to be careful about the formula. You have to be careful about the sign convention. If you are using incorrect sign convention, if you are using, suppose this is given for mirror, if you are using the lens equation, your answer will be wrong. So you have to be careful about the formula. You have to be careful about the equation that you are using. Now let's move on. Study the data given below showing the focal length of 3 concave mirrors A, B and C and the respective distances of the object placed in front of the mirror. So first of all, we can see that mirror A is focal length is 20 centimeter, object distance is 45 centimeter. So this distance is basically greater than the radius of curvature, Okay, Now next mirror B is focal length 15 centimeter, object distance is 30, so it is equals to 2r. Okay, sorry, equals to r, or 2f equals to r, right? Next is mirror C, focal length is 30, object distance is 20, so this is less than f. Okay, the object distance is less than f. Now in which case the mirror will form a diminished image of the object? Justify your answer. So the mirror will form a diminished object only when the object is far away from the mirror, far away from the radius of curvature of the mirror. So you will get a diminished image when the object is placed at 45 centimeters. So case answer for 1 is option A, mirror A. List 2 properties of the image formed in case 2. So 2 properties of the image formed in the case 2 is given by, 2 properties are given by, number 1 real, number 2 same size as the object. Right, it is placed at 2r, placed at 2f, or placed exactly at the radius of curvature. So if your object is placed at the radius of curvature, we obtain real image, we obtain inverted image, also you obtain the same size as the object. So everything you can mention. Okay, real and inverted you can mention as 1 point. Okay, and same size as the object. So magnification is 1 also, that you can mention. What is the nature and size of the image formed by mirror C? Draw a ray diagram to justify your answer. So the nature is, because it is less than f, it is virtual and erect, and I have drawn the ray diagram, but still I am drawing once more shortcut. Quickly I am drawing a ray diagram, if you please don't mind my drawing. This is focal length, this is radius of curvature. So any ray parallel to the axis after passing through this will pass through the focus, and anything passing through the radius of curvature, they will pass through this. Now they will not cross in this direction, they will intersect somewhere behind. Okay, so virtual, erect and magnified. Okay, so these are the property of this image, and this is the ray diagram that you need to draw show the image. Now let's move on. Another case based question. On the basis of the circuit, okay, so here they have given this circuit and they have asked you to solve this problem. So the circuit itself is the case. On the basis of the circuit, answer the following questions. Find the value of total resistance between the points A and B. Okay, find the resistance between the points B and C. Okay, so first of all, what is the resistance between the point A and B? So here 4, 6 and 16 are connected in series, right? So these two are connected in series. So simply to find the resistance between A and B, say RAB, we just need to add them, 4 plus 6 plus 16, that is 26 ohm. Okay, so 26 ohm is the resistance between A and B. So R AB is 26 ohm, right? Now let me calculate RBC. Now while we are finding RBC, here you see that two 8 ohm resistances are connected in parallel. So two 8 ohm resistances are connected in parallel. I showed you how can you calculate two resistances connected in parallel. That is nothing but R1 times R2 divided by R1 plus R2. In fact, if both the resistances are equal, then it's simply R by 2. Okay, if both the resistances are equal, then you can even simplify this further by writing this as R by 2. Okay, so this is the RBC. So 8 divided by 2, that is 4 ohm. 4 ohm is the resistance of RBC. Now what else was given? So first question is 26. This is 4. If in case they are not given equal, then you can use I1 I2 R1 R2 divided by R1 plus R2. Okay, but since they are given equal, so this is very simple. Okay, that is 4 ohm. Now calculate the current drawn from the battery when the key is closed. When the key is closed, everything is connected. Now RAB is 26, RBC is 4. So what is the total resistance? Total resistance is RAB plus RBC, right? So total resistance is 26 plus 4, that is 30 ohm. Okay, and what is the voltage given? Voltage given is 6 volt. So what is the current? Current will be V by R, that is 6 divided by 30, that is 1 by 5 amperes. Okay, or you can write this as 0.2 amperes. Right? Now what else? There is a OR question also. Okay, so if this question was given, as long as also there is an OR question for option C is given. Okay, for only option C there is a option given. The above circuit 16 ohm resistor or the parallel combination of the two resistors of 8 ohm, which one of the two will have the more potential difference across the two ends? Justify your answer. So they have asked you to find the potential difference across these two resistances or between these two resistances. Now current is same. Current, what is the current? We have calculated the current in the previous case. Current is 0.2 ampere. Okay, so the voltage drop across 16 ohm resistance will be, voltage drop across 16 ohm, okay, is 16 times 0.2, that is 3.2. Voltage drop across any parallel of the resistor, any parallel of the resistor means RBC. Okay, so I am writing BBC. So resistance is 4. So obviously as you can see, the voltage across 16 ohm is more. Now you can answer this qualitatively without doing mathematics, because the the resistance of 16 ohm is much higher compared to the resistance which are in parallel, that is 4 ohm only. So the voltage will be 4 times across 16 ohm compared to 4 ohm. Okay, that is also a qualitative answer that is possible, because if you are using this method, that means you have already solved part 1 of C. If you have already answered part 1 of C, no need to answer part B, which is an optional part, right? Now let's move on. Now let's look into 5 mark questions. Okay, this is also sometimes, now there is a very less demarcation between case based questions and 5 mark question in physics. Okay, even though there are some in some cases might be they will ask you what is the doctor prescribes, but that is also, in that case also, it is a 3 mark question only. So the demarcation of case based 4 marks and 5 marks and 3 mark questions in physics sometimes is very blurry. Okay, that is very quite clear in chemistry and biology topics. Okay, but in physics there are not so much case based. Okay, there are mostly some situation based or some circuit based question. Okay, if you check all the previous year questions, you will hardly found any proper case based questions. Okay, a student focused the image of a candle flame on a white screen. This is also a case based question, but this came as 5 mark question, not 4 mark question, right? A student focused the image of a candle flame on a white screen using a convex lens. He noted down the position of the candle screen and the lens under the position of the candle, 12 centimeter. position of the convex lens 15 centimeter, position of the screen 88 centimeter. Okay, so what is the focal length of the convex lens? So they have given you that for object placed at 12 centimeter, you are getting the image at 88 centimeter, whereas the mirror the lens is placed at 15 centimeter. So first of all, what you have to do, you have to find the object distance u. Okay, u is the magnitude of u. Okay, magnitude of u is position of the lens minus position of the object, so that is 38 centimeter. Okay, and v equals to position of the image minus position of the lens, that is 38 centimeter. So both of them are 38 centimeter. So the focal length will obviously be half of that. You can use the formula, no problem. 1 by v minus 1 by u equals to 1 by f. Now we know that in this case real image is formed, so v is positive, u is negative because u is always negative. So 1 by f equals to 2 by 38, or f equals to 19 centimeter. Okay, you can simply answer that based on logic as well, because the object distance and image distance are equal. That means object must be placed at 2f. So only when object is placed at 2f, we obtain the image exactly at 2f. Okay, but on the other side of the lens, in case of lens, it is on the other side. Okay, so if u is negative, v becomes positive, but for mirror, both of them are happening on the same side, right, for 2f distance, that is r distance. Same is valid for lens, but in this case the mirror is image is object on the other side. Where will be the image formed if he shifts the candle towards the lens at a position of 31 centimeter? Now the object is moved to 31 centimeter and they have asked you where the image will be formed. Now what is the object distance? Let me find the u first. U is 50 minus 31, that is 19 centimeter. So the focal length is 19 centimeter. You are placing the object at the focal length. What will happen? There will be an image formed at infinity. So the image will be formed at infinity. Okay, the image will be formed at infinity. So if you place the object exactly at focus, you will get the image formed at infinity. Now question number 3 of that, what will be the nature of the image formed if he further shifts the candle towards the lens? So now the distance is less than focal length. So less than focal length means image will be, for answer for part number 3, image will be virtual. Virtual. Okay, what else? It will be erect, obviously, as we know, and what else? It will be enlarged or magnified. Okay, so virtual, erect and magnified, these are the nature. So now they have only asked you the nature, so virtual is correct answer. You can mention these also. Draw a ray diagram to show the formation of image in the previous case. Now this is a lens, not mirror. This is little tricky. Now this is a virtual image, that's why I have used dotted line to show the image. Okay, so this is AB, suppose this is AB, then this will be A dash, B dash, right? So this is the ray diagram for this case. Simple. But whenever you are drawing ray diagram, don't forget to show arrows. Without arrows, ray diagrams has no meaning. Now, question number 97. So, slowly we are moving towards the end. An object of height 5 centimeter is placed at 15 centimeter in front of a concave mirror of focal length 10 centimeter. At what distance from the mirror should a screen be placed so that a focused image is obtained on it? Find the nature and height of the image. So, the object of height. So, height of the object is given 5 centimeter, okay, in front of a concave mirror. So, U is given minus 15 centimeter, okay. Focal length of the concave mirror, is also negative, so minus 10 centimeter, okay. What distance from the mirror should be screen be placed so that a focused image is obtained? So, first of all, we have to find the V. So, 1 by V plus 1 by U equals to 1 by F, okay. So, 1 by V is, we don't know the V, plus U is minus 15 and F is minus 1 by 10. So, it will go the other side. So, 1 by V equals to 1 by 15 minus 1 by 10, right. Minus 1 by 15 will become positive. So, the LCM is 30. So, 15 is 30 minus 3, 30 is 30, and then it will become minus 1 by 30. So, V will be minus 30. So, we have calculated V. Where we have to place the image, you have to place the screen at a distance 30 centimeter from the mirror. So, we have to place the screen at 30 centimeter from the mirror. Now, find the nature and the height of the image. So, nature is obviously it's a real image because it's a concave mirror, and because V is negative, it's a real image, right. So, nature is real because V is negative. Now, magnification is given by minus V by U. So, magnification is minus 30 divided by minus 15. So, magnification is minus 2, sorry. So, magnification is minus 2. So, height will be 5 times minus 2, that is minus 10 centimeter. Now, what is the significance of the negative sign? Negative sign indicates it's a inverted image, okay. So, the image will be downwards, that is why minus 10 centimeter. You can keep the negative sign in this case, you can remove the negative sign, does not matter as long as you are explaining that the image is, final image is inverted, that is why this sign is negative, okay. This is the final height, right. Now, let us move on. Question number 98. A current of 1 ampere flows in a series circuit containing an electric lamp and a conductor of 5 ohm when connected to a 10 volt battery. So, a current of 1 ampere flows in a series circuit containing an electric lamp and a conductor of 5 ohm. So, you have a lamp and a conductor of 5 ohm. When you connect this to a 10 volt battery, a current of 1 ampere flows through it, right. So, 1 ampere flows through it. Calculate the resistance of the lamp. So, suppose the resistance of the lamp is X ohm. Now, they are connected in series. So, what is the total resistance? Total resistance must be X plus 5. In series resistance, they are getting added, right. So, we know that V equals to IR. So, V is 10, I is given 1, R is X plus 5. Okay. Treat physics numericals as algebraic problems. Okay. Don't worry too much. Only the formula are based on certain laws. Otherwise, it is nothing but algebra. If you start thinking this is completely different, then you will be in trouble. So, X is 5 ohm. Okay. Simple algebraic method, right? What change in current flowing through 5 conductor and the potential difference across the lamp will take place? Now, okay, calculate the resistance of the electric lamp. Now, if a resistance of 10 ohm, this part is done, up to this part is done, so electric lamp has a resistance of 5 ohm. Okay, now if a resistance of 10 ohm is connected in parallel with this series combination, so a 10 ohm is connected in parallel. Now next is, the lamp was already there, okay, resistance was already there, and now you have, what you have done, you have connected a 10 ohm resistance in parallel. Okay, what change, if any, in current flowing through 5 ohm conductor and the potential difference across the lamp will take place? Give reason. So initially the entire 10 volt is acting along this, right? Now 10 volt will be acting along this as well as this. Now if you consider only the current, nothing will change, because in parallel combination each component is getting the same potential. So current and potential across the 5 ohm resistor, even the lamp, will not change because it is connected in parallel. That's why when we are suppose we have one light, I want to install one more light, then I will install that light in parallel with the existing light so that both light glows equally brightly, right? So since it is connected in parallel, the total current in the circuit will change, obviously, but current through this individual components will remain unchanged. That is very, very important. Total current in the circuit will change, total power consumed in the circuit will change, but the power current through individual components will remain constant because it is connected in parallel. So in parallel the voltage across each component remains constant, so that's what you have to mention, that in parallel combination potential across individual components remain constant, so current through 5 ohm ampere will remain same. Similarly potential difference across the lamp will remain same. Okay, now question number 99. So this is 99 and then we will score a century, one more run away from the century. How many of you are still with us? Mention in the comment below if you are still watching question number 99. Find out the following in the electric circuit given in the figure. So what all we have to find out? Effective resistance of two 8 ohm resistances in parallel. So I have already told you how we can calculate the resistances in parallel, so you can use that formula. Now since these two resistances are equal, so the equivalent resistance is 8 by 2 is equal to 4 ohm. We have already done that. If they are not equal, then R1 R2 divided by R1 plus R2, right? Current flowing through 4 ohm resistor. So they have also asked you to find the current through 4 ohm resistor. Okay, so to find the current through 4 ohm resistor, we have to find the current through the entire circuit, because this is the connected to the entire circuit. So what is the total resistance? Now so far I think you have understood how we can find the total resistance. Total resistance, check all the resistances. So this is a 4 ohm, this is equivalent to 4 ohm, and they are connected in series. So total resistance is 4 ohm plus the equivalent 4 ohm, so that is 8 ohm. So 8 ohm is the total resistance of the circuit. Now voltage applied is 8 ohm, so current in the circuit is V by R, that is V is 8 volt divided by R is 8, so 1 ampere is the current through the circuit. Now whatever current in the circuit, the same current will pass through 4 ohm, so current through 4 ohm will also be 1 ampere because it is connected in series, right? Now potential difference across 4 ohm resistance. Potential difference across 4 ohm resistance, simple, current through 4 ohm times its resistance. Simple, current is 1 ampere, resistance is 4, so 4 volt is the potential across 4 ohm resistance. Power dissipated in the 4 ohm resistor. Power dissipated in 4 ohm resistor, power is nothing but, you can use any formula. I am using I square R, you can use VI, whatever you want, you can use the same, no problem. let me use the common Vodafone Idea formula. So Vodafone is, we have calculated 4, I we have calculated 1. So the 4 watt is the power dissipated in this 4 ohm resistor. Difference in ampere readings, if any. So you should remember that both these amperes are connected in series in the main circuit. If you see, both the current ammeters are connected in series in the main circuit. Okay, if this ampere was connected here, then there would have been some difference, but both are connected. One is the starting of the battery, one is the ending of the battery. It does not matter where the ammeter is connected, as long as it is connected in the complete circuit. So both the readings will be same. So you have to mention that current through A1 should be equals to current through A2, and that should be equals to 1 ampere. Okay, now let's move on. Last question. Century, you are going to score century, you are going to make a history. Okay, what type of circuits, series or parallel, should be used when you have operate different electrical gadgets in your house? List 2 reasons for your answer. So what is the which type of circuits you will use? Obviously, to get use all the gadgets in your house, you have to use parallel combination. I have told you multiple reasons throughout this journey. I told you that in parallel combination, each component gets the required potential difference. Okay, even if one of the components stops working, the other components keep on continue to work. So these are the 2 reasons. Okay, each component will get the equal potential, so they can function independently. Secondly, if any one of them is fused, then rest of the devices does not get affected through that. Okay, so they will still continue to work. So these are the 2 reasons that you have to mention. Now for question number 2, find the current flowing through the following electric circuit. So they have simply asked you find the current, no component, nothing. So they have asked you to find the current. Simple question. Okay, so this is a 3 ohm resistance. Okay, and in between there is a part. So this part is complicated. This part we have to solve. So how can we solve that? These 2 resistors, if you see, these 2 resistors are connected one after another. So these are basically connected in series. So 6 ohm plus 6 ohm connected in series. So the equivalent resistance will be 12 ohm in this case. So 12 ohm is the equivalent resistance for these 2 resistors. Now 12 ohm and 6 ohm, they are connected in parallel. So 2 resistors are connected in parallel. So the equivalent resistance is 6 times 12 divided by 6 plus 12. So I was telling you this formula from the beginning. You can directly use. Okay, and you can find Req, not 1 by Rq. This is the direct formula where you can calculate Req in one step. So it is 18 6 times 12. Okay, so 3 2 is 6, 3 6 is 18, 2, so it is 4 ohm. So now we can replace this entire combination using a resistance of 4 ohm. So the entire resistance can be replaced using a 4 ohm. So the total resistance is 3 ohm plus 4 ohm plus 3 ohm, that is 10 ohm, right? So 3 ohm plus 4 ohm plus 3 ohm is the total resistance of the circuit, and the voltage given is 4.5 volt. So current is basically V divided by total resistance. So V is 4.5 divided by 10, that is 0.45 ampere. So 0.45 ampere is the current in the circuit. So we have completed all 100 questions, right? Congratulations to everyone who are sitting through all the 100 questions and want to score a beautiful marks. If you have followed all the questions, I can guarantee you will score a very good score in your upcoming midterm exam. But if you are wondering what you want to do after your class 10th is over, then worry not. Diksha is presenting Diksha Scholastic Aptitude Test or DSAT. Here you can take a class of 1 hour after the class is over. They will give you a one question paper that you have to answer based on the marks that you are getting. There will be a counselor available to you for absolutely free of cost. So that counselor will give you a free counseling that what you should try, whether you should go for engineering courses, whether you are suitable for medical courses, or you are suitable for any other courses rather than engineering and medical. They will give you an honest opinion. So once based on your score, if you take admission in one of the Diksha's colleges, you can get up to 90% scholarship on your PU2 fees. So why are you waiting for? Go and register your name for DSAT now, right now. Also, if you are thinking how you can get all the question papers, all the discussions, all the notes, you can go to our website, click on the link below, and then you can get and download all the questions. So I hope that you have enjoyed our midterm score booster series. So I am Soumik. I am closing this lecture today, and hopefully we will be seeing you soon with new series for your final board exam very soon. Till then, tata, goodbye, and if you have not yet subscribed to our channel, don't forget to subscribe to our channel. Click on the like button. See you soon.